So recently my father passed away, and I just wrote down a few things just because it's easier to get through. And so this was actually as she contacted me about the keynote. And so this was at the end of July. And it was an interesting, so I was his primary caregiver. I had moved back to Minnesota to help him. He had been diagnosed with dementia. And the last kind of 6 months have been just day to day trying to help him have a good day. That is the goal. Every day, just what can I do to help him have a good day? And as he passed away, there was a nice recovery of him as a person and his life and the arc of his life. And I started to think about his work and his life. And he was a pioneer in the work on cancer. And that historical trajectory and how we are on a trajectory in the space of neurotechnology. And so I'll just start by sharing kind of his trajectory and take it from there. So recently my father passed away. I was close to him and cared for him at the end of his life. As I reflected on his life, I found his career journey followed an arc of history and had interesting parallels to my own. He was born in Calcutta, India in 1939, graduated from National Medical College, and in 1964, he immigrated to the United States. West Side Story with Rita Moreno had just won the Oscar for Best Picture, and my father saw this movie as something of a primer for upcoming immigration to the United States. His port of entry was New York City, and he came to this country with a few dollars in his pocket. He hoisted his suitcase on his shoulder because the handle had broken, and with images of dancing gays and gun veterans from West Side Story on his mind, he entered the U.S. with a mix of trepidation and excitement. The U.S. was at the height of the Civil Rights Movement in the 1960s. Immigration and Nationality Act of 1965 came about the year after my father arrived. The act formally removed discrimination against many immigrants, including Asian immigrants, from immigration policy. However, in 1964, the my father would have been one of only a few hundred Indians allowed in the U.S., largely for educational purposes. A couple years later, he moved to Buffalo, New York, where he started his specialization in oncology and community-based cancer research at Roswell Park Medical Center at what turned out to be a seminal time. In the 1950s and 60s, the frontiers of medicine were infectious disease, measles, smallpox, polio. The vaccinations and antibiotics were eradicating these diseases, and focus was turning to cancer as the next frontier. Post-World War II, the U.S. was investing in scientific progress as a strategic imperative and driving funding into research to push forward new frontiers. It isn't lost on me that this is the framework that is under attack and being dismantled today. In the 1960s, the foundations of clinical studies as we know them today were created: informed consent, clinical study protocols, institutional review. And my father was at a preeminent institution at Buffalo, New York, where he was learning to drive research on this frontier. In 1971, Richard Nixon declared a law on cancer, which opened funding opportunities in the U.S. There was a small clinic in central Wisconsin, Restorative clinic, that was growing its research capabilities. They were successfully whaling grants and had support from the government to grow research related to the local farming community. And their father saw an opportunity to develop a research program and create change for a population in need. This was the beginning of his illustrious 35-plus year career leading clinical research for the community clinical oncology program. And most of us, even as lay people, to develop effective treatments for many cancers. My father's work led advances in methods to identify treatment course based on bench testing of tissue samples, but he also planted the seeds in the next generation for scientific frontiers. And I share a couple of the historical aspects that really framed up his career, and there's, of course, my history as well. And amongst his things, I found this photograph. It's the 1967 local residence in Buffalo, New York. The gentleman at the bottom right there, he's a very famous scientist at the arc of cancer. Like father was there in the back, and you can actually see quite a diverse residency group there at that time. So I use that framing to think about us, and why is this the neurotech moment? What is happening in our sphere, and what has enabled this moment? And so I kind of framed this in three pillars. And one of the key pillars is government programs, which seems very relevant to where we are here today. And I did a little bit of research that many of you may know about when the Blueprint program came about. The Blueprint program started really not that long ago, in 2003. And the recognition was about the need for cross-disciplinary collaboration in this industry and the importance of that. It started with some foundational research work. The 2009 Human Connectome project really was enabling that table of, hey, can we create some open source content that would really be a good framing and foundational work for the space? The Neuro Initiative recognized that maybe private-public partnerships would be beneficial. If innovators could connect with industry, could that be a good partnership to enable innovation? And the Blueprint MedTech, as Sri mentioned, this being the third year, came about in 2002, and the goal has been to enable translation, to recognize that to get these innovations out to millions of people, we really need to get to that translation state, and how do we do that together? Another key change in the space that is enabled at this moment is what I like to call enabling technologies. And so what do I mean by that? A lot of times when we think about technologies, we can kind of dream and think, oh, we can do low power, and we can do high bandwidth communication, and we can make devices small, and we can do all of that in one. But really understanding what exists there, what's been proven. If you're working on implantable devices, what kinds of electronics are safe and reliable for implants, and how much power consumption can there be? Those are the enabling technologies. And if you find, as you're concepting, that you have to dive deeper and deeper and create more of that first principles on this physics level stuff, recognizing where you're spending your energy and your efforts versus where you're building upon something that already exists is really important. In my time in the neurotechnology space, the early neurostimulators, so the first essential tremor deep brain stimulation approval was in 1998. For Parkinson's, it was, I believe, 2002. In those early days, the integrated circuits on those implantable devices were fairly simple. They allowed for pretty simple waveforms. The communication methods for those were near-field communication, so they were coil-based communication, which meant that you couldn't get information out of a device unless the patient went to see the clinician. Nowadays, we have this expectation that things are very interconnected, and we can stream things, or we can monitor things while somebody's sleeping. But at that time, you could only get that data and download it when, for many years, when the patient went into the clinic. There's been a lot of progress in biomaterials and understanding what is safety, what is safe, and how do you measure that as well. There are multiple aspects of biomaterials. There's compatibility and making sure material isn't toxic to the body, but also the stability of that material over time, especially if you're working in the chronic implant space. So how do you select that material so it's not deteriorating in a way that's detrimental to the body or causes problems down the line, as well as detrimental to the function of the device, especially in an electrically functional device that's implanted. I mentioned connectivity a little bit. That connectivity has changed so much, how you interconnect with each other, and the standardization of things like Bluetooth-led energy. When I started my career electronic, Bluetooth-led energy didn't exist as a standard. We were working on distance communication and moving away from that near-field coil-based communication system. That the standardization of Bluetooth-led energy that enabled communication systems at the kinds of power that the implantable batteries could accept. There's been innovation in battery technology and powering and the size of those batteries. So all those things have enabled connectivity, Wi-Fi, and then as well as the ecosystem of our tablets and our iPhones and how we interconnect with the world. Not only is it our tablets and our iPhones, but we can turn on and off the switches. We can change our thermostat with connectivity. And then, of course, machine learning, artificial intelligence, and the explosion that we've seen. How are we going to connect our devices, enable our devices with an ecosystem of AI or machine learning, and where is that going to take us? Those are all innovations that have really stood up in the last 20 years. And the final pillar also is talent and the development of talent. When I did my electrical engineering degree back in the late 90s at University of Minnesota and UW-Madison, the biomedical engineering programs didn't exist. Those programs came about in 1998, 1999. There were, of course, other programs that have been around for a lot longer. But it's taken time to figure out these cross-disciplinary intersections that have formed new disciplines. I've come to appreciate the neuroengineering talent in the spaces that I work in. And when I was a student, when people told me, well, biomedical engineering is an application, we go to mechanical engineering, electrical engineering, and then we can always apply to biomedical engineering. There were some truths in that. I certainly found teams that are very cross-disciplinary, and I need electrical engineers, mechanical engineers, material scientists, chemists, like all different disciplines. But the neuroengineering talent is really developing people at the intersection of electrical engineering, computer science, and neuroscience. And there is a uniqueness in these cross-disciplinary spaces where the universities have been able to develop programs and develop talent that has been able to drive us into new frontiers. And these are the things that form our foundational neurotechnology. When we think about the last 20 years and the arc of history that we've been living, why is this moment now? The computational power is there. The electrical capabilities are there. The governmental programs are there. The talent is there. And this creates the unique ecosystem in which we're working with that has been proliferating our startups and our suppliers and and creating a foundation for neurotechnology. And of course, there's funding that has to go with that.
Yes,
Funding is interesting in this space because there are some applications, in particular, if they say BCI, that get lots and lots of funding. But for most of us in the startup space, we're working with incremental amounts of funding and showing proof principles and going along a systematic journey. This was taken from the MedTech strategist, just published in the July-August timeframe. You could read into it and say, maybe funding was down the second quarter of 2025. But you can also take a look and see how much that funding is distributed across many different organizations. So funding really drives what you can do and how do you get to that next proof of principle that drives your next milestone and your next round of funding. So I'll pivot a little bit from talking about the history to talking a little bit about an approach. And I really like this tree analogy. I talked about those enabling technologies, but it's not just technologies that enable what you are trying to do. And I call these the enabling roots. Are you building a new surgical procedure, bringing in almost a different specialty in the clinician domain? Or are you building upon something that exists? One of the things about Synchron that was great was we were building upon an infrastructure that existed in angio suites of deploying devices in the blood vessels of the brain. You can see right there, that's an approach that has some enabling roots. It has a physician base that's excited about what we're doing. The neurologic approach, of course, is quite different, where you're implanting through surgical robotics. They have to take on building that infrastructure, give them the leadership, maybe they're building hospitals. I don't even know, but they can think about that with the funding levels that they have. So you have to balance where are you at and what are you building upon and what are your enabling roots. Do you have existing clinical proof points that you can leverage to do your clinical study? Do you have things that will develop into endpoints? Do you have existing reimbursement structure that you can piggyback on? So what is the framing of your enabling roots that ground your startup or your technology to be successful?
Yes, sir.
And that's what's in the roots, you kind of develop a strong trunk. So the opposite of this, if I were to show an opposite program, would be to see lots of it as happening. So if your work is really separated, you'd be seeing a lot of separate saplings and they wouldn't work together as a company. But that core of your trunk being really strong gives you a platform. So when we talk about platform technologies, how is that is being leveraged as a core that then enables your branches and are used to talk about as future applications. Many of you are familiar probably with the biodesign process. I just picked up the Stanford biodesign process just as an example. Of course, there will be talks about needs screening and the importance of starting and understanding the user need. It can be tempting in this space to start kind of bottoms up and pick up a technology and be like, what can I do with this? And that does happen, but understanding that need and what you're going after is really important. And then the invention phase is about concepts. But I'll talk a bit more about the implementation phase and their strategy development. Because as you develop concepts, that can be pretty unconstrained. But if you're going to kind of frame it into this tree concept and really understand where am I leveraging in this pretty complex environment, complex space, where am I leveraging things that already exist and building upon them? And where am I going to spend my energy building anew? That's really your strategy of implementation. And choosing the right partners and choosing the right strategy are really key to your success. And I would say, don't just think one step ahead, but at least try to think 2 steps ahead. If you try to kind of eat the whole enchilada, that's also not a great thing. But if you can kind of think, all right, well, if I make these choices today, then 2 steps ahead, how do these impact me? And that kind of gets to cost of change as well. I don't know if you guys are familiar with cost of change. That as you go through the process of creating the prototypes and getting further and further into a design controls or regulated process, the less freedom that you have to change. Where design freedom is going down because your project costs are going up. And if you're working in particular in an implantable space, which is my expertise, that implantable device in particular will be harder and harder to change as you go down this path. So how do you solidify things that require significant cost to change them, and that keep your options open so that you can adapt as you learn? This is a space that's very iterative, right? So you can't really get your true user needs and your design inputs until sometimes you start implanting. And then you start to learn, and you have to be able to change and iterate. But how do you do that smartly so that you're not incurring this huge cost of change curve, which can impact your timelines? One of the questions that comes up very often is, well, how does tech move so fast? Like, we're kind of go, and there's kind of a perception that, like, well, med tech is slow and tech is fast. Like, why is that? Is it because it's a regulated space? One of the reasons for that, though, is a lot of tech is largely software. And in the software space, that cost of change curve that I was just showing in the previous slide just isn't as impactful. They can change a lot. The word is agile, right? They can group their things together. They can create backlogs. They can, they call it refactoring, where they can adapt to this change pretty quickly. Software, even in the medtech space, though, is to be evaluated for risk and for harm. So within software change in the medtech space, I would generally say probably takes at least somewhat longer than in the tech space. You can, however, be smart. You can architect your software to enable that change and plan for that change. You can architect it so that you are targeting off of, okay, these are the things that create risk, but these are some things that I can change that don't create risk. This is particularly interesting in this brain-computer interface space, where mostly we're taking sensing information and operating digital devices. You're not introducing, in a BCI application that I was just working in, we're not introducing energy into the body. So what aspects of patient harm are you creating if you're just taking information out? It's not completely black and white, but it is a way to think about partitioning your risk and architecting your software so that at least a portion of what you're doing can move quicker and can be more adaptable to change. If you are building hardware, in particular implantable hardware, this is what really drives the timelines. And it's really that ability to build and to test. And one thing I think a lot of people don't realize is not only are you building your prototypes, you are maturing the manufacturing line to be able to build your prototypes at the same time. So that's the thing that I think people don't realize or understand, that unless you're building upon, but even if you're building upon existing manufacturing methods, those methods have to come together and be able to build what your design or your product is. Those things can take a lot of time. We're driving that manufacturing maturity to be able to build your product. And then this is where global supply chain comes in. I've been told multiple times, I give talks at various academic forums there, and you just don't really have some pains with global supply chain. So your global supply chain is impacted by, climate change. A tsunami happened somewhere, like big global events, tariffs. These are the big economic things that are outside of your control that are happening that can impact your global supply chain. Understanding, are you building upon materials that are pretty easily accessible or components that are pretty easily accessible? If your device is more external or what you're working on is more software, you probably don't have to worry about this as much. If you're working with suppliers, it's good to be aware of that and to ask. In the implantable space, sometimes you have very unique suppliers or limited choices and suppliers. As well as, of course, if you're vertically integrating, even if you're vertically integrating, you create a lot of materials from the brace yourself. Right? But it's important we're interacting with the supply chain and understanding that we have to be able to source that material for a time. And then there's a balance for that is your last turn. Where are you going for? When you get your next round of funding? So how much do you want to purchase and kind of keep so that you're protected a little bit from changes in the supply chain? That can be a challenge within the balance, because that's the thing when you're locking up some of your funding, of course, buying up stuff. And then if you're working on implantable devices that require tremendous reliability, the reliability of your phones and it's feel pretty good, or you don't have to restart them that often, but it's not good enough for something that's implanted. You really need very, very high reliability for something that's implanted. So that can also drive certain vendors, certain kinds of testing to make sure that you have that high reliability for. So what this said is it's a little bit of a timeline story to kind of my journey at Synchron. So I was at Synchron for 4 years, and when I came in, I was the 5th employee in the US and the most medtech trained employee at Synchron. Synchron is built out of University of Melbourne, and it was primarily kind of more academic oriented team that did great things, but didn't know how to build a company, build more of a company, really, and the different kind of attributes that have to go with that. So I was going to be the first industry trained person to come in. The first thing that I did was set up a supply chain. I knew that implant timelines are long. I think one of the things that was interesting was I found people that were already implanting, so it wasn't the implant done. And the reality is it's safe. The early implant they built such that it was safe, but it wasn't really manufacturable. And so a lot of work had to be done to develop a stentrode and the other implantable technology that could be manufacturable and could enable Synchron to go into a pivotal. And one of the things when your device isn't manufacturable, you're having a lot of variability from part to part. And if you're going to work in a sensitive space like sensing, that variability might show up in your data and your outcomes. So one of the things I was really focused on early was supply chain. And these partners, if you're partnering with people, they last for years. 8 years, 9 years, 10 years, 15 years. These are long-term relationships. And so investing in those relationships, understanding, are these people that I can work with? Do they understand the risks that I'm taking? Are they vested in what I'm doing? And trying to build that relationship was great advice, actually, that I got early on, and I really focused on that. It was pretty interesting to come into a startup and be able to frame up and change the whole supply chain makeup for Synchron. The second thing we focused on was the quality system. You can buy out-of-the-box quality systems, and that's great. But understanding the quality system with respect to what you're doing and what you're working on. So, for example, we had team members that came in from working on orthopedic implants, and they were used to, very mechanically engineering-oriented organizations, and they were like, okay, all of your test plans need to be done at this point, and then we'll do the review. And he was like, I've got software operating at a different timeline than my systems team. I've got an electrical team that's operating different than my mechanical team. I've got all these different teams and 50 different people writing test protocols. There's no way that I'm going to be able to drive all of them at one time period. And then that's the best way to operate, or the most efficient way to operate. And so working hand in hand with the quality and regulatory team to piece that out, keep breaking the problem apart and piece that out so that piece by piece by piece we could be getting ready instead of, again, trying to eat them all in July. I think there's a saying, how do you eat the whole elephant? It's like one bite at a time. So that is like the theme here. And then the process of development. We went through a process of feasibility and asking some key kind of high risk questions and trying to answer those questions maybe through some technical reports, like some modeling and some calculations when we don't have parts, or trying to piece together maybe an early prototype just to address very, very specific questions. So that was kind of the feasibility process. And then we went through a process of what we called characterization, which was when we were getting our first devices off that early manufacturing line and just trying to exercise a lot of different aspects of the device and the system together as best we could. And then the kind of finally go through verification. If you're doing this while we're driving deep bench and preclinical methods, and they become your foundational tools. So as you mature and then you start to see issues even later on, you have the tools to go and address those, and the engineering talent to go dig deep and address those. So building up that capability was really important. I can kind of think about, about 3 years ago when we started to get that first lab equipment in, and over time, building more and more depth and capability. And then another thing that's about development is you will probably have to, and it will be tempting to use those early devices coming off the manufacturing line to do your preclinical work. But often what ends up happening is the manufacturing oil is still maturing. And so then as you do that preclinical work and working with animals, you start to see those things from the devices showing up in that preclinical work. That's all, I kind of like to say sometimes people feel really down when they find issues. And I'm like, if you're not finding issues, then maybe work isn't happening. So that is part of this process, that resilience to, just keep working the system, keep finding issues, and have a culture of surfacing those issues, talking about them, and tackling them together. And then the third thing I'll mention is what I'll call applied research. And that was a recognition that through the early feasibility, the EFS process with the FDA, the process of development from a technology standpoint continues through those early clinical studies. For cost of change reasons, like really drive down the safety of those implants, make sure your base functionality, so your electronics are looking as good as you can. That through that process of applied research, especially in the software space, we can still enable change and learning and keep developing and keep improving your surgical procedures. There are ways that you keep learning and improving through that applied research process and through the clinical studies, which is a really important aspect of development. So I'm going to close just with a couple of examples. I don't have any affiliation with either of the companies that I've chosen to highlight. I've Synchron recently, and just something in an exploratory mode, I was very, very heads down with my personal situation and work situation, and so it's been fun to kind of see what's going on, and just wanted to share with you a couple of companies and share with you kind of how I think about these companies and how they're approaching things. So one interest I had is Restorative, so I became a migraine therapy. And they kind of came out of some off-label clinician work that where the clinicians were taking spinal cord stimulation systems and tunneling the leads up and creating, I think they call it like a halo effect to address migraine. And they saw a good therapeutic effect, but they saw that the procedure and the devices were not withstanding this application. And so I think I thought it was really smart. Restorative went after a cranial mount device that was very integrated to remove this therapy and studying this therapy more deeply. And I guess what I saw was in the first early rounds of funding, they went up and making this device. And this is a great device. I guess from my eyes and lens, I see it grounded in a lot of those rooted principles that I was talking about. So I've seen a lot of MedTech practices. It looks to me like some standard lead technology, and I'm sure there were things that they had to do to make it into one form factor for their needs. But it's grounded in a lot of MedTech practices. So I think it was really smart, and they seem to use their capital really efficiently to drive hard into a pilot clinical. And so they did a pilot clinical in Australia and got great results. And that's what drove their Series B funding. And so I came across their story and I was like, wow, that's really great that they were able to build a device, get to that clinical data, really efficient about and drove hard into that. It happened to me. One of the people that worked in the clinical study, and I'm like, oh, that's amazing. And he was like, it was exhausting. It's so exhausting. So I got a little bit of insight there, but that just kind of wanted to share their story and how they approached it. The other one that I chose to highlight was precision neuroscience. I think many of you may be quite familiar with them. They're very well known, well funded. And just to give a little bit of an overview of what they're doing, the top picture is showing, their primary innovation, which is to create a slit in the cranium and introduce the neural interface, that kind of silver color is so kind of like stylet that would push the neural interface down and then be removed. And the neural interface could have a lot of electrodes. And then I would say the other two pictures that I'm showing are vision or concepts from their website of an electronic module that then connects to that neural interface. And then the bottom picture showing, how that electronic module will extract information from the brain and be used to connect with digital devices. And they had a recent Nature paper come out, which is a great combination of their work. And one of the things they're doing well is what I was talking about. How do you eat the elephant one bite at a time? They've really separated their work into separate work streams. And I recommend going and looking at that paper because I saw a lot of headlines on, even flashed up on LinkedIn and then amongst people who are pretty technically savvy. And it's easy to read the headlines or to look at the videos on their website and be like, wow, they already developed the electronics module and it's all done and it's all integrated. But going through the paper is really helpful. And what I found was they were separating the neuroscience work stream and the neural interface characterization by using these non-significant risk clinical studies. So these are studies where the patient may be going in for like an epilepsy study or something, or we're having a person in the cranium removed for other reasons. And then Precision is going in there with their neural interface. They probably had a system of equipment that's outside of the body, but connecting into that neural interface and gathering and capturing data. And that's very useful information. It's giving them a lot of design inputs and information about design of the neural interface. It's really allowing them to manage that kind of data and try to do some decoding type work with it. And also from a fundraising standpoint, they're showing that they've been working with tens of patients at this point. I think that 50 plus patients that they've been doing this with, by piggybacking on these types of clinical studies. So I think they're very smart in terms of showing the amount of information you're gathering, and they're gathering great design inputs for their development in their system. In terms of the procedure itself, they've been doing that preclinically. So they've been doing that with their animal studies. And they're now showing that they can take take a device like this and bring that style in and implant it. But the piece of integrating it and all that interconnects to the electronic module that then has to be implanted, that part they're not showing yet. It's not to say that they're not working on it, but I think they've been smart about what they're publishing and how they're talking about it. And how they're taking it one step at a time in terms of bringing that system together. So how do you break that system apart? How do you do experiments that address key questions as we're working on it? And one of the things I mentioned is it can be tempting to think, an animal study, I have to show my sensing and stimulation function and do that in the same test where I'm going to do my procedure the way I would do it in a human, and I have to evaluate all of those key questions in one animal study. Those might be very, very similar, different experiments. Something where you're looking at something mechanically and procedurally could be very different than looking at the neuroscience aspect. And maybe I'll just mention from my own experience, taking a human-sized centroid, the blood vessel of a human, and trying to find that same size blood vessel in an animal near brain tissue is really difficult. I think the comment I got from someone that's an expert in working with many different animals was like, I don't know, maybe a silverback gorilla, like that really wasn't a blood vessel like that would have all of those key aspects. And so you have to separate the problem up, ask your key questions, and then bring it together at a later time. So that's a kind of a process of integration. So hopefully I shared some things that you can take away that are tangible. I think we're at a great moment. There's lots of suppliers, there's lots of talent out there, there's lots of technology out there, and really probably the challenge is figuring out what is the right approach for you and what kind of funding do you have that are your goals, and how do you put that together. Thank you.
Good. So I think we have time for maybe a few questions. If you don't mind, try to shout. Otherwise, I'll run up with the mic here, too.
I could shout.
Okay, thank you.
Thank you for that. I work in an academic team, Stanford University, Blueprint MedTech, and many of the things said were right up the alley of advice people getting from our answer. So they really, a lot of things register. One thing you didn't talk about very much that I'm curious about is how, particularly coming from an academic side, they've been clueless about many of the things you talked about. How do you develop the right team? How do you find the right subject? You know how to be right coach talent from the Johnnies, or how do you do that?
That's a great question. So, I had an interesting journey because I think I was in New York and I'm from Minnesota. And people always told me that tech talent in Minnesota was so prolific. Like you go to the coffee shop and you meet a quality engineer. I do drop-in dance classes and I met my mechanical engineer. But I didn't really appreciate that until I went to New York. And so, I think a key learning I had was that medtech understanding and talent matters, and understanding, being in a location where you can find that. Now, of course, Stanford, I think you're in a great location, but there are a few locales where you can find that kind of talent. That was kind of my first learning. And then I think Becky started a nice conversation about language. So about medtech, language is a thing. And so you speak a common language, and when people don't come from that framework, they're not as familiar with that language. It takes a while to get used to that. So I think hiring talent from those areas that do have that medtech experience, I will say some of my best hires have been these neuroengineers. So I was doing internships with people at Medtronic, and then they were going back to do their master's degrees or PhDs with faculty that I had worked with, and then they were coming back. And so that kind of talent that's been cross-trained, or a lot of times a neuroengineer that then came into Medtronic as someone that has a few years of experience, they speak the language of medtech, but they also neuroengineering. Those were some key hires. Then I had some learning that there's the language of medtech, but someone maybe that's developed a product that's not chronically implanted. if you're working on a chronic implant, that's a key skill. Some people who haven't worked in a space of active implantable medical devices, so you might be familiar with that acronym AIMD. That's kind of a whole different thing, like thinking about electrical function in a device that's implanted chronically. So it was really through the process at Synchron where I was like, okay, I don't have enough just medtech people. Like, I need more medtech people. Okay, so we're not the medtech people, but now I need that electrical engineer that understands a chronic implant. I have to look very specifically for that. And it was a process over time of understanding, like, this skill is missing, this piece is missing, and building the team over time. I think in whole, probably seeded a good version of my talent. It took probably a couple of years. Not I was building from scratch. I mean, yeah. And I was probably in a more mature phase of this, I guess, a little bit. But yeah, it was a good couple of years to figure that out and get the right people.
Yes.
Yeah, hi, Matt Steve from Radiance Hopkins. Thank you for a great talk. Could you speak to your experience at Synchron, or maybe some of the case studies that we chose to speak to the milestones that you targeted in order to get to next phase of the timeline?
Yeah, I mean, I can speak to, like, I thought, so Sheer Electronics that's like, I think, a very good typical medtech journey. Like, it's not just get your device out there, but get to those clinical proof points speak volumes. It can sometimes be preclinical data, but understanding your investment community. So that's also a thing, like, just speaking from Synchron, but also looking and being familiar with other startups and their boards. Like, the BCI space, some of their boards are very tech-oriented, so they might be looking for something really different. They're looking maybe for lots of data, the kind of the Elon Musk bent of things as maybe a little bit of a different space. So understanding who are your stakeholders and what are you looking for versus another startup that I'm familiar with. They kind of started to imagine more of that tech future, and then everyone was like, just stay focused. You got to get clinical data, right? So figuring out that balance of who are your stakeholders and what are they looking for, how to look for those outcomes, and then who are you be going to for your next round of funding, and what is that population looking for?
Thank you. I know we do have probably just time for one more question. There is a question on Zoom. So I'm going to ask, I guess, if they can unmute and ask their question. Let's see if that works.
Hi, this is Rhonda Video. And I then credits to Yan Brown. IEH. No, probably ranges and progressively because as I selected. Like if one sitting today, first test ball, you're getting well as DJ on all the nobody stuff and jury try to fix you, replace your neurotechnical. Like what is your indicator? Should it have been some configuration on my best theory? If you want that, I say switch shifting to be a base and be good and well all and we really know that we would have taken that to the for what example, we tell about we don't know if it works. If not like people not the action call, if you become something difficult, just get it to you. I just think them will fall. Let me give you an example. I could look at that when you tell about they were girl well, reading about this way today. Make me hot. Never remember it feeling out the voice then. With the population get together. So we fell out and one is a good let it like your theater. If we ever keep with the of all that, then that make my eyeball. Then there's the eat. That one eye or you're there. That then they fell out and now boss. Nothing word deep down these. To a lot of now as we AMS, the light of buggy, it AMS and we should find a lot of that in the environment. They have your community.
Yeah, that's a great question. I didn't talk to the health economics perspective a lot. I think, that has to come into the frame as you develop your pitches, and it gets to the, at least think a couple of steps ahead, and you don't have to have everything figured out, but understanding, that macro level of, is this headed in a direction that's very impractical, or we'll have to pay the long way to try to get buy-in for it, or is this, piggybacking on a pathway that's paved already, and amenable to, that there are some roots there already. I think understanding that landscape is beneficial to that holistic picture as you develop your pitch is very funny.
Great. Well, I knew others have questions, so hopefully you can try to catch Dr. Banerjee later this morning. I want to thank you so much for taking the time to come. Just a quick thing, as you were speaking, so my father landed in Buffalo, New York for school in 1966. He does remember your father. And I'm born in Buffalo, so well, thanks again. I hope you have a time to talk to folks here. Those of you who are on the next panel, led by moderator Dr. Goodwin, please come on board onto the stage and we'll get started.
So good morning, everybody.
That is good.
Yeah, let's go section audio tools. You should have the geographic. It's not this. Yeah, I don't know about it. Yeah, but yeah, push me. So that is yeah, watch us. Like we gotta do the script cautions because we got meetings. I essentially talk. Set us. We had the scale. Thing. Striding down. Good morning, everybody. How are you? Me. My name's Roy Goodwin, and I'm coming from Duke. And essentially what I'm going to do is to provide a little bit of a primer for some of what our other panelists will be discussing in terms of basically patient-centered innovation and meeting patients where they are. We have a very esteemed list of guests. Some of them are very, very dear mentors to me over the years. And so I have the privilege of kind of moderating this session. So with that in mind, I'll kind of get started. So these are disclosures not really relevant. So when we want to define patient-centered neuro innovation, or basically meeting patients where they are, it's kind of made up of a couple of key concepts. The first of which is combining neuroscience, technology, engineering, and design. And basically placing the patient's wants and needs as the inspiration for innovation. And so in that vein, we want to treat patients as co-creators and collaborators, not just as end users. And basically the focus is on the meaning, the relevance, and the practicality, but from the patient perspective. And so I want you guys to kind of keep that in mind as we kind of go through this, and as you go through the rest of the session. The kind of the benefits of it is that, these patient insights can lead to improved effectiveness, adherence, and functionality, as we all know, enhanced design accuracy, relevance, and more importantly, the therapeutic impact. And you can encourage iterative refinement through patient feedback. When we think about the traditional way that innovation is performed, a lot of times we start from the can we make it work paradigm. And we're looking at kind of metrics such as technical excellence, the optimization of it, basically the innovation for the sake of innovation. And that's why I kind of read that talk by Dr. Banerjee before. But when we look at it from a patient-centered view, right, it's like, can patients live better with this, right? And the focus is more so, are we functionally aligned with the patient's values, goals, and their priorities as needs? And so when I think about those metrics, it's things like sigma to noise ratio, precision and accuracy, optimization, right? But one of the things that I think we need to make our priorities is basically comfort and ease of use, adaptability, integration into the daily life, and the things such as making things functionally meaningful. And I think that if we do that, then we can basically design technology that will empower patients and actually impact their lived experience as well as healing. So if you take some of the lessons from some of the other innovations just through the years, you can see where these things have failed to meet consumers. So I don't know how many of you guys are familiar with the egg tuber. So this is this failed because consumers found it labor-intensive process without much culinary utility. How about smell-o-vision? Right? This was actually something that was pioneered back in 1960s, 1970s, and it failed because audiences had nausea, headaches, confusion. Basically, they had a hard time getting all of the sense, across the entire theater at a meaningful time, so it matched up with what the story was. And so it also impaired the theater owners because they had, basically had problems with their ventilation systems with all the smells. What about these X-ray shoe fitting. So this is basically where you put your foot in a shoe. You put your foot into a device that is an X-ray shielded box. And what you do is then you basically take an X-ray and then that tells you basically, is this the perfect shoe, for you, right? And what this found is that it's, that we don't really care about what the patient what the consumer says in terms of comfort. You just care if the X-ray fits. Right? These are places where it failed innovation. What about from a Neuro Innovation standpoint? What about high-use electrode shock therapy? Right? Treats anxiety, depression, exhaustion. Right? It failed to meet patients because dangerous shocks, frightening to users, and it was replaced fortunately with clinical electroconvulsive therapy, right? What about tranquilizer chairs, right? Turn used to calm your consumer, right? This actually failed to meet patients because they were dehumanizing and uncomfortable. A lot of psychiatric, as a side note, a lot of psychiatric facilities tried to use this to calm patients. Let's just say it didn't work. Why do I bring all this up? Because it reminds me of one of my favorite quotes by Raphael, which is that basically the jelly to my burger, the knife to my soup, the burger to my sushi, and the ketchup to my ice cream. Some of these were worthless. And I think that we have to keep that in mind when we think about patient-centered innovation, is that are we doing the thing that's going to actually make these patients' lives better? And that's focusing on answering the right questions that are most important to patients. And these are some of my mentors here, two of which are on the panel. So just to give you the background of where I am, I basically am a spine surgeon, and I do a lot of patient-centered spine research ranging from degenerative diseases, trauma, infection, deformity. But my bread and butter is actually spine tumors. And spine tumors are common, and it's important to just know that these are patients that usually have a poor prognosis and they're at risk for low athletic quality of life. And so the goals are really to kind of maximize their quality of life, right? And that goes from understanding their goals as well as their expectations through patient input. Now, fortunately, and I'll tell you this, and don't let him know this, but I had this really great mentor named Dr. Henry Bum, who taught me basically that, I need to listen to my patients. And so when I got to Duke, I basically formed this spine tumor support group. And this spine tumor support group served as the cornerstone for all of my understanding and my goals in terms of developing innovations as well as programming for these patients. And what I did is I allowed them to basically identify the patient-centered clinical, basic, and translational research questions that I ended up investigating. So when you ask them, these are the things that they said were their priorities. And really it was basically how am I going to be able to rep, local tumor control, pain, XYZ. And so when I first went in, I thought of all of the clinical questions like this, basically surgical techniques, drug resistance, metastatic progression. But what I then turned all of those questions into was from the patient-facing standpoint, right? I want this surgery possible. How do I actually make this decision? Will this tumor get worse? Will it come back? Will I be in pain permanently? Will I be able to walk again? Am I going to be survivors? Am I going to be a burden on my family? And taking all of those prompts from patients, I then turned all of the research efforts that I did and made it very, very patient-centered to address these questions. And so I'm going to give you a really brief example of this. This is a 35-year-old dog, 3-month history of back pain, 2 unsuccessful beta biopsies, has a malignant neuroendocrine-like sarcoma. You can see there the tumor, it's causing severe spinal cord compression, and it's where those red arrows are. It ends up undergoing this 2-stage procedure. Basically, I had to do what's called an en bloc resection. You take the whole thing out as one piece. And then I did this complex reconstruction. And afterwards, what the patient told me is, he said, I just want to be able to dance at my niece's wedding, but my priority is that she got it all. And so he asked me, he's like, how do you tell what's tumor, what's not? How long will it take to find out if you actually got it all? And I said, well, I study your imaging, study its experience. Usually it takes about 2 to 4 weeks for pathology to come back. And he was like, you don't have something just in the OR while you're right there that can kind of figure this out? Like, why do I have to wait 2 to 4 weeks for this thing? He's like, listen, I don't want to have to get another surgery. I just want to figure it out. And so what he was really saying is that, that you lack an objective, non-destructive, intraoperative tissue identification system. That's how I took it. And really, you want to get it so that way it can give me some real-time data. And so I collaborated with a colleague of mine that's in the brain tumor space, Dr. Khan, and we developed this intraoperative tool that uses fresh and defined parameters to differentiate tumor from normal tissue. And it can do this in real time in the OR. So I can literally, basically, after I do the resection, scan the resection cavity, make sure that I got everything, and then I was able to round it. And it uses this spectra. I can give you more details on this, but the goal of it is that when we took this device and we took out the specimen, we took one out, and you can see from the wavelength spectrum that basically that's what mine looks like, and then that's what it looks like with tumor. So that way we could actually tell if this thing, if we ended up getting it all. The second perspective that he gave me, which was also fun, is he says, how are you going to see around things that you can't move, like my spinal cord or arteries, to make sure that you got it in? And I said, I have some tricks. And he said, but it's hard to see behind that important stuff, right? And I said, yeah. So the main clinical problem that I translated that to is that there's limited, safe, and linear trajectories within the surgical workspace. And so we basically wanted to design kind of minimally invasive devices for those nonlinear trajectories. And so we came up with this tactile robot, which is basically a robot that is able to move and track. And my colleague, Dr. Khan, wanted to use this for pituitary surgeries. I wanted to use it for spine tumors. And when we combined that with the tumor ID, we came up with the NAV ID. Now, I could go through, we did this value proposition in terms of, okay, this is the patients that we can treat. These are the surgeons. These are the other applicable markets. These are the way that we can affect patients, industry, and surgeons in terms of low frequency. But all of these things were from the surgeon perspective. What I really wanted to know was, what's this going to do from the patient perspective? And because I was keeping the patient-centered focus of it, what I realized that he was asking me about is the distress about, oh my gosh, I have to have this huge surgery. Am I going to have to get one again? And distress is what's known as the unpleasant emotional experience that's associated with psychological symptoms and basically the patient's ability or inability to cope effectively with their cancer diagnosis and treatment algorithms. This is the distress thermometer, and it's part of the research that I do in conjunction with a lot of these kind of technological advances, where you can see that basically the no distress thermometer in the preoperative, the distress thermometer is red in the preoperative, and it goes all the way to 30 days, which when you do this across patients, like, and we did this across all of Duke, you can see that most patients have a lot of distress up to 30 days, which usually corresponds to when they get their diagnosis back. This is the NavID team for anybody who wants to know. Just a shameless plug. I'm just going to put that there. But you can come talk to me afterwards if you want.
Sure.
But the reason why I bring that up in terms of that patient-centered focus and distress, and this is kind of how I'll close before I introduce our first speaker, Dr. Ann Brasberg, is that for us to tackle some of the most challenging problems that patients face, it's all about attitude. It's all about how we approach it, right? We can take down the biggest problems and the biggest challenges as long as we have this focused approach and we incorporate basically all of the key elements that will make us basically successful in creating the most optimal thing for patients. So I'm going to leave you with these 3 takeaways, right? True innovation meets patients where it focuses on the meaning, relevance, and practicality from the patient perspective. Patient-centered Neuro Innovation enhances function, efficacy, and adoption, and it fosters a culture of patient collaboration that will transform our technology and advances into patient healing. The most important thing, I think, is balance. We need to balance all the technological innovations that we make with basically keeping that patient-centered meaning and focus, and I think with those 2 things together, I think that's how we can actually impact outcomes. So with that, I'm going to say thank you just for this primer in terms of where we're going to go, and then I'm going to invite Dr. Brasberg to basically give our first talk.
Oh, great.
Yeah. Thank you very much, Drew, for inviting me to come and give a presentation. And specifically, I was asked to present a specific case. So she said to me, you know, I heard about this case, I saw this thing, and I want you to come and talk about it. So patient-centered, what happens to us as a surgeon, as a neurosurgeon, everything we do is for our patients. And when we're clinician scientists on the side, everything we do as a scientist is based on our struggles treating our patients. So we are very much a back and forth from the bedside to the lab, back and forth, trying to always figure out what we're going to do next to help our patients. And hopefully we bring a lot of that from dealing with our patients. Now, how we do it and how well we do it really depends on how we think these problems through and who we can collaborate with. And the better our collaborations, usually the better the product that we produce. So we really truly believe in collaboration in its true meaning. We want to collaborate across disciplines, bringing that power to the questions we have. Hopefully we have good questions because we center those questions on our patients and on our patients' needs. So it really is a true back and forth. And that's what we end up doing, running to the lab, running to the hospital, running to the patient, running to the operating room, and so on. And sometimes these things occur all at once. And so I'm going to present a case for you where that all happened at the same time. So I had two presentations. I'm going to go to the second one because in the interest of time for you, so I'm going to try to move this. So I'll come back to the other one if I have time. So I'm going to present this in full disclosure. There's going to be operative pictures here. So if you have a problem, for the non-medical people in the field that are in our audience and so on, if you want me to hit a switch or something or say, this could be operative, you're going to be looking at the insides of people, please don't hurt that. These are very nice seats. We don't want to mess them up. So this was an interesting patient. This was a 17-year-old who had gotten run over by a motorboat. And basically, unfortunately, alcohol was involved in the driver of the boat. This was a very competitive surfer who was behind the boat in the water. And unfortunately, the person driving the boat was intoxicated and backed up over him. He got hit by the prop in his legs, got treated at home in Texas right away. They saved his leg. They saved his life. They realized his sciatic nerve had been really traumatized very, very badly. And I got a call from one of the surgeons saying, we've helped his leg, we've done what we can, but we can't do anything with that nerve. Any chance that you could have a look at this and see if you could do something? So I said, sure, send him up, send him to Hopkins, we'll see what we can do. So they sent him up and we took him to the operating room, and this is a few days after the injury. We explored it, and what we found was a 7-centimeter gap in his nerve. Now, this is way up in the buttock region. And when you have a big gap, you can't quite pull that nerve together, so you got to splice it like you would splice a wire. And we harvest nerve, or we harvest wiring, so to speak. We fill in the gap. Sometimes it works, sometimes it doesn't. The younger the patient, the better the outcome. But a gap with more than about 4 to 5 centimeters is problematic. A 7-centimeter gap is huge. And high up in the buttock means it's going to take years for regeneration to occur down to his leg where he needs the regeneration. So everything was not in his favor, so to speak. And the question was, what do you do in a situation like that? Is there anything to help? Well, this is what it looks like. This is a beep. You're looking inside a body. Please don't hurl. So what you're looking at is the buttock here. Okay, the patient's prone. This is the top of the butt. This is the top of the thigh. And this is his sciatic nerve explored. Now, what you see is the two ends are actually overlapping. And rather than a big gap, here we see that the nerves are actually overlapping. And when I go to trim this and get rid of the stuff that really is, it looks like we'll be able to do a direct end-to-end repair, which is in his best interest, and not have to graft that. Now, how did we get from a 7-centimeter gap to an actual overlap where we could do a primary repair? So the first thing to think about, he's 17 years old. Now, even though he's 6 foot something and a surfer and a dude all the way, he's 17, he's a minor. His parents are out in the waiting room. They've flown up from Texas, and we go and we have a talk. And I talk to his parents and I tell them, if I have to graft this, he's not going to get an outcome. He'll never get that sciatic back. I really have to get the ends together, not use cable grafting. And we said, got an idea here. I'm not sure if it's going to work or not, but it's an idea. This is my colleague, Dr. Greg Osgood, who's an orthopedic surgeon, and he's gotten his hands something called a giggly saw. And what we're going to do is we're going to shorten his leg. We're going to take out a part of his femur, shorten him so I can do an end-to-end repair. And then once it kicks in a little bit, we'll regrow his leg. That's the concept. And the question is, can you sell that to the parents in the waiting room? This is not exactly things that are well known in the literature. As a matter of fact, there's nothing I can say that's been done before.
Do I think it's going to work? Maybe. Do I think it's going to be bad, good? I don't know, but it's an idea, and we kind of have to make a decision now. And so we sold that to mom and dad, and this is putting in a rod across his femur. We've taken out a chunk of bone. We're going to put a rod across, and then we're going to shorten his leg. So again, I need to get those nerves to overlap. And that's the rod across the femur. And you see us squeezing together his femur and literally shortening his leg. I need the volume up. Can you guys get me volume on this? Yeah, no, I do need volume for a later one. So I just want to see if the volume plays. Okay, so we'll use it for later. All right, so that's shortening it. Here's the repair. So we now did an end-to-end repair. This is some of the muscle being brought over top. But we've repaired the nerve directly end-to-end, little micro sutures, some tissule glue over top. And that's what it looks like. Literally, that leg is 7 centimeters shorter than his other leg. Now, he's not going to stay that way. I'm going to give him 1 month, and then after a month, I'm going to re-break his femur, and I'm going to put in what's called a growing rod. That rod has a gear in it, and very slowly as the gear turns, we're going to re-stretch him out. We're going to regrow him to where he was. So that's going to start about a month, month and a half after the original surgery, and it's going to go on for a couple of months. And the way that rod works is we put the magnet over his leg, and the magnet drives the gears in the rod, and every day we're going to stretch a little bit, and we're going to grow him back. So that's the concept. Now the question is, with that concept, how long should it take for his nerve to regenerate? When can I expect a response? When can I start looking at this thing to see if it's working? Now remember, we put the nerve together, but it's the inside of the nerve that has to regrow. Those axons, otherwise known as copper filaments, like in a regular wire, they have to regrow all the way down to the target. So here's his repair way up here. There's his calf, below the knee is target, calf muscles, shin muscles, and so on, and it's about 19 inches. So we think about regeneration at about 1 inch a month. We've got at least 19 inches to go. We also have to add a month for the axons to grow across my repair. And remember, I'm going to re-stretch him out. So that's another 3 months for the re-stretching, right? Because I'm going to regrow that leg. So when you start adding all of that up, it's going to be at least 2 years before those axons reach his calf, before we have any idea if those axons are getting in. And then it's going to take time for those axons to grow into the muscle and give him motion. So I'm sitting there thinking and telling the parents and everything, the kid and everything, I think it's going to work, maybe it's going to work, but it's going to be 2 years so we have any idea. Not exactly great. 2.5 years for motion is what we're expecting. So I've got some time to think about this before we expect a result. So now I go to the literature, I've done that, I've stretched him back out, and I start reviewing, has anything been done on this, in the past? And I start seeing these articles. Now, Elizarov, a Russian, he talks about limb lengthening, and limb lengthening is actually a common thing in the United States as well for people who are having problems with height and so on. So the idea of lengthening a limb is not new. The idea of shortening it and then re-lengthening it, a little bizarre, okay? But this lengthening isn't so new. The problem is when I go to the literature, lengthening causes nerve injuries. So I've used a concept of limb lengthening to help a nerve repair, and the concept I've used causes a nerve injury. So the question is, what do you do at that point? So I did the obvious thing. I met with the lawyers at Hopkins. And I said, we've got a serious issue here. And it's going to be 2.5 years till I have an answer, but I may have done the exact opposite thing of what I should be doing. So if you go to that literature, it's not reassuring. There's nothing specific on what I've done, but the things of like what I've done don't look very promising. So now an interesting thing. 8 months post-op, he sends me a video. And he says, "Doc, I think I'm moving my foot." Now it's 2.5 years before anything should happen. This is 8 months. And he shows me this video and says, "My foot's moving." And I go, "I'd love to believe you, but I don't believe you. So I think what you're really doing is maybe moving your hip, and that's what's going on." So I tell him, "Hang your leg off so you can't move the hip and everything. I want to see that foot move," it's his right leg, "without your hip moving or anything like that." He says, "Okay," sends me this video. So I'm looking at that going, okay, so he's got the fishing wire. You can't see the fishing wire. So I call his parents and I said, "Guys, can you go check, on him? I really want to know whether that video is legit or not." And they said, "Yeah, no, we were there when they filmed it. He's moving his foot." Now he's 8 months. 2.5 years is what that should take. Do I believe it? I don't believe it. What do I do with that? Well, the bottom line is, he was right. We brought him up. I said, "I'll pay for the flight. I want him up from Texas back to Hopkins. I want to see this for myself." And he was moving his foot. And he was using our nerve repair at 8 months to move the foot, which is against everything we understand in biology. So what the heck is going on? How did we accelerate neural regeneration in this patient? What were the variables? What do we need to look at? Well, first one is nerve stretching. We know that if you put tension on a repair site, worst thing you can do. But what if the repair site is fine and I'm stretching the whole nerve? Does that stimulate neural regeneration? When we grow, we stretch our limbs as we grow and nerves regenerate. So maybe that nerve stretch was a hell of a stimulus for him to beef up the neural regeneration. Was it the magnet? Now there's a lot of data in the literature, in the basic science literature, that electrical stimulation early on in nerve regeneration helps a nerve regenerate, stimulates it. Tessa Gordon's work from Edmonton, Alberta. And that's now moved into human trials with people sticking electricity on nerves all the time when they try to regenerate. Maybe it's the magnetic field that I was driving that growing rod with that was stimulating the nerve. Was it the fracture? Remember, I re-broke his femur, put that growing rod in and kept it fractured for a couple of months while I regrew it. Think of all the factors that are from that fracture that could also be hitting the nerve and stimulating the nerve. Is it genetic? Is there something about this particular individual who, if you studied him just with any other nerve regeneration in his body, he simply regenerates quicker? Is there a phenotype? Is there a genetic predisposition to the way we regenerate our nerves? I think the most obvious thing, the clearest variable here, is simply everything's better in Texas. So if you injure your nerve, go to Texas, just live in Texas, and things will go better. So what do you do with this? So I'm sitting there with a kid who just blew us away in terms of his biology. What do we do next? So we take this to the lab, and we try to break down those variables and study them individually, set up a system, we set up a sheep model of this, and we try to break it down and say, where are we going to go with this? What is the various technologies? What's the various biology behind all of this? And how do we take advantage of it for our next patient? So you have to go variable by variable, controlling all of this, obviously, in a laboratory animal, and then you move it to human. And we're in the process of doing that. So I've worked very closely with Amit Hoke, one of our neurobiologists at Hopkins, and we look at all the different elements of neural regeneration that we can bring to bear on this. Now that first one, for example, nerve stretching, and for an audience like this, it's very important. How do you stretch a nerve? So there is technology already out there from UC San Diego of a little implant that can stretch a nerve. So we start adapting that and saying, can we take it and adapt it to this type of a scenario where I do a primary nerve repair and put in a device to stretch the nerve. So we start thinking about this from the individual patient. This patient generated so many questions and will continue to generate So 2.5 years for nerve to regenerate, and in less than a year, he is back on a surfboard, surfing away. Those are the kinds of things. Those are the kind of things that we always say it's such a privilege to look after patients. It's such an honor because as you look after patients, it's the patients, what they're bringing to you are these questions that are just fascinating. And if you pay attention, you get these wonderful questions to go to the lab with, to go and collaborate with all of our scientists with, and try to bring to the next patient. But it's patient by patient, watching, listening, and learning. So I'll present the other one if we have time afterwards. I don't want to take other people's time. So, thank you.
Thank you. It's a pleasure to be here, and it's been one of the greatest privileges for me for the last 3 years now to be involved in following the great progress of Neurotech Harbor and the Blueprint. And I think our goal is to really change the world through neurotechnology. And the topic this morning is what do we learn from patients, or making it patient-centric. So like Dr. Belsberg, I'd like to give you an example of where a patient didn't follow the rules, and one of my teachers when I was a medical student used to say that the answer generally when clinicians are faced with phenomenon is we see this. That's their answer. That's the professorial answer. Yes, we see this. This happens. But the question is, what do you do about that, and how do you learn from the patient? And I think that was our message today, is to give some examples of patient-centric. So if I can advance this slide. And then the disclosures are important because I would say that the companies, that's just core to what we're doing today, is that's how you evolve change. It's both academics and companies working together. The problem that most of my clinical career has been focused on is malignant brain tumors. And this graph just demonstrates how difficult, how dismal the median survival is measured in a few months, and almost all the patients are dead within 2 years. That's what we've been faced with for most of the time. I'll show my next talk a little bit how that's changed, but basically that's the picture for the most common brain tumor in patients, and almost universally fatal tumor. So one patient came along that really impacted us and affected us, and this was a gentleman who's 39 years old. He had been the administrator for the neurosurgery department at Hopkins. A few years earlier, in his mid-30s, he had left that job as the administrator for our department, went to Sinai Hospital, was married, had 2 kids, a 3-year-old, a 5-year-old, and he was driving home from the hospital, and he stopped at a red light and woke up in the hospital after having a grand mal seizure. His MRI, his CT scan MRI showed a left frontal tumor, and he transferred his care to us at Hopkins. I operated on him and removed the tumor. It was a glioblastoma multiforme, which was devastating to us and to him and his family, because it was someone who was very well known to us, close to us, just a person at his prime, and we knew there was sort of a death sentence, and that no matter what we do, what the outcome was going to be. We treated him at the time with systemic chemotherapy, BCNU cisplatin, and he came back a few months later after the radiation and chemotherapy with an infection. We thought, like, oh my God, it's not bad enough that he has a glioblastoma, now he also has an infection. So we tried treating with antibiotics, didn't work out, so I wound up taking him to the operating room, cleaned it out, tried to preserve the frontal bone, and he came back afterwards a month or so later, and it was still purulent material, and so he said, we have no choice, but we have to take out the bone. So now we took this young man with these young kids who's, still functional, but we knew had a very limited time span ahead of him. And by taking out the frontal lobe was bone rather, not the brain at all, is a horrible thing because you could see his brain pulsating. So it was impossible for him to interact with people because they'd look at him and nobody, even a neurosurgeon would notice that it's such a bizarre thing to actually talk to somebody and see their brain pulsating in front of you. So he was somewhat miserable and knew his prognosis. He kept asking me when he can have a cranioplasty and have it repaired, and I was thinking, he's going to die in a few months. The last thing I want to do is take him back to the operating room now for a third or fourth time and put in artificial material, and then he's going to die anyway, and that'll probably get infected, and we're in this terrible cycle. Let me just wait it out. But gentleman was very persistent, and he said, I just can't function like this with everybody seeing my brain pulsating, and I want to get back to a more normal life, even if I only have a few months left. So we took him back to the operating room, did a cranioplasty, and then waited for the inevitable, because we know that graph, that time frame, that 8-9 months out, that it was going to recur and that he would die soon after that. But instead, we did the cranioplasty, and that's 1991. This is a scan from 2019. And tumor never came back. It just, we kept waiting. I got scans all the time. He'd come to clinic. I'd apologize to him because he'd come to clinic, first every 2 months, then every 3 months, every 6 months. And he'd wait a long time, after he got a scan for me to review it, to inevitably tell him that it had come back, but it didn't. And I'd apologize for how long he had to wait. And he said, I don't care. He said, just keep giving me this news. I don't mind how many scans you do. And long and short of it is, he's still alive. There's no obvious tumor in resection site. It's been 34 years, not 9 months, since that time. And he's continued to work. He raised his family. He has beautiful grandchildren now, not just his children all grown up. I think what's written in red there is that for generations, I brought him to the Hopkins Medical School class to teach the new medical students about hope and about the lack of predictability and about that you don't shut the door on people and tell them that they're definitely going to die or something else like that, which many physicians had told him, and he was very explicit with the new student doctors that you don't do that. But the question is, what does it mean? And do we just do it? Dr. Folkman said, the professors all said, we see this. Occasionally you see an exception. Or do we say, what happened to this man? So we sort of thought about it. We said, well, the only variable, and we reviewed the pathology, many, many times. It was a glioblastoma. The only real variable is that he got this infection. So we thought, should we go around infecting all our patients? Maybe that's the solution, but that doesn't work out well, and you don't stay in practice very long doing that. And also people have actually tried that and lost their licenses doing that. It's not a good, because most people just die from the infection very quickly. But the question is, could we find a way to harness the immune system and say, gee, can we simulate what the body reacts to an infection and harness that in some way or another to change the way the body responds to this horrible malignant tumor. After, I'm going to just show you very quickly what amounted to 20 years of following this line. And my point today is just that if we learn from patients, what happens, like Dr. Belsberg's example, that you start to say, well, can we extract from that how we can help other patients? So just to very rapidly just say, well, can we augment the actions of the immune system to help us? And in the laboratory, and this was a Justin Haynes, who's was a postdoc fellow in my lab, now is professor of biomedical engineering, and has started many companies in this space. But just shows basically, and with Drew Pardell has been a huge participant in this, another person who's taken neurotechnology and technology through and really changed the world for the better with it. But basically in the laboratory, we could see with microspheres that we could cure otherwise incurable cancers by simply harnessing, by putting cytokines and other methods. And just to very rapidly, not get into a big scientific discussion, but could we reverse immunosuppressive strategies? So Mike Lim, who is now chairman at Stanford, really was one of the leaders in taking checkpoint inhibitors with Drew Pardell, showing in the laboratory how effective they were, and doing clinical trials, which actually didn't show a benefit for brain tumors. It revolutionized the cure of melanomas, lung cancer, renal cell carcinomas, but all the clinical trials, and there was a tremendous number that Mike really led nationally and internationally, didn't show a benefit. So the question that then took us back to, like, why is it failing? Why aren't we seeing it? And I just say that this whole checkpoint inhibitors is revolutionized, but this review article, about 2 years ago said we need innovation. We need entirely new approaches with checkpoint inhibitors. And so we took that challenge. And the question became from Demetrius Mathias, who now is at WashU, but was in our laboratories at the time, is it possible that we have this all backwards? We're saying, let's treat in a standard way, which is surgery, radiation, and chemotherapy for brain tumors. How do we learn, basically, from our failures and from our patients? And then we give them chemotherapy, radiation, and we say, let's see if we can't juice up the immune system and have them wipe out their tumors, like we think is what happened to our patient, who 34 years later is still alive and functioning. And maybe we're doing it backwards, though. Maybe we're treating with radiation and chemo, we wipe out the immune system and then say, hey, let's put a checkpoint inhibitor that juices it up. So we went, and I'll be very rapid with this, we went to the laboratory, and this is Science Translational Medicine, and I actually would point out the date on this if it's there, or it'll come up in another place. This was quite a while ago, 2016. And I'm going to come back to that as the final point. But we showed that if you take, in our animal models with checkpoint inhibitor, you take chemotherapy, systemic chemotherapy and a checkpoint inhibitor, you can see you shift the curve to the right, you get some long-term survivors. But the best thing was local chemotherapy, so you don't wipe out the immune system. With the checkpoint inhibitor, we had the large number of cures in these animals, and we certainly shifted the curve to the right. So we said, why don't we see whether that works clinically? It's great in the lab. This is very powerful. It's a very highly cited paper. It's an important, because it would change our whole paradigm, the whole way we treat brain tumor patients. And I will say that the 2016 business is very significant because I was incredibly excited by this finding, and it took 7 or 8 years to get through all the lawyers and the different companies, because it involved both the companies for gliadel that were producing the local chemotherapy and Bristol Myers initially, and then another company that was producing the checkpoint inhibitors. And all the lawyers kept getting into a room and saying, who's liable if something goes wrong with the patient? And it drove me out of my mind, because what I said to them in those boardrooms, conference rooms, was that in the meantime, every patient who has this disease, since we've started these discussions, has died. So there is no bad thing that can happen to these patients that the company is going to have liability for. We're sitting on a potential change in the way we treat these patients. Can we just move forward? And it took several years, and we're at, so I'll come to that in a second, but we're at the point that it's being tested clinically. And this actually is one of the most exciting experiments. This is the one, again, from that paper. And you took the long-term survivors from those different ways of treating the tumor, and then you look at it, and the bottom line is that if you took the checkpoint inhibitor alone and re-challenged those animals, there was no tumor. If you put systemic chemotherapy in the long-term survivors from that and re-challenged, they immediately grew tumor. This is looking at the brains of the animals. If you took systemic chemotherapy and the checkpoint inhibitor and looked at, you re-challenged them, tumors grew right back. But if you took the local chemotherapy and the checkpoint inhibitor and re-challenged, there was an immune recognition. So don't wipe out the immune system and don't treat the way we currently treat patients. So to test this idea has taken years. But the good news at this point is that this was the conclusion, is that we designed a clinical trial, and after several years of fighting to make it happen and getting the funding and so on, it is being tested. And we will have an answer, and it's either right or wrong. It's a testable hypothesis, but it just shows how you go from patient observation, sometimes the good fortune that you see in a patient like Dr. Belsberg's case, in my patient, and then just not just saying, wow, that's a good day, but saying, how do we then make that think about how we should be treating other patients? And what we're trying to do so much with Neurotech Harbor is to take all these observations, all these things that we learn from patients, and both learn about what their needs are, as Dr. Goodwin so amply showed, what does a patient want from us, not what we want to do, but what are they actually asking about, and then also learning from the successes as well as the failures. Thank you.
Hi, my name is Rena. I'm currently an undergraduate student at Johns Hopkins, but before I was an undergraduate student at Hopkins, I was actually a patient at Johns Hopkins Hospital in their children's department, and so I was being treated for something called functional seizures, which are also called psychogenic non-epileptic attacks or psychogenic non-epileptic seizures. And during that time, one of my high school teachers did not think I was going to make it to graduation. So how I made it from graduation all the way up to being an undergrad at Hopkins. So when I was in middle school, I had my first seizure on the lacrosse field. So my vision goes blurry, and I would forget just the basic rules of lacrosse. I've been playing since I was a third grader, and I would not remember a single thing. And so these symptoms got worse, so I quit lacrosse. I was like, okay, maybe I'm doing too much. I quit lacrosse, and then I move on to school and church, and I would also have these other reactions. I would forget English. I would forget how to walk, and sometimes how to swallow. I would also have migraines and random jerks. I also lived in a very rural area, and so there was no pediatric neurology care. They took me to the emergency room. They couldn't find any abnormalities. They told me, just go home and good luck. And so I got referred to my pediatrician, and my pediatrician actually thought it was stress-based, but I had no explanation for the physical symptoms that were happening to me. Is there? I don't see the clicker. Oh, is it? This one, okay. Green button. Oh, very cool, okay, thank you. So I lived in a rural area. So I live on the very East Coast in Ocean City, right there. The closest pediatric neurologist to me was in Baltimore at Hopkins. But in order to do this, they would have had to travel, my parents would have had to take a day off of work, taken me up to Baltimore, so it'd be a whole day worth of travel. And this is just really impossible, so the local adult neurologist decided to take me in. And that's when they decided to do an EEG for me, and they diagnosed me with epilepsy. Epilepsy is different from psychogenic non-epileptic seizures because those seizures, you can see abnormalities in the brain through the electrical signals. However, PNES, it doesn't show those signs. However, these EEGs are only 30 minutes, meaning that there's no guarantee, even if you have epilepsy, where you can actually capture an event, meaning we are relying on patient description, family history, and et cetera. And for me, in my case, the adult neurologist thought I was too young to be diagnosed with functional seizures. So over these 2 years, I was put on anti-seizure medication, going from 25 milligrams all the way up to 525, seeing no reduction in seizures. They started to get worse. I started falling. A lot of my peers were really scared. A lot of them also made fun of me, but a lot of them, I think, just didn't know what to do and didn't know how to help me. And honestly, the doctors didn't know what to do either. And they told me that my next steps were to continue to take more medication, and eventually, if the third medication failed, I would be a candidate for surgery. However, because of COVID, everything became telemedicine. So I was actually able to see a Hopkins epileptologist who told me, in the pediatric department, who told me I probably don't have epilepsy. And so I was misdiagnosed for 2 years. Instead, they told me that I had to do a video EEG, which is a 4 to 7 day monitoring in contrast to the 30 days that is typical for diagnosing epilepsy. And so here's me in my sophomore year of high school. I'm on Zoom class. I'm in AP US history in the unit. But they took all these electrodes, placed them on me, and they would try to stimulate these seizures. So this included sleep deprivation, taking off my medication. They even brought a flashing light machine to try to trigger my seizures. And all this was to try to figure out whether I had epilepsy or not. And so eventually, they were able to find out that I don't have epilepsy. When I found out that I don't have epilepsy, it was very challenging because for the last 2 years, I had been told that I have epilepsy, that there was a path. I just had to take my medication and hope it works. But in order to treat functional seizures, you have to face the anxiety head on. This became more of a personal struggle, or you had to face your personal challenges rather than rely on medication. And this was after and that's why a lot of patients struggle with transitioning from getting epilepsy, the misdiagnosis rate is about 30%, and a lot of patients struggle from getting a different diagnosis as they've been told for a lot of years that they have a different condition. And now all of a sudden, they're being diagnosed with a psychiatric condition, which comes with a new set of stigma. And now a lot of people also feel like it's their fault. With epilepsy, you have that abnormality in the brain. I think it's very understood by a lot of people. I felt like when I got diagnosed with the psychiatric condition, I somehow felt this is all my fault, and I could have done something to stop it. However, the gold standard treatment for this condition was cognitive behavioral therapy, and also the closest location was Hopkins. And it required my parents to take me every week to Hopkins, so a 2.5-hour drive from Ocean City all the way to Baltimore every week, and that is not something we could have done. So I also got sent home from Hopkins saying, now we know what you are diagnosed with, but we don't know if you'll actually get better. However, if you do solve it, you'll become smarter, so try your best. And so where medicine failed, God didn't, and I continued to once I got here, I had really bad anxiety, but I started praying more. I started prioritizing community rather than stressing a lot about school. And it was a lot of discipline, but it was very blessed. It was a huge blessing because I eventually got to grow and manage my anxiety even without the gold standard treatment. And now, 5 years later, I'm able to do research. I'm actually able to play lacrosse again, even though I quit, and I get to mentor high schoolers in Baltimore. So without any seizures. So I have my last seizure, I think I had 1 in the last year, and this was a 2 to 30 seizures a month was my previous rate. So not having to do that, not having to be in and out of the neurologist, and getting bad news after bad news every single time, I now get to have the freedom to be able to explore different things. And my story is rare, but also not rare. It's not rare because 81% of patients with psychogenic non-epileptic seizures have a 81% of patients with psychogenic non-epileptic seizures see over a 50% decrease in their seizures after getting the correct treatment. But what is rare is for me, it took me only 2 years to get my correct diagnosis. For most functional seizure patients, it takes 5 to 7 years. And that is mostly due to the inaccessibility. If you go back to the first slide, here's all the level 3 and 4 epilepsy centers in the United States. There's about 300, including pediatric and adult. This is the only way to diagnose functional seizures. And in order to be seen at this center, you must have usually you have failed epilepsy treatments. And so that can range anywhere from 2 to 7 years. And for me, it took 2 years. So in our lab at the Neuromedical Control Systems Lab at Johns Hopkins, we seek to try to reduce that time so we can make sure we can treat an actual treatable condition at the right time. So I'll leave it to Dr. Simon to talk more about what we do in our lab.
Thanks, Rena. The animations didn't come here. No worries. Just want to thank you for coming and sharing your story. So I'm an engineer, so unlike these esteemed clinicians that learned from their patients and turned their observations into innovations, sometimes maybe we have a place for engineers to come and play in your sandbox. So you heard from Rena this misdiagnosis. And to me, I actually study epilepsy. We build models from brain imaging data to understand disease, to help with diagnostics and therapeutics. That's what my lab does. And Rena came to me a few years ago, I think the freshman year, and she must have sought out, doing research to help her understand her own condition, what was going on in her brain. And she came in at a time we had just developed a solution to her problem. So just to give you an idea here, 30% of patients are misdiagnosed on average on that very first visit. So in the United States alone, over 1 million people enter the ER having to have claimed to have had their first seizure or seizure-like event ever in their life. And when they're finally referred to a neurologist, on that first visit, what happens is they ask the family and the patient, describe your event, what happened. They do a medical history, a physical exam, an MRI scan to look for possible abnormalities that might have caused an event that might be a seizure. And then they do this 20 to 30 minute routine EEG, as Rena went through when she saw her neurologist in her hometown. And what the doctors are looking for on that EEG are potential indicators of epilepsy. So there is no biomarker, 100% reliable biomarker for epilepsy. This is why this is challenging, right? But on the EEG, if they're lucky, they might see in that 30-minute window, not a seizure, okay, because they're just at rest, are these spikes or discharges that are abnormalities that indicate epilepsy, or other types of slow waves on one side of the brain and so forth. But over half the time, they don't exist, because these are sort of random events at the end of the day. And that 50% chance of seeing something on the EEG, also through visual inspection by a neurologist, lead to ambiguity, right? If the MRI is normal, I don't understand normal medical history, the event sounds like a seizure, but the EEG shows me nothing, what do I do? Well, typically they will prescribe medication, because the error of sending someone home who has epilepsy without anti-seizure medication is much more risky than sending someone who doesn't have epilepsy with medication and just those medications don't work for them. So with that said, when Rena had joined, we had just developed a software. We call it EpiScalp. EpiScalp takes in that same 30-minute EEG, and it doesn't have to have any abnormalities in it, and it'll produce a risk score. A score from 0 to 1, that if it's 1, you highly likely have epilepsy. 0, you're not likely to have epilepsy, unlikely. And one of the things I write up here, it's not in there, but is there's no requirement to have abnormalities in that scan to make a risk score, okay? So if we're not looking for abnormalities, what is EpiScalp actually doing? Well, we believed in the lab over several years that we might have found a biomarker, an EEG marker of what's going on in an epileptic brain that doesn't happen when you don't have epilepsy. And it's based on sort of network pathology. And we use that biomarker, we construct this biomarker from the EEG data and make a call. And what could be the potential impact of this? You heard her. If we can actually help clinicians make a reliable decision on that first visit, you avoid years of taking medications that you didn't need to take, getting the right treatment, and recovering much, much sooner. So here's a sample of the patient's very big clinical need that we heard the neurologist and epileptologist saying this is a big problem, but the solution was in how can we better read the EEG, and that's where computation and engineering can play a role. All right, thank you. And so I think our last panelist should be on Zoom, Dr. Steve Schachter. Steve, are you there? Tina, do we have Steve on Zoom?
Yes.
Perfect. Go ahead,
I'm
Steve.
here, thank you. Okay. Well, let me also thank Rena for sharing her journey with us. It's quite brave of her, particularly since functional seizures, the scientific understanding of them is at its infancy. It's almost like the way that epilepsy was understood over 100 years ago, when it was attributed to evil spirits or possession. today, the usual explanation is, well, it's all in your head. And that's of little comfort to people that are suffering so much from functional seizures. What I'd like to do is to emphasize a couple of general principles about listening to the voices of patients. And it's based on many, many years of sitting in the office, listening to people with epilepsy tell their stories, understanding that they're all unique and all to be understood in the context of the patient's life. I've published a series of books called Brainstorm series, which are the largely unedited words of people living with epilepsy or people who live with family members or friends with epilepsy. And the latest in that series are people who live with functional seizures. And you come away from reading those books amazed, full of new ideas of potential innovations, and so much respect for the patient's voice. But one of the lessons I've learned is that patients may have a tendency to please the doctor. And I think it's important in listening to patients that you establish a collaborative relationship with the patient, that they are a co-creator, as an earlier speaker said, and that you are not, that the doctor is not in the role of an authoritarian figure or in the pocket of pharmaceutical companies like you see on this white jacket, but rather that you are equal partners in figuring out the best approach for that patient. Where that conversation takes place is very important. I learned a long time ago that when a patient's in the office or in the hospital, that they're not as in touch with their daily struggles, with their unmet medical needs, as they are at home or in the workplace. And so creating an environment to facilitate the patient's voice is quite important. Also, the words we use are important. For people with epilepsy, if you say, how's your epilepsy? They'll say, it's okay. If you say, how are your seizures? They'll say, very bad. And so whatever the neurological or psychiatric condition is, we need to use words that resonate with the patient's thinking. Another important aspect is, as a physician, not to minimize or belittle a patient's experience because we're taught it's a mild side effect, for example. Oh, yeah, this treatment might give you mild double vision, but it shouldn't be too bad. Well, if you've ever crossed your eyes for more than a few seconds, it's very uncomfortable and very unpleasant. So we cannot underestimate the significance in the daily lives of people for consequences of treatment that we believe are mild. Likewise, the concept of invasiveness, and with apologies to Dr. Brown and Salzberg and other neurosurgeons in the audience, we have a tendency in medicine and medtech to use the term minimally invasive or non-invasive. Earlier, we saw a slide of a film that was put onto the cortex or on the dura, and it was minimally invasive. Well, it's minimally invasive if to the surgeon, but to the patient, it may not be minimally invasive or non-invasive. So we need to remember or catch ourselves before we use specific lingo that may be interpreted differently by a patient or their family. In epilepsy and in functional seizures, the concept of being in control of yourself is very important because The seizure, whether it's an epileptic seizure or a functional seizure, takes over control of the individual. And so being in control is a very important concept to people living with seizures. And in this era of medtech development in epilepsy, where we have things like the vagus nerve stimulator, deep brain stimulation, responsive neurostimulation, when you talk to patients, they have a concept that these devices may be usurping some control from them. Here is a self-portrait of a woman living with epilepsy. And by the way, all the art and sculpture I'm showing were created by people living with epilepsy. And in this self-portrait, she's trying to express the fact that she doesn't feel fully in control anymore of her body or her mind. So we need to design medtech for people living with epilepsy or other disorders where self-control is important, to design it in a way that gives the individual as much control over the use of that device as possible. And the last point I want to make is that the patient's voice may be different than the caregiver or family's voice. And that's particularly important in conditions like epilepsy, where a family member or a caregiver or a friend may be called on during a seizure to render help. This is a table from a paper where doctors ask patients and their caregivers, how long would you like to know in advance that you're going to have a seizure? And this is important because we're now in the era of seizure prediction, not yet commercially, but many, many groups are working on different ways to predict the onset of a seizure in advance, which would allow the patient to get into a safe place or administer a quick-acting anti-seizure medication. And for example, if you look at the line 1 to 6 hours, 18% of the patients said that would be fine and desirable for them, compared to 3% of the family members. And this just is an example of how it's important it is not only to solicit the viewpoint of the patient, but for those conditions that do involve family and friends, caregivers, to take their perspectives into account as well. So just a few very high-level lessons that I've learned and wanted to share as we think about the patient's voice. Thank you.
Maybe Linda, if you could let us know about timing.
I think we're done.
Yeah, we're almost done. Well, I'll
Take a few more minutes. That's
yeah.
okay.
All right, as long as you're good with it, like I'm good with it. All right. So thank you to all the speakers. I think that was absolutely fantastic in terms of the patient-centered focus. I really wanted to just kind of ask a couple of quick questions of the panelists in terms of how, number one, how can patient values influence and focus the direction of neurotechnology and development? And then I think more importantly, how do you try to empower your patients to be collaborators, especially in light of kind of that most recent talk there in terms of technological innovations and kind of coming in? You know, a lot of the advances that I think, you know, we're seeing are things that you've, some are observations, some were technologies, but then how do you actually be more proactive of engaging that patient at the forefront as you're kind of making your innovation?
Well, I think I'll talk as not the clinician. I heavily rely on my clinical collaborators because I don't have access to the patients, right? But only through my clinical collaborators I might have some access to them, but it's through just
months, maybe years of working with them and collaborating with them to understand not just their needs, but patients. So when we have access to patients, we ask a lot of questions, myself and our students, but really we heavily rely on the clinical collaborators in our center.
I think that, Rory, one thing is that it's not a one-way street, it's a two-way street. And that I look at the interaction with the patient as it's so important to start by listening to how they present their own history and story and what it is they're looking for as solutions in terms of degree of aggressiveness and intervention, and then presenting options rather than a single course. Now, the pendulum swings in society. It used to be in the old days, a doctor would just come in and say, hey, sweetheart, this is what you're going to do, and don't ask questions. And, there's no discussion, and they're like Dr. Schachter showed that cartoon. They're the king, and you just listen to them. And then the pendulum swung the other way, is that we just present options to patients, and they're terrified because they're not experts. We spend our whole lives studying these diseases and these problems. We know the nuances of the different treatments, and we know what works and doesn't work, and what sort of price they're going to pay with the consequences of what we're recommending. So the balance that I've found is to listen, because patients really do communicate what the problem they're trying to solve is. Firstly, you so amply showed, and then to present options, and then ultimately the question is, well, which direction? And to say, well, if this were my family, and to think that way as well. If this were my family, I would probably choose option number one because these reasons, although option number 2 and 3 are also viable alternatives. And then that really does both provide the guidance that the patient wants because they've come to a specialist, they want to know, but it also says that it's okay to choose different alternatives. And so I think that's the fundamentals, I think.
Yeah.
I just think also in considering like invisible struggles too, like I think for rural patients, travel is a huge deal. I know when I got into Hopkins, the amount of people that came up to me talking about how Hopkins saved their life and all these things, but they had to stay at a hotel, and we also had a fund. I remember one of my classmates was diagnosed with cancer, so we had a fundraise for him to be able to stay in a hotel, so his mom can stay in a hotel while he's in the hospital. So while listening to the patients, also thinking about like what other barriers are there in addition to the primary health challenge that they're facing.
I swear you just stole my question just right up under me. I literally was going to ask you about what are the challenges from your perspective that you saw, and then also, sorry, Dr. Pelker.
I'm going to go back to the patient relationship.
Yeah.
Mm-hmm. So how many people here have ever waited in a waiting room to see a physician and had to wait? Really? Unbelievable. Yeah.
I never have.
Yeah. If you come to our clinic, we are definitely not on time. And the reason for that is because patients travel. They often travel a long way to see us, and we're going to take the time for the individual patient that they need. And unfortunately, sometimes that's longer than what was booked, and we get far behind. But when the next patient who's been waiting a long time and is angry and so on, then gets all the time that they need, they usually forget about how mad they were. So what you start to realize is you have to develop that relationship. And the days of, I'm going to tell you what you need, just listen to me, those are long gone, and rightly so. One of the things we've learned, though, is patient has the disease. That's step one. Not you, the physician, and not you, the family, although the family does suffer with the disease, but it's the patient who has the disease. The second thing is, it's the patient who has the disease. And often in discussions, as we get to know our patients, we have to kind of separate out a little bit the patient's wishes, the patient's understanding from the family or other members who might be present. And a lot of times that takes time. Concrete examples, I can give you 2 recent ones. I deal with neurofibromatosis, which is this unfortunate disorder that produces multiple tumors. People think of the elephant man syndrome and so on as an expression of it. When we deal with this, and patients will travel from far and wide to get surgery for it, we realized that a lot of times the patient has a very different belief system and want system from the family around them. So for example, a patient 2 weeks ago who had a very large, ugly, horrible tumor that we had to remove. At the end of the discussion, I said to the patient, is there anything else? Family member piped up right away. What about this tumor that's sitting on the face? Can you remove that? Can you do something with it? It looks horrible. And I said, well, we'll talk about it. And then I said, we have some personal time that we have to spend with the patient, just one-on-one. Family leaves. We discuss that with the patient. By the way, that tumor on your face, is that a problem? Not at all. Get this big ugly tumor out of me that's the major problem. I'm not worried about that tumor on my face. Family members are who have to look at it, unfortunately, but the patient wasn't worried about it. Second example is last week, taking out a malignant nerve tumor, and it's a very malignant tumor. We have lots of research protocols. We are talking about that somebody from our research team will probably come just in the pre-op area to consent the patient for harvesting tissue, where we just tumor bank the tissue. Family member came to me and said, please don't do that. We don't want him participating. We know it's already so stressful. We don't want researchers coming and talking to him and adding anything more to this operation. But day of surgery, I kind of forgot that. The family member had said that to me. And in the pre-op area, got into a little bit of a discussion after I consented the patient for the surgery that one of our research coordinators may be coming by because we're going to potentially hold a piece of the tumor in our tumor biobank. I have never, in 35 years of doing this, had a patient refuse that. The patient, who's very invested in their disease, if there's even hope that they can help someone else, even if it's not themselves, are usually very thankful. So those discussions with the patient and understanding what's important to the patient, that's preeminent, always.
I was just going to ask one more question and then ask the audience. But basically, I just wanted to know if each of the panelists could just tell me either one obstacle that could be removed or one opportunity that could be taken advantage of that would make it easier for you to basically empower your patients to keep your innovations patient-centered. Don't start all at once.
Yeah, because I'm a completely different perspective. As an engineer building, like say, in my case, software, and being in academia as a professor, we are not in the current traditional setting of how we get judged, evaluated, and promoted. It's never about innovation or translation. It's about how many papers do you publish? What are your discoveries? Are you going out there giving academic talks? And so young professors especially are kind of discouraged to innovate, but yes, they're some of the best because they think out of the box. They haven't been sort of molded by the institution. And so for me, removing that barrier for engineers or just in general any academics to encourage innovation translation would be huge.
This is from a patient perspective, but the reason why I was actually able to go to Hopkins to get that EMU was because there was a pediatric neuropsychologist who would travel to the Eastern Shore of Maryland once a month and set up a pediatric concussion clinic. So I happened to get a concussion, and my doctors, because they didn't feel qualified to do epilepsy, what they thought was epilepsy at the time, epilepsy plus concussion management. So they sent me to the specialist, but because he, as a pediatrician, so he works at Children's National, Dr. Gerard Joya, because he traveled once a month, my parents and I were able to save up so much travel time, and he told us that the biggest barrier he saw with patients from the Eastern Shore is that they're traveling 3 hours there and back, taking days off work just so their child can get good care. And so what if he was the one once a month? He got a second home in our little beach town, and he's like, what if I'm the one once a month that can come and see maybe 5 patients a month? And that has alleviated so much burden. So I sometimes wonder, okay, where can technology and where can also physicians come into these areas that are underserved and show them that we do still care about them, that they don't have to travel all these distances. And I think part of the great thing about technology is now that with Zoom and all these other things, we can connect people all across the world. So I would love to see more of how we can make sure that bridges gap, or the gap is closed.
How many people here are engineers? And how many of you as engineers have ready, almost all the time access to clinicians? Yeah, so I would have loved to have seen the whole audience put up your hand. So one of the, I think, barriers is simply that barrier, where you've got departments of biomedical engineering or engineering up at the university on main campus. You've got clinicians in a hospital somewhere. Maybe some of those are clinicians who do have labs, but those labs are out in the hospital. We all know the water cooler story. Nothing works better than the water cooler, where when you are getting the water and you're just chatting and you have those discussions, great things happen from those informal discussions. So one of the things that I think is a huge barrier is the inability to have those water cooler discussions. And one of the great breakdowns is to get rid of that separation. So one of the things that my boss, my ex-boss, Henry Brem did was to do that. At Hopkins, we have our biomedical engineering group who are physically right there with the clinicians in the Meyer building at Hopkins, where we're seeing each other every day, where we can walk and literally talk to each other every day. I'm looking at Amir down there, who's a biomedical engineer, who the contact is immediate, it's daily, and the ideas get shared. To me, one of the greatest barriers is my inability to know what the right hand is doing if I'm the left hand and vice versa, to know what's going on, down the street or on the other campus in biomedical engineering or at APL or something like that. If I don't know about it, I can't take advantage of it. And if they don't listen to the clinical questions I have that I struggle with my questions, or my patients rather, we can't have that back and forth. So to me, that's the barrier that needs to be taken down. You have to be working together in close proximity to share ideas.
Dr. Schachter, I don't know
Yes,
if you,
thank you.
wanted to comment on it or if you're still online.
Yes, I'm still here. Can you hear me? Can you hear me?
If not, I was going to say I'll let you have the last word, Dr. Brown.
Okay. Dr.
Can you,
Schachter,
I'm
maybe
sorry,
come back.
can you hear me?
Beautifully.
Okay. I'd like to, oh, and there I am too. I'd like to emphasize the last point. I think that was Alan made it. I'm biased because my practice was entirely people living with epilepsy, difficult to control seizures. And if I had to summarize what I've learned is the biggest barrier, it's communication. It's effective communication. We need more time in the office. I have found it helpful to socialize with patients, with people living with epilepsy. For example, at epilepsy foundation events, or fundraisers, or whatever it might be. The more you talk with the patient and the family, the more you're in a position to choose the options, as Dr. Brem said, choose from among the options that, and allow the patient to choose from among the options that makes most sense from them. And I'll just finish with, I got in the habit when I first met a new patient to say, the very first thing I would say is, how can I help you? Leave it very unstructured and go from there. I think young trainees have a tendency to fire off a series of questions that they're taught to ask. And I think it works better to allow the patient to focus in on anything they feel is important in terms of effective communication. That is, to me, the most difficult barrier. And all the trends in medicine now work against that, limiting time in the office, making the doctor take a review, ask questions that are unrelated to the patient in order to be able to bill a certain amount. I think we could do much better as a profession listening to our patients and building that therapeutic alliance.
And Dr. Brem, final words.
Thank you.
I know we're a little bit over time, but yeah.
I'm going to speak very quickly. We lived through, people have forgotten already, but we lived through one of the worst medical crises in medical history and humankind history through the COVID epidemic. And, but some good things came out of that catastrophic event. One of them, of course, maybe the most important, is the RX program by the NIH, which accelerated technology development that led to vaccines and led to rapid diagnosis of COVID that helped abort that horrific pandemic. And therefore ultimately led to the Neurotech harbor and the Blueprint being created so that we can do that for the brain, spinal cord, peripheral nerves, and rehab and so on. So that's a good thing that came out of that experience. The other thing is really building on what Rina mentioned is telemedicine. That technology, the engineering was there before COVID, but most of us never paid any attention to it or used it, and certainly patients didn't. That revolutionized the way we took care of patients. We all of a sudden started being able to see our patients again by telemedicine. Post-COVID, I think that does, Rina, what is so much of what you asked for, which is it gives access to people, to specialists and people to think about their problem. It doesn't matter where in the world they are, and it's instant access, and you get their full thought and concentration, and therefore it benefits. To me, that's such a wonderful thing as a brain tumor specialist, that patient doesn't have to spend a fortune and come to Baltimore and so on and so forth, but rather in 10 minutes in their living room, they can get the best of our expertise as to whether we can help or not help. The problem is, the frustration is that, and so, yes, to Dr. Schachter's point, there's nothing like being in a room with a person, and that's the ideal environment. And we now give everybody an option, and it's split. Half the people love telemedicine, half the people love it in person. But what's happening is, since COVID, is all the legislation that enabled us to be able to do that has started disappearing. And so now states have gone back to legislation that's saying that a Hopkins physician or Baltimore, Maryland physician can't talk to a patient in Tennessee or in Oklahoma or some remote area because it violates state laws. You're practicing without a license and so on. So I would think very pragmatically that one thing that's very frustrating that gets in the way of innovation and best care for patients would just be to break that down. That's a legislative, man-created problem of blocking people from having access to the people that they want to see. And we could fix that. That's an easy fix.
I just want to thank all the panelists.
Washington, D.C. It's easy. As soon as the government stops shutting down, we can fix that problem.
Exactly. Well, I wanted to thank all the panelists, and thank you guys for watching and listening.
So we are almost done with the morning program. Some of you might have to take a bio break. Just go ahead. So just very quickly. And I'm here to introduce the next session. I'll be sitting here with 3 colleagues. My name is Paolo Bonato. I serve as co-PI on the Cinta hub, for the Cinta hub. In Boston, led by Steve Schachter, who was just part of the panel discussion, as I trust you notice. I'll be accompanied here by 3 colleagues, Yusuf Yazdi and Mike Dempsey, who are in charge of our innovation programs, and Elise Camera over there, who is going to join us on the podium. You might want to see the slides and stay on the seats, but they will participate. So I'll make a short introduction about the type of conditions and focus on applications that we receive over the last few years since we actually started this initiative. And then Yusuf and Mike talk to you a bit about how the program actually works, the process by which the applications are considered. And then Elise will actually tell you a bit about impact through a survey that she analyzed very recently. Here we are. So this is a list of conditions that are the focus of individual applications. A colleague of mine, I'm an engineer by training, a colleague of mine in the department, oftentimes we discuss numbers, and he likes to joke with me that I pull up a wall of numbers. So this is a bit of a wall of word, right? And the key here is, like with numbers, you want to focus on sort of take the big picture, but then focus on individual conditions. And so I don't expect you to read all the conditions here, but here what we wanted to do is to give you a sense of the breadth of different applications that we're focused on. I really appreciate the previous panel when there was so much emphasis on really driving solutions on the basis of conditions. And I was thinking, as I was listening to the keynote speaker earlier, that each one of these conditions is a bit of an elephant that you have to take in small bites. And so, you know, for my specific work, I work at Spalding Rehabilitation Hospital. There is a lot of focus on my group on stroke. And there is such a variety of presentations, but so much that technology can do in this context. Over the past almost 25 years that I've been at Spalding, there has been such a dramatic change in the way stroke is managed in the acute phase. And so, and we see that oftentimes, thanks to technology, research, and on the biology side, and we see that in the clinic. Despite the fact that I'm an engineer, as Dr. Bram was mentioning earlier, this interaction between engineers and clinicians is very, very fundamental. And I'm privileged as an engineer to actually work in a clinical environment and see, witness on a daily basis the needs of these patients. And this applies to all the conditions that you see on the screen. You can think of conditions, neurodegenerative conditions such as Parkinson's disease. You can think of cerebral palsy. And we do have a pediatric program, and we do see these family comings with kids affected, either their motor system or their cognitive function and their ability to communicate with others and grow and have a fulfilling lives. There are, of course, very dramatic conditions, as the brain tumor that Dr. Bram has discussed earlier, and traumatic accidents that really cause major neurological conditions, as the somewhat beautiful from a scientific point of view examples, but devastating from a clinical standpoint, that was actually presented earlier by our colleagues from Johns Hopkins. These conditions affect a substantial portion of the population as a whole. Some of them, from one side, fortunately, they affect a relatively small number of individuals, but with devastating consequences. The fact that they affect a limited number of individuals doesn't really matter because of the depth of how dramatic is the impact on the lives of these individuals, the conditions they're affected by. Some other conditions affect a humongous number of individuals. We can think of the general category of pain, related to a variety of different conditions that have led over the past few years to the opioid epidemic. We can think of stroke, again, really close to my heart, as a condition that affects about 5 million survivors today in the US, with a variety of presentations from very mild impairments to really devastating impairments that affect both motor, cognitive, speech, communications, and so on and so forth. This translates into billions and billions of dollars. I recently led a panel discussion through IEEE that many of you are familiar with, on the impact of research on not just the healthcare impact, but the economy, right? And you can see here the large amount of money spent actually addressing the clinical needs of these individuals. You can think of lack of compensation, lack of the ability to actually be part of the community in an active role of those individuals. And you can see money really piling up because of that. So even a major investment is the investment of NIH in research when compared to the type of impact that we can have economically and on money saved, where in fact we're spending a humongous amount of money for taking care of these individuals, right? And so we hope really excellent results, at least in some cases, unfortunately not as good in others, right? It's almost like a drop in the bucket. These are examples. Again, this is a wall of words, right, as my colleague would like to call it, right? So, of projects, right? I don't want you to focus on any specific ones at the moment, right? And so perhaps you can pick one in this list, a very long list of projects that we've been supported that's very close to your professional activity or close to your heart because you might have had a family member affected by some of these conditions. I was really grateful for the testimonial that Rina, that Dr. Sarma brought today, her sharing of her clinical experience, her journey, right? So, and this is among the type of projects that we've been supporting. Can we do a better job by properly diagnosing epilepsy so that we provide effective interventions from the very beginning as opposed to go through the journey of figuring out, as Dr. Sama has mentioned earlier, that the patient was misdiagnosed. The same happens in a variety of different conditions. In Parkinson's, that's not actually unusual, that even when it refers to specialized center, oftentimes in the early phases, we have to deal with the misdiagnosis of these patients, right? And a lot of referrals, in fact, are based on that. Can we do a better job by developing technologies, you will see in the titles here, to improve motor functions in these individuals with neurological conditions? Can we prevent the consequences of, for instance, brain hypoxia at birth by developing technologies that actually allow shunting in a more effective manner? So these are the type of projects that we have been supporting over the past few years. And then Yusuf, I think, is coming to the podium to tell you a bit more details about where the projects came from and the way they were managed.
Thank you, Professor Bonato. Really inspiring morning, and you're getting a picture of the whole journey, starting from a clinician observing an anomaly in their patient, like Dr. Brem and Dr. Belsberg mentioned, but not letting that go, taking that hint and digging into it, asking why. We have this expression in the design about asking the 5 whys, repeatedly asking why. What is the root cause of that anomaly that you observed? And digging into that. And from there comes the most amazing opportunities for life-saving innovation. But that's just the beginning of the journey. And Dr. Banerjee went through her example of the many issues that need to be addressed to be successful in this. And Dr. Sarma mentioned some of those technological innovations that are required to turn that inspiration, that moment of that observation into something that turns into a real product. And this program, this Blueprint MedTech program, is really all about that. It's about taking that inspiration and doing all the 1,000 things that need to be done to turn into a real product in the hands of clinicians, saving lives. Dr. Bonato mentioned the wide range of clinical areas that this program is addressing and the wide range of technologies that the program is tapping into in order to create those amazing solutions. And the program has also been very successful in tapping into a wide range of innovators across the United States of America, across many states who have proposed projects to the program. And the funding has also been very well distributed across the country. It's something we're very proud of. So through the 2 hubs, we're able to manage effectively a wide range of projects. And if a project comes from one of our hubs as the origin, because we have this dual hub model, we're able to avoid conflicts of interest and have the alternative hub managing that project. In addition, the projects are not just, the funding is not just going to academic innovators. And this is one of the very special aspects of this Blueprint program that I think the NIH has really created and designed a program that is incredibly innovative in the way and where the money is being spent. The money is being spent primarily in academic organizations, in startups, and in small businesses. And small businesses meaning usually newer spin-outs. There's a few larger companies, academics, not-for-profit organizations, labs, et cetera, that are getting funding, which shows that the review committees are not biased toward one or the other. They're not excluded, but I think we're very proud of the fact that the funding is going to many ongoing operations and startups and supporting them. And the previous slide was about the ones that were proposed, and these are the ones that were awarded. And the journey, I think this is one of the very innovative aspects of this program. To start from a proof of concept and take it all the way to the point where the project is ready for the critical phase of testing it in real patients, evaluating in real patients. There are many, many steps in between that need to be addressed. And typical NIH programs, typical academic programs do not address these. They do not really prepare projects. And this is where a lot of wonderful innovation that could be life-saving is lost. And that's why the design of the Blueprint program is to help the teams navigate this pathway. And you'll hear from my colleague Mike about the resources that the program provides to enable that to happen. But there are many, many things. I won't go into all of them, but I just want to point out a couple of things that are critically important. One is the philosophy of de-risking, and that is to look at the solution that you're proposing and say, what are ways that this thing could fail? And those ways that it could fail come from the world of medicine. They could fail medically. They could also fail from a business perspective. They could also fail from a technical perspective. And they could also fail from an entrepreneurial perspective, meaning the team itself, there's issues that come up. And we have dedicated mentors to help the team identify those risks and address those risks. Another important element of the program is accountability. This is a very unique aspect. I don't know how many here whose careers are built on NIH funding, but how much is there of this? How often have you had to have quarterly reviews of your proposal, of your progress? And how often have those quarterly reviews been meaningful in the sense that the funding could be pulled if you don't achieve your milestones, if you don't have a good explanation for why you haven't achieved a milestone? So I think this is another very innovative aspect of this program, and I think we should all commend the NIH for designing this program and for implementing this program. It really is a breath of fresh air and fundamentally different from historically how NIH has funded research. And I think a critical part of that are these quarterly reviews and annual reviews that we take very seriously. The people on those committees are not just academics. They're people who focus their lives on innovation in medtech, people from industry, from academia, investors, et cetera, on those committees. And they help the hubs and they help the NIH make decisions about continuing the funding or killing the funding, or sometimes it's about pivoting and correcting some serious mistakes. And the process of selecting funded projects is also, I think, something that the Blueprint program can be very proud of. It's a very rigorous process, and it involves assessment of criteria in all of those categories I mentioned. Not just finding something that's amazing science, not just finding something that's amazing breakthrough medically, but it also has to be something with a solid potential to actually make it into a viable, marketed product being put into widespread use for many, many patients in order to achieve real-world impact. So I think those criteria are brought into play from the very beginning of the process. It's a process designed to iteratively look at deeper and deeper into the project and answer those questions, to give the NIH, who has the final word on approval of the projects, the confidence that this project, if funded, at least it has a solid potential both for clinical impact and commercial viability. So with that, I'd like to invite my colleague Mike Dempsey up to talk about one of the critical components of the program, the mentorship. Thank you, Mike.
So in general, I'm going to talk about the resources that are available to the teams, but I'll immediately disclose a bias that I think the most important resource that's available is our people. That's our secret sauce. And the origin story here is in March of 2020, when the country was shut down and NIH tapped CIMMIT, my group in Boston, specifically, and the POC-TURN network more generally, to think about COVID tests. So we started calling friends of Mike or friends of Bob or friends of Andrew, and everyone wanted to help. And most people were sitting at home trying to make do with their time. And we ended up with an incredible network of people who were very experienced in the medtech industry and all trying to give back. In that first phase, it was to solve COVID testing. But as has been mentioned, that has evolved into an ongoing corpus of people who are willing to work with innovators to make them successful. So let's go through some of the details. The structure is we have team leads. They are someone who is paid for by NIH, who is a resource to the teams. And you can think of them as a for-hire CEO or a for-hire general director. They don't do what I call drive-by consulting. Those of you that are in academia probably have the benefit, plus or minus, of your tech transfer office, who gives you great advice. You probably have some alumni network, some investors. They come in and they say, oh, well, like you have to figure out your intellectual property strategy. And I'm an IP lawyer and I'll give you 30 minutes at 2:30 on Tuesday to help you. Or I'm a product developer and here's what you have to do for a QMS system. Or I'm a regulatory expert, here's 45 minutes on a random Monday. And you as the innovator are like, okay, I've got $10,000, 2 hours, and I can't figure out what to prioritize. I don't know what is more important. Is it the IP? Is it the technology? Is it the voice of the customer? So what the mentor brings to these teams is the experience. The average team lead has got 35 years of experience in industry. Something like 92% of them have started companies. 100% of them have had P&L responsibilities. And they're doing this to help the patient of the future. The team leads are supported by PFs, project facilitators. They tend to be younger in their career, and they're the bird dogs. They're the people who are working with the team day in and day out. All right, you said you were going to do this. Where is it? They're running the project management. They're interfacing between the team and NIH and the CINTA and NTH groups. They're the folks who are getting the stuff done. Here's a little bit more about the TLs, the team leads. One way to think about it is these people are, as a phenotype, folks who have been successful in whatever they've tried to do. So they've successfully exited multiple startups. They've successfully run major corporations. They are tenured professors. They are, in a position where they're giving back. They're giving back to the next generation. And they want to give back in a way that their advice is respected. They want to give back in a way where advice that they give or guidance that they give is followed up on and then it's measured, and they want control. We have this notion of, are the teams coachable? If the teams aren't coachable, if they're not accepting the advice that the TLs are giving them, we tend to look disfavorably on that, and it's hard for the teams to go on and raise more support and funding from us. An example, Mike Harsh, he's got a very corporate picture there. Former CTO of GE Healthcare, had $1 billion product line responsibility, hundreds of employees. You know, fairly typical for the TLs, has a degree in engineering and then gone on and done more stuff. I don't think Mike is in the audience today. Claire is another pretty corporate picture. Medtronic, 31 years, MBA, another mechanical engineer, works really, really intimately with the team. Here's the rebel of the group, Andrew DeMeo, PhD, started multiple companies. During our experience from 2020 to 2025, Andrew left our little nest, started and sold a company, came back into the little nest. So these are the kind of resources that you all are getting as winners of the Neuro Blueprint program. There are other resources that are available. Money is one of them. You know, when you don't have money, money is really all you care about. I get that. I was the PI for many years. So yeah, mentorship, that's great, you know, but I really want the money. And by the way, why is the contract taking so long? Where's my notice of award? I completely get that. But what we find is after the money flows, then it's the mentoring that makes the difference. That's what causes the project to be successful. There are a whole laundry list of available specialized resources that are available to the teams. I think in the next slide, yes. This is a little more telling. Basically, a lot of people need help with regulatory advising. A lot of people need help with manufacturing of prototypes. Lots of business development work. And I'm surprised this is as low as it is, but one of the resources that I would encourage all the teams to take advantage of is understanding where your reimbursement is coming from, understanding your pathway to payment. Because without that, you don't have anything. With that, I'll hand it over to Elise, who will talk about some results.
Hi, everyone. Thank you so much for hanging in there with us. We're almost there to lunchtime. I believe I am the last speaker standing in the way between you and lunch. I'm thrilled to be here today. You've heard a lot about the program, the mentorship, and the resources, and building this ecosystem of support. But what does that mean, right? The focus and the theme for today's annual meeting is moving from innovation to impact. And so how do we evaluate impact? One of the ways we did this was to create a comprehensive survey that was given out to the team leads. Thank you so much for filling it out. It was, they did it over the summer, towards the end of the summer, at a busy time of year. We had 14 teams respond. 8 of those teams started in the first year of funding, 2023. For reference, there were 9 teams who started that year, so this is a pretty incredible response rate. 4 responded who started in 2024. There were 6 teams that started that year. And then already 2 teams that just started this year have responded. And looking through their results, I was very happy to see that they've already made progress that can be measured and evaluated. One more thing, this is very early, right? This program's been going on a little over 2 years. And so take that into consideration when you look at the results. But I think you're going to be impressed with what you see. There were 4 broad domains that were evaluated by this survey: regulatory, technological, intellectual property is part of that. We have clinical and then commercialization. So this first one, regulatory, when you have an inventor who has a new technology, regulatory is often kind of one of the scariest items on their mind, like how are we going to get through the FDA? You saw that the most utilized resource is regulatory consulting. Part of this is a lot of teams want to wait until their product is finished before they ever talk to the FDA because it's scary, right? Once you talk to them, your testing plan, like they know what you're doing, you're on their radar and they're afraid. But we're encouraging teams to talk to the FDA at an earlier point in time. And for devices, this is also encouraged by the regulatory experts. So 8 out of the 14 teams who responded have had at least one formal interaction with the FDA since starting in the program, which is incredible. 3 of those teams have had 6 or more FDA interactions. So some projects require a lot more feedback than others, but that's encouraged, right? And so you're encouraged to do these as necessary, and your mentor will walk you through that, and the mentors have been walking teams through this. The second group of bullet points really speaks to the impact of the projects that are being funded by this program. So many breakthrough devices, right? So they can get support for applying for breakthrough device designation, either through the program, they've already applied for it, some have already gotten it. So this gets them more one-to-one and FaceTime with the FDA. I can't see this. And then regulatory preparation. Oh, okay. So, before you're ever allowed to test your device in a human being, you have to get permission from the FDA through an investigational device exemption. There's so much work that goes into this. All those regulatory consultants are helping you set up a quality system. A lot of academics don't know what a quality system is, or they know that it exists, but what is actually involved, all of the work required to put that together. So you need that in place before you go to the FDA with your pre-IDE. It's encouraging that teams are prepared to submit their pre-IDE. One additional team already has their IDE application ready for submission. Okay, so clinical. This is something we've heard a lot about today with the patient-centered innovation. Maybe when you start a program like this, you think, I have this device and we're going to test it in this patient group and we're going to demonstrate safety and efficacy, which is absolutely the main point still. But through that process of designing your clinical trial, it's critical to think about who are your stakeholders, right? It's not just the patients and the clinicians, but it's their family, it's patient advocacy groups, payers, investors, right? At the end of your clinical trial, you want to make sure that you have designed your trial appropriately such that you're adding value for each of these stakeholders. And so half of teams are actively engaged in drafting their early feasibility study clinical trial while taking into consideration all of these elements that I was mentioning. What else? Oh yeah, and site preparation, ethical approval, teams are in various stages of submitting and getting approved and identifying clinical trial sites coming after your IRB. IP, this is a fun one. So another maybe misconception, I'm not sure, is you come in, you have your invention, you patent it, and then you test it in patients. But one of the interesting aspects of this survey is we asked the team leads to supply us with 3 top successes that the teams have met with, as well as 3 challenges. And one of them is, I would like to quote one of the team leads on this, Tony Curro, that submitting, you're getting your patent protects the novel features of our device, right? Ensuring long-term value for translation and commercialization, and provides a strong foundation for discussions with both investors and industry partners. But then we also heard about this culture of surfacing issues. I love that phrase from Dr. Banerjee. That was incredible. Another thing that comes up through reading the challenges is, oh shoot, our device didn't work the way we thought it would in preclinical tests. And so you need to adapt your device, you need to change it, potentially invent new supportive technology around that. And I think these numbers speak to that. So not only were 11 new patents issued, but there are new invention disclosures, right? Before teams ever came in, they would have already disclosed this to their tech transfer, but the fact that they continue to be submitting new inventions means that they're benefiting from the supportive ecosystem of the program. They are inventing workarounds and potentially new technology as well to make sure that they're meeting the needs that they have identified. Economic development, a few teams that started as an academic team have launched a company. And then economic development, this is early days, I'm going to keep saying this, but teams are taking this money and they're adding employees. And so I expect these numbers to go up dramatically going forward. And then finally, the commercialization, the funding, and the investment milestones. Teams are meeting with investors. Being part of this program is evidence that they are in a select cohort, and that in itself is appealing to investors. The number of meetings ranged dramatically from 4 to more than 100. We also heard many and lots, so we'll have to fix that in the survey going forward. Lots of new strategic partnerships are being formed, have already formed as a result of participating in this program. Of the teams who have raised additional non-dilutive funding, the average amount of that was 1.2 million, and small data points, right? Like average is probably not the appropriate statistic to be presenting here, but it's still encouraging that through participation in the program, teams are able to attract not only additional non-dilutive funding, but equity funding as well, with an average of $2.7 million raised. And that's it. This is our last quote. But basically what I wanted to conclude with is that what you've seen here today and what we've been talking about is that the Blueprint MedTech model of supporting neurotechnology innovators through federal funding, expert world-class mentorship, and resources, it's really having an impact. We're working to accelerate translation of these life-changing neurotechnologies. That is all. Thank you for your attention, and thank you so much.
Really quick announcement, not going to talk or give a speech or anything, just wanted to thank all the speakers of the morning. We have a really nice long lunch break. Lunch is a hot lunch straight ahead where breakfast was, and there's nice tables inside. Take a right.
Hey, everybody. Good afternoon. If everybody can get seated, we'll get restarted with the program. If everybody can get seated, we'll get restarted with the program. Thank you. Thank you. Hello. If everybody can get seated, we're going to restart the program very shortly. Thank you. All right. All right, good afternoon, everyone. Hope everybody enjoyed lunch. So now we're back, a little behind schedule. We'll try to catch up as possible. So my name's Santosh Venkatesha. I work here with the Neurotech Harbor team, along with many of my colleagues. As you heard from the program right before lunch, as Yusuf, Paolo, Mike Dempsey all shared all the great metrics about, how we recruit teams. From our solicitations, we receive applications all over the country. The funnel in terms of reducing, coming down from pre-proposals, applications, eventually awardees. Along the way, we run a program that's called the Seedling Program with Ivar and myself. He'll be speaking at the tail end of this panel session. You'll hear from him. So just wanted to highlight, beyond the information that is already shared about the program, the Seedling Program is an opportunity for teams to receive a $50,000 award for over 6 months. It's essentially our accelerator program that runs within the Blueprint hubs that's really aimed at trying to address key gaps that are identified in teams during our review process. So these are teams that are seeking optimizer funding, but reviewers have identified perhaps a gap that's in their clinical indication in terms of what's the proper beachhead market for their particular solution. It could be regulatory strategy that they need help. They haven't considered the input from reimbursement specialists. A number of different gaps that could land them into the Seedling Program and be diverted into our program for that, essentially to work on those gaps. And as you've heard, we have the same mentors that were presented in the earlier morning talk, work with these teams over the course of 6 months and provide their input along the way. And we have various programming that we have within Seedling Program that's offered throughout the 6 months as well. But I just want to highlight that, from an outcome standpoint, the most relevant thing to share today is probably that 11 optimizer awards were recently given over the summer. That's our latest cohort of full optimizers. So these are the teams that receive full funding up to sometimes $500,000, $1 million a year over 3 to 4 years. Of those 11 awardees, 7 came from the Seedling Program. So it's turned into quite a pipeline of wonderful projects, wonderful teams. And today, we wanted to steer this panel into a different direction, have some of the awardees and innovators come up and present. So we have 3 teams here with us today. And we'll just start off with Dr. Amir Mabachi from Hopkins. He's got a project he'll talk with you about, and we have 2 more speakers after him.
Thanks, Santosh. Thanks, Ivar, for the invitation. Luis, if you don't mind releasing the screen.
You want a mirror or extend it?
As long as it's sound. Thank you.
Okay. Let's see.
Perfect. Thanks, Luis. Good afternoon, everybody. My name is Amir Mabachi. I'm an associate professor at Johns Hopkins Neurosurgery, affiliated with many engineering departments as well. Just to be mindful of your time, I'm going to start my timer. So today we're talking about a neural implant for patients with acute spinal cord injury. If anything we talk about resonates with you, feel free to check out Hopkins Medicine. If I can figure this out. hopkinsmedicine.org/neurosurgery/happiest. This 1-minute video can tell you way more better than I can. So just bear with me and let's take a listen together.
Paramount for traumatic spinal cord injury, SCI, yet few diagnostic and treatment tools exist. Despite the vital significance of tissue perfusion, there are no reliable methods for assessing blood flow and preserving at-risk tissue post-operatively. A new class of wearable and implantable ultrasound devices to help SCI victims is being invented and developed by the Johns Hopkins Heppius Interdisciplinary Team. One of these, the multifunction spinal cord implant, Music, is an ultrasound sensor implanted over the dorsal spinal cord during a decompressive laminectomy. Music is capable of A. High-resolution imaging of microvasculature, B. Continuous monitoring of tissue perfusion, blood flow, and autoregulation, and C. Enhancing blood flow through use of therapeutic ultrasound. At Johns Hopkins Heppius Innovation Lab, we believe in the inexhaustible potential of sound in medicine and neurosurgery.
Great. Now that I told you what I'm about to tell you, let me dive in. So, as I've talked to about 30 key opinion leaders in the field, here's what they're telling me. And I do want to acknowledge Tanya Fernandez, our mentor, has been on most of these calls with me, especially the ones that went wrong, just being there for each other. These key opinion leaders are telling me that when a patient has spinal cord injury, car accident, motorbiking, battlefield, NFL, so on and so forth, hockey, they come into the clinic, they're taken to surgery. These surgeries are called decompression because swelling happens in any in any trauma, including neurotrauma. As you think about traumatic brain injury, as you think about stroke, as you think about spinal cord injury, time is brain, so is time is spinal cord. So here's what the surgeons are telling me. Their patients look good in the operating room, good. But I don't have to tell you, when they close their patients, not everybody is doing well, right? So something is happening to these patients when they're closed. So wouldn't it be nice if there was a gadget that allowed you to kind of understand whether these patient conditions are stable, getting worse, or getting better? So we picked blood flow as a surrogate for that, and I'm going to use perfusion and blood flow interchangeably. What I essentially mean by perfusion is small blood flow in small microvasculature. And so when I talked to the surgeon that we started this lab together, Nick Thitter, he mentioned that when these swellings are happening, what we do, our patients come in, we take these bony landmarks out, these bones called lamina, so we do laminectomy, and we let that swelling happen. In rare occasions, also, not in rare occasions, in parallel, they also drain this CSF, cerebral spinal fluid, that's meant to protect the spinal cord and brain. They can increase the blood flow. If you overdo that, though, the patient can go blind, the patient can have stroke. If you under or overdo that drainage of CSF, that can have major complications as well. I was in the room with Nick. He said, I know just how much to drain this CSF. He walked outside. All the chief residents who have done this for 7 years are like, this is very subjective. We need a device to do this better. And so from that point onward, my mission with Nick was to quantify this. So Nick encouraged me to think about it this way. We're not changing anything from the workflow. He's already there. We're right on top of the spinal cord. What if we use a little sensor that allows for monitoring and potentially modulating the blood flow at the site of injury? Remember I said part of the standard of care, the treatment exists and people increase the blood pressure? Well, that's a very systemic parameter, right? When you increase the blood pressure, a systematic parameter, physiological parameter, you just don't know what's happening locally. And what we want to know is, can we have a better parameter that we quantify things locally? That's what this device aims to do. And so let me summarize what I did by just showing you the prototype we built. So our team has secured, or did secure, $13.5 million from DARPA, Defense Advanced Research Project Agency, about 4.5 years ago. And the outcome is what you saw. I happen to have one of these in my pocket in case anybody wants to have a look. It flexes, sits right on the spinal cord. it modulates this, monitors and can potentially modulate the spinal cord blood flow when the patient is after the surgery in the ICU settings. That's when most of the surgeons are losing sleep on how their patient is doing. That's when things can go wrong and are going wrong. It even happens at teaching hospitals such as Hopkins. I can only imagine what happens elsewhere. So the device was built over the past few years after this form factor. When you look at the top left, the electrodes that are FDA approved. And when you look at these, I want your attention to these gray structures right here. The distal end measures the blood flow at the healthy side as a reference, and the proximal ones measures the blood flow at the injury and the secondary injury when the injury starts growing. So the injury, the penumbral neighboring zones compared to the healthy ones. And here's how it looks like when you implant this in a pig. And so we monitor that about 5 to 7 days. We've gone as far as 15 days as well. Warning, there's blood. And I'll probably, in the interest of time, I'll skip through it, but I just want you to see how much this fits into the workflow. This is the outcome, which I can show you a little more. This is a demo on a moving pig. Keep in mind that patients in ICU, human patients, are not moving. They barely are moving, and they have a lot of cables attached to them. So believe it or not, doing it in pigs is actually more difficult because we have been in situations whereby, our vets and our ethicists tell us to go for partial injury. We are monitoring them 15 minutes onwards, so the pig gets up, runs around, sees a snack, pulls the whole cable. But what we want to do is we want to be able to see the spectral Doppler that I wish we were able to hear the sounds, but you kind of have a sense of these blood flow. To somebody who's trained, a neuroradiologist, this is every day. But to you, you may be like, why am I looking at this? So I'll address this in a little bit. So here's what the outputs look like. On the top right, you see our AI, machine learning algorithm, detecting the hematoma, which by some accounts is too late if the patient has that. But the ability to see whether there's hematoma, whether it's stable or growing, that's a key factor. You also, I want to look at this colorful color map. I'm not sure if you see, but I'm hoping you see a tiny little bit of a microvasculature right in the middle. Typically, when people want to see microvasculature, they actually inject contrast agents, which when we talk to FDA, as part of our TAP program every month, they mentioned if you want to do continuous monitoring, continuous injection of these contrast agents, that's a big no. So DARPA pushed us hard to be able to monitor these microvasculatures without any contrast. And when we did finally got to that, they actually said, we didn't believe you're going to get to it. But then they didn't share that until we got to that point. Again, you might say, so what? I don't want to look at the imaging. Even radiologists agree that ultrasound is hard to read. But if we were to show you a diagram like this, that day 0, day 1 to 4, up to 14, the patient's blood flow velocity is stable, getting worse, or getting better, what you want to know, and this should be easily to everybody who is in the course of the patient treatment, what you want to know is whether the patient blood flow locally is stable or out of control. So I want to wrap up by mentioning that we received breakthrough designation pathway January 2023, and we've been part of the U.S. FDA TAP program, Total Product Lifecycle Advisory Program. They've been phenomenal. Tanya and I were just talking to them last week, and they mentioned to us, and they put it in writing, that if you do this many pigs, healthy, controlled, you're able to get to IDE, which is exactly what we need to get into first in human. In terms of competitive advantage, when I first started this, I realized that there's nobody out there who measures the local blood flow of the spinal cord right at the site of injury. And that's usually when people say you haven't done your homework. So, we have gone out and about, talked to many different people, and since then we've identified some of the other products that can be comparable elsewhere in other organs. We have 12 different patent applications and a team that's behind this. These are the summary of everything we addressed through the seedling program. Very helpful. I think a few things I want to mention is that we reduced this to 1/10 of the price and 1/3 of the width so that it can be miniaturized. We were asked about taking the device out in an ICU setting as opposed to surgeries. that was addressed. We did many more studies looking at what does this mean? Is there a clinical outcome relation to this? So on and so forth. I'd like to wrap up by acknowledging the team. Nick Theodore, our CMO that I mentioned, as well as Tanya Fernandez, our mentor as well, our Blueprint. We happen to have a very close industry partner, reimbursement consultant, regulatory consultant. And as I mentioned, we've been talking to about 30 key opinion leaders, none of them who are very happy to have their name and reputation behind what we're doing. And with that, I'd like to thank you for listening.
Thank you.
Thanks, Amir. Thanks, Amir. Really great. So next up, we're going to have Matt Johnson from Autonomous AI.
No audio. Yeah, thank you.
All right, well, thank you so much for the opportunity to present. Really exciting to be here today. I was inspired by the morning sessions. And so I'm going to tell you a little bit about what we're developing at Autonomous Medical Technologies. So the development of this technology started in 2019. There was a need recognized by the DoD to essentially bring the ability to provide advanced ultrasound guided procedures near the point of injury. This is due to the difficulty with evacuating patients to places with higher echelons of care. And specifically, they wanted to be able to provide rapid central vascular access in the battlefield and do this with lightly trained medics and do this to enable various life-saving interventions. Could be rapid fluid resuscitation, advanced catheters, and other types of technology. As the team worked on this, it was immediately recognized that medical expertise is not just a problem with access in the battlefield, but it's a problem all across the country. Sometimes in major hospitals like Mass General Hospital that we partnered with in this work, and that the work spun out of, sometimes in rural care centers. And so with that, the company was formed in 2022 to commercialize this technology. And it was really formed to democratize access to ultrasound-guided procedures. As we spoke with about 100 users, we came up with a very long list. Vascular access was our first go-to because of the initial work from the DoD and widely recognized need. However, as we spoke to people, they would say, hold on, could you use this for nerve block? Well, hold on, what about joint injection? We came up with a list of about 20 and went through a long process of sort of down-selecting potential applications. The one I'll be speaking about today is one I'm really excited about because of the potential impact to patients, and that's nerve block. So to get specific, and this was a major pivot that we had as a result directly of the Seedling program, there are 320,000 hip fractures in the US per year. Nerve block is a standard of care for pain management. And for those who are not familiar, a nerve block requires a person to typically visualize a nerve under ultrasound, guide a needle into that location, inject an anesthetic, verify that the anesthetic envelops the nerve, and that stops the pain conduction signals temporarily. This is the accepted method of pain management. Unfortunately, about 19% of hip fracture patients actually receive a nerve block. This was puzzling to us. And so we conducted a lot of interviews through this Seedling process and found that the crux of the matter was that emergency physicians, which is where hip fracture patients present, are not actually frequently trained in nerve block administration. So the workflow for this means that they need to call anesthesia. Anesthesia comes down. It has to be a subspecialist of anesthesiologist that's familiar with this particular nerve block. They administer the nerve block, off they go. Therefore, 80% of people frequently receive opioids for the pain mitigation. This has a lot of downstream impacts that many people are probably familiar with, results in poor patient outcomes, increased hospital costs. And so it's worse for the patients, worse for the hospitals financially, and therefore a good entry market. Now, specifically, the problem we're addressing, like, why is this hard? Why not just train emergency docs how to do this? And the reason is because two things, really. Interpreting ultrasound imagery is tricky. As the previous speaker noted, it's kind of grainy imagery, especially when you're looking for weakly reflecting things such as nerves. And guiding in a needle coordinated with a 1 millimeter thick needle ultrasound is also something that requires not only a fair bit of training, but a lot of reps to stay current on. And so what our technology does is it brings together ultrasound, artificial intelligence to interpret the ultrasound imagery, and handheld robotics. And what we're seeking to do is really package the smarts and the training for a particular procedure into the device. I brought a more recent prototype here. And so we package it all into this handheld robotic system. And this way we can give somebody a couple minutes of training. In our initial testing, it was literally 2 minutes of training. And if you've ever used a stud finder and slid it across the wall to identify where a stud is, you put it in the right spot, you push a button, it fires in a needle. So essentially what we've done is we've married a stud finder to a nail gun. And my old boss at Lincoln Lab hated when I used that analogy because it was like, it's too simple. It's a highly sophisticated blah, blah, blah. Okay. But so that's what we're building. And the way that it works is the ultrasound is automatically interpreted by the AI. There's an expert view that's available that's shown on the top there. And so they can choose to look at this expert view if they want to see how the machine learning is operating, or they can use what's this novice display, which is kind of what I referred to earlier. Basically, put it on the patient in about the right spot, slide it medial lateral until the yellow box is inside of the goalpost. It does a bunch of computations to make sure there's no critical structures in the way. Once you're in the right spot, you push a button. When you push a button, the right-hand side of the device, which is where most of the robotics live, has been adjusted already to the right depth for the needle injection. And needle advances out of the cartridge, applies suction to the backside of the needle. If blood is detected, that's detected by some optics in the handle, and that's the ultimate feedback signal for the vascular access device. The reason that we're in this program developing for nerve block is because the workflow and the shape of the device are a lot different for nerve block, but a lot of the similar machinery will be in place. And so last chart, here's a very quick example. You can see the display. This is on a pig model. Person lines it up. Here's what the AI is identifying, artery, vein. You'll see the right index finger here. Pushing a button. Once that pushes in, the needle goes in. This says success, which means blood flashback is detected within kind of tens of milliseconds. And the person is off with the races with advancing a guide wire. So we're looking at a technology here that the goal is we'll be able to sell this into emergency departments. It'll be financially advantageous for them. We can give them this device, and therefore they'll be able to really use it as a rapid upskiller, a force multiplier for all sorts of procedures. Nerve block is a big one of those. And so we're looking at saving hundreds of thousands of people from ever being introduced to opioids in the first place. Very excited about this, and super, super grateful to Blueprint MedTech for helping us kind of through this process. Thanks.
Thanks, Matt. Those great videos. Next up, we've got Dr. Andrew Chen from Motif.
Just take this. Hi, everyone. Thank you so much for having me speak here. So my name is Andrew Chen. I'm a senior scientist at Motif Neurotech. I'm excited to present about our project, minimally invasive implants for treatment-resistant depression. So here's a bit about our team. So our founder and CEO is Jacob Robinson, who founded Motif out of his lab from Rice a few years back. And some of you may have met Landon Minch, who is our product manager and has been to this meeting before. I joined Motif this year. My background, I'm a psychiatrist as well as a computational biologist, and I'm on the science team at Motif, working on projects including that supported by Blueprint MedTech. We're also joined by a wide variety of consultants and advisors. So the problem that our project is trying to address is the growing problem of mental health. And in particular, 10% of American adults suffer from major depressive disorder. A substantial fraction of those, about 3 million people total, do not respond to 2 or more adequate trials of antidepressants. And they fall in a category called treatment-resistant depression, a very debilitating illness. So there have been some emerging opportunities to be able to treat treatment-resistant depression by neuromodulation. And this is based off of regulating patterns of brain activity, or motifs, if you will, that regulate mood and attention. A particularly important network is this one here, the central executive network. This controls executive functioning as well as our ability to engage with the external world, and tends to be underactive in patients with depression. Now, you might see these two large red blobs here, paired red blobs here at the top. This is the DLPFC, or the dorsolateral prefrontal cortex. And, sorry, is this, because you're cutting in and out? We're good? All right, fantastic. And is a common target for neuromodulation treatment. So this actually is the target for a commonly used intervention in treatment-resistant depression, which is TMS, transcranial magnetic stimulation. Here we're using this big coil over here to generate a strong magnetic field that penetrates into the brain and is able to induce an electric current in the DLPFC. This works fairly well for depression. However, it does require the patient to go into clinic for the use of this specialized machine, and a TMS course can be quite lengthy, about 4 to 6 weeks of coming in 5 days a week. The other issue with TMS is it doesn't tend to last that long. So of people who do remit from TMS treatment, about 50% of them will relapse within 6 months. So this is a place where we identified a potential problem, and our device is meant to mitigate. So at Motif, we have created the DOT, which is this video? Great. Which is a miniature epidural implant that is able to electrically stimulate the DLPFC, similar to TMS, but is able to be done from home and can treat the patient from the comfort of their own home over a long-term maintenance therapy. So the device is tiny. It's less than 1 centimeter in width and can be implanted in a standard surgical burr hole in a procedure that could take as little as 20 minutes. Once implanted, it rests above the dura, where it's able to stimulate the cortex underneath as well as record signals. So the implant itself does not have a battery. It is externally powered by a headset that sits inside of the hat. And the hat is able to transfer power wirelessly to the device, as well as communicate the stimulation protocol. So unlike most wireless power devices, which rely on electromagnetic induction, this technology actually uses the magnetoelectric effect. Much of the development actually came out of Jacob Robinson's Rice lab. This is particularly well suited for miniature devices because it has increased efficiency with transferring power to such small devices. So not only can this device communicate both uplink and downlink with the device, it also transfers power to be able to deliver electrical stimulation to the cortex underneath. There, we will be able to stimulate the DLPFC similar to TMS, which we expect will propagate down to downstream depression-relevant brain networks. Not only can it stimulate, but also has the potential to record, which will be similar to ECOG or electrocardiography, which we expect will be very important for being able to personalize closed-loop protocols for patients. Now, the Blueprint MedTech program was very helpful in terms of being able to support our development of this device. We were part of the Seedling program back in Cycle 1 in 2023, 2024, and there we worked with advisors Anthony Kuro as well as Sunira Bhatia to identify and address 6 gaps in our program. Now, many of these gaps had already been on our radar, and we had been actively working on them, but we still greatly appreciated the advice and perspective from the program. But I'll point out one particular gap that was identified by the Seedling program and I think has been extremely valuable for the development of our company. So just to give you a sense for what these gaps look like, the first gap was about proof of concept of electrical stimulation in vivo. We had not had that yet, and to address that gap, we conducted studies in pigs as well as intraoperatively in humans, where we stimulated motor cortex and with the DOT, and we were able to show that it was able to provoke muscular contractions. Second and third gap had to do with clinical trial design as well as our clinical expertise. In response to that, we completed an FDA pre-submission as well as expanded our clinical advisory board to include neurosurgeons, neurologists, and a variety of psychiatrists. Now, the gap that I was mentioning earlier that was pointed out by the Seedling program, this was about diversity in our patient candidate population, as well as patient views on technology adoption. So not only did we formally interview 5 patients to get their perspectives on our technology, but we established a community advisory board, which is currently directed by our lived experience expert, John Nelson. This includes both patients as well as caretakers of patients, and we continue to meet regularly to get their perspective on our technology. We'll mail them prototypes, wearables, like just getting their sense for what would it actually be like to use our technology out in the real world and integrate their feedback into our designs early on in the process. A couple other graphs I just mentioned. We also got the perspective of psychiatrists, doing a 15-person interview series with interventional psychiatrists, and got actually quite optimistic feedback about the proportion of the population that would be willing to use this device once it's had the track record. And then lastly, some points about regulatory, IP, and quality management. So that was back in 2024. So since then, we've actually had quite a few developments. In this past year, we've now developed a functional wireless dot, and we have tested that out in simulated use in pigs primarily, including stimulation safety studies. And we also did a simulated cadaver study where neurosurgeons were able to implant the device through a burr hole in a procedure that only took 15 minutes on average. We still continue to complete some experiments with V&V as well as these longer-term biocompatibility studies, but we expect them all to be done by the end of this year with our IDE submission early next year. In addition to these engineering milestones, we've also had quite a bit of success with fundraising. So last year, we completed our Series A with just over $18 million, and we got $5 million from an ARIA grant this year. And in terms of upcoming milestones, we have our first clinical study, our first in-human study planned for next year, an early feasibility study, followed by Series B fundraising. And here is where Blueprint MedTech fit in, their seed lead program back in 2023 to 2024, and the optimizer, which will begin this year. Our milestones for the optimizer program mostly revolve around recording, development of our headset to be recording compatible, training our clinical staff to be able to use our device and the recording, as well as developing the software that will be used in line with our device. And one of the reasons why this is one of our focuses is that we think that recording will be very, very useful for patient care in terms of being able to create a mood biomarker to track patient mood status. So in addition to more conventional biomarkers that we could obtain using devices to track sleep or physical activity, heart rate, things like that, the placement of our device over the brain will give us access to longitudinal recordings of DLPFC dynamics. And this will give us the capacity to be able to actually see what is going on inside the brain, discover new biomarkers that we can use to inform care. Not only could this be very useful for patients to have a better sense for what is going on with their own internal brain state, but we imagine that it would be very useful for psychiatrists as well, to be able to create like a patient dashboard to understand which patients are doing well, which patients need more monitoring, having early warning signs of relapse before patients get worse. So with that, I'd like to give some acknowledgements. Thank you so much to the Blueprint MedTech program for your invaluable support over these years. I'd like to thank the rest of the Motif team as well as our collaborators and you for your attention. So if you have any questions, feel free to get this QR code from me or my email. Thank you.
So, Andrew, I'd like to suggest the acronym CDM for Continuous DLPFC Monitoring. Will I get the credit for that? All right. Please join us. We need to switch gear at this time to Q&A. First of all, I'm incredibly proud of you guys. I remember my first meetings with you, and it's an honor to see how far you guys have gone. So I will ask you start with a question, and this is a question for all three of you, and I will let the audience go ahead and ask you a few questions before we wrap up. So looking back at your journey through the seedling program, what has been the single most transformative, I would say, moment or insight that has changed, how your trajectory through this neurotechnology life cycle? I'll start with Matt.
Okay, sure. So for us, it was more of a slow rolling wave that became undeniable rather than a specific moment. Our original application was for adductor canal knee block for nerve block associated with knee replacement. This was based on what we thought was extremely robust literature search, saying there are 700,000 knee replacements in the US per year. A small percentage of people actually receive nerve block for these, and a lot of them take home a lot of opioids. Therefore, we're kind of looking at, 500,000 people introduced to opioids that wouldn't have been. And there was also fairly robust literature that a significant percentage of those form long-term long-term addictions. So we had the combination of, we could do increased billing. We had high usage rates, and we thought we had need based on the people that wrote the papers. What we found as we spoke with surgeons who did knee replacements was that there are some other pain mitigation techniques that they actually felt comfortable with. We weren't there to argue with them whether that was kind of incorrect or not. I'm an electrical engineer. I'm not qualified to do that for certain. But at the end of the day, some of them were like, yes, this is something I want to give my anesthesia team because, they say they always get the nerve block, but do they really? I don't know. From family members who have had joint replacements, I can say they don't always hit the nerve block. Nonetheless, they were like, yes, how much will it be? They weren't pounding the table. But when we spoke with emergency physicians, they were the ones that were saying like, oh my gosh, this is amazing because you don't understand how chaotic my life is. If it takes me an extra 20 minutes to do a nerve block, or if I'm unsure about it, like it's just not going to happen. I've got 5 minutes to treat this patient and sort of triage and move on to the next thing. So they were the ones like standing up, pounding the table, like give me this thing. And so that really completely changed the trajectory of this. And it was an important lesson for me that sort of you can read as much literature as you want, but it really comes out to sort of pounding the pavement. And I'll credit Maxima and Christine with that big time, our mentors, for really pushing us for, you've got to hit this number of interviews. You've got to cover these disciplines. Even when we said, we think we have a pretty clear picture, they were like, that's nice. No, you don't. Go. And so, it was like a good football coach, I guess, sort of just pushed us and got to an answer that we're really excited about.
All right. How about you, Andrew?
Sure. And just saying, does this work?
Can everyone hear you?
No.
You want to share?
How about now? Okay, great. And just saying that brought back so many memories from being in the emergency room and desperately looking around for providers that could do ultrasound guided procedures. So super important. From our end, I think I just want to emphasize again that GRAP that was brought up by the Seedling program in terms of creating our community advisory board. I think that has been really like a fundamentally key, important part of who we are as a company. And I can't take credit for it because a lot of this was done before I came to the company. But when I started, I was so impressed by how much like the patient experience really did factor into our design decisions. Sort of my like clinical philosophy has been it's so important to understand like what are the actual factors that will like increase patient adherence to any sort of treatment. Like the best medication for a patient is the one they'll actually take, and understanding those like little but important logistical things that will impact whether a patient will be able to comfortably use your technology, and being able to integrate that early on in your design process, I think has been so valuable to us.
Okay, thank you. How about you?
So Ivar, let me just ask, like, are you thinking technologically or are you also thinking logistically? Because I can answer it twofold.
No, everything.
Okay.
Like what's
Sure. One perspective I may be able to add, because I'm the only person on the stage now that also has an academic full-time job, and that's rarely 40 hours a week. It's 100 hours a week, right, in itself. We do have very academic entrepreneurs, looking at Youssef, Rolf, Nidish, Shri, they all can attest to the fact that, as an academic, when you go to the higher-ups, and I was blessed to have Dr. Brem, Nick Fielder, all these people who are entrepreneurial. But the academic system first says, oh, you want to spend 10 hours a week doing this? Where's your grant? Like, where's the effort? So whereas this was very entrepreneurial, like I was kind of pushed out of my comfort zone to spend this time in the anticipation of maybe getting a grant, right? So that right there was kind of me coming out of my shell as an academic, being a little more entrepreneurial. And then the second thing was, like, as academics, we usually kind of hold on to our data, being like, well, have you published this? Don't talk to this person, that person yet. Whereas I think as soon as I started sharing and going out and about as part of this Seedling program, I soon learned that, nobody's out there to get you, especially at this stage, especially if you're working on neural implants like class 3. It's kind of really your brainchild. So you got to own it and work on it. And getting out of that building kind of took me back to my training 10 years ago. Like I started doing customer discovery with Bob Story, Chalk Montague. So it kind of felt like this time I actually had a device. And customer discovery, you're not supposed to talk about the device, right? Whereas here I had a whole list of 9, 10 different comments, 12 different comments that I needed to ask about my device. So I think those are the two perspectives I wanted to share as an academic. If you want to be an academic entrepreneur, you're walking a fine line. And I'm grateful for the Seedling program to push me to do that because, we're in a way better place than a few years ago.
Thank you. So not that you don't merit to be here. You're incredible. But this is exactly why when Santosh was trying to compile this list of folks, I wanted somebody from the academic sector to be here. And that's really why I recommended you. Just, it's good to have a flavor of the perspectives of people from the business and the academic or startup and the academic sector, different challenges and so forth. So it's really an honor to have you here. I have more questions, but I'd like to have any member of the audience shoot you guys some questions. Is there a microphone around? We can circulate microphone. By raise of your hands, if you have any questions for any of those three gentlemen.
Everything was very clear.
Very clear. But I have a question for you. So if you can be more specific, I'm sure you've really shared some of those already. How did the mentorship, the resources, and the feedback system, maybe networking with the ecosystem of the Seedling program and the Blueprint MedTech, how have they shaped your readiness for the optimizer phase that you are in now?
So I think it's allowing us to charge into tech development with a lot more confidence, just as a result of having spoken with so many people. And another big plus of the optimizer program is when we go pitch VCs, we're able to not only say that we have non-dilutive support, which of course they enjoy hearing, but we're also able to really answer questions at 2 levels deep, 3 levels deep, 4 levels deep. If we get somebody that's like, oh yeah, well, what's your CPT code? Then we know that, and so we can really adjust the clinical need. I think that was probably the biggest thing we took out of it.
Yeah, I think it's definitely, in addition to the direct feedback and mentorship we got during the program, but also knowing who to bring on board to our team. So expanding our advisory board so we have exactly the right expertise to be able to guide our development, hiring a full-time quality lead to get through that QMS. So all those things were very helpful.
Yeah, I can say that, well, first of all, echoing everything you mentioned as well, I think the mentorship is the single most thing that stands out to me as I reflect on the Seedling program. I've been out there trying to go to different incubations. I've been part of the VMP program as part of the tech transfer office at Hopkins. I've gone to the CDL in Toronto, and I think this felt different because we had the ability to work together for a long time. So, having a sounding board that, Tani was able to tell me, I know where you're going with this, but maybe before I would even get there at this point, you're kind of telling me, I know where you're going, maybe not this time, right? I think that's that relationship building is something that only takes time. And, mentorship is available. We just don't tap into it. But I think having a mentor that's sounding board to you saying that at this point, I know where you're going, but that's nonsense or for another day. I think that's what we all need more so as entrepreneurs because, it's just a risky business we're all in. We just kind of want to maximize our chances of networking and success. So.
Great. So how has your team evolved between the Seedling and the optimizer phases? For instance, have you needed to add more expertise, maybe change your internal structure or how you collaborate or, with others, strategics and so forth? Has there been changes?
So we've been doing a lot of hiring on the engineering front. That's partially, there's the financial aspect, of course, that's really important. There's also the sort of having clarity of vision because we're always competing for people that could go work at a lot of different places that would give a lot better work-life balance and pay a lot better than we can. So they really need to buy into the mission, which means we really need to be bought into the mission. And so this sort of confidence that we've gained through all of the mentorship has been big for that. Also because there are probably 6 different disciplines that need to come together to make this little handheld robotic system. Accruing enough funding through any single support mechanism to hire 6 full-time people is kind of a showstopper. And so by being able to sort of have part of the engineering resources go toward the vascular access device, part of the resources go toward the nerve block device, that allows us to actually assemble a full team where they're fractional people split across. And just from a real kind of practical nuts and bolts perspective, that's been a huge game changer. And yes, that's the biggest change now is we look at our team now versus 6 months ago, and it's triple the size, I think. We started with 3, so that was easy, but yeah.
I think I may have already partially answered this with my last response, but yes, I want to echo some of that as well. I think interdisciplinary collaboration is very important, especially with a small company. We have people that wear many, many hats, but we have since been expanding our team. I guess actually I'm actually one of the people that has since joined Motif, since the Seedling program.
Yeah, so what I was expecting going into this whole journey was, kind of as time grows, having less engineering techs, right? And having more commercialization mentors and reimbursement consultants, regulatory consultants, and animal surgeries or human trials, right? So that I was expecting. What I was not expecting was some of the funding cuts we experienced. So because of that, our team downsized. But the ability to bounce back up was something that I experienced, that I tried to push myself. And, I feel like I grew up as part of that process. I've developed a more tough skin as part of that. But then if we were to break it up, as I mentioned as early on, the number of engineers are reduced, but then the needs are more so in, who's going to pay for this and how, and, have you identified this regulatory pathway and that? So kind of there are more human power behind our meetings with FDA every month, the reimbursement calls and so on and so forth. So change the nature of the breakdown. So.
Well, listening to all of you, so I definitely know that you guys really favor the mentorship. You favor the regulatory support, reimbursement specialist, and everything. Now, there was something that we didn't do in the first cohort, which we started from the second cohort, and I think one of you made a mention of that. We are trying to get them to learn how to face investors. By show of hand, would you have wished we had that during the first cohort? This is really pitching and actually practicing and learning how to navigate that investment, world.
It's a good skill set. I kind of went out of my way to kind of learn them, and so I've asked that question many times and exposed myself. So I think it's a good practice, but also leaving it to the entrepreneur to kind of like coming to the table for some things as well may not be a bad idea as well.
All right. Questions? Oh, we have one at the back.
This is probably mostly for Andrew and maybe Amir. Could you give, I guess, two-part question. Do you have an estimate for the total monetary needs, financial needs you're going to need to go from start to all the way to a product adoption? That's one. Like, what does it take to get through the long journey, or any comparables that you use, right? And then two is, how have you approached seeking supporters, investors, angels, whatever, that would stick with you through that long journey?
Oh, yeah, that's a great question. I admit I actually have not done that pen and paper math to figure out how much total money, but our Series A, we raised about 18 million. I think we're hoping to do a lot more than that for Series B. It takes a lot to get to a PMA stage for a device.
2.4 to first in human. 2.4 million to first in human. I have some estimates. Happy to talk offline about it for sure.
All right. Any other questions?
I just want to make a comment. Thank you all of you. It's a kind of a plug for Blueprint, since we're talking about funding. This program is very impactful when you all come with your challenges to us, right? It is not a place where you should be selling to us because A, we cannot be bought.
Because there's no money that can buy us. But more important, and I've made a plug for Amir before, this program worked very well because he's transparent and brings to the table challenges. There is no investment capital out there that is not only deep domain expertise, but also works with you along the way to make you more fundable. So continue to take that opportunity so that we can make all of you more successful.
Bob, you have a question? No, oh, waiting. Okay. Well, maybe we should round up. Thank you so much. Please show a round of applause for great sibling mentors, teams. We can go back. So, in rounding up, I can't help but to reflect back to the very beginning, almost 3 years ago, when I was first challenged by the hubs to come up with a definition for seedling. We had a name, but not really how to operationalize a criteria for the seedling team. And then to fast forward now, 2.5 cohorts later, it's just been amazing to see you all and to see the incredible journey most teams have made. I'd like to really express my gratitude to Bob for introducing us to a lot of these mentors. The mentors, without mentioning your names, I'll say big thank you to you. I'll thank Paulo and Steve, who introduced me to John Collins and Penny, two incredible pillars of support for the seedling program. My immense gratitude to our administrative team at Howard, Maria Ippolito, who does all the dirty work for me, and all the challenges with administrative support. Hassan, who is our intern over there, who liaises with the teams. Santosh has been an incredible assistant to me. He does all the navigations with the hubs. My gratitude also to all the members of the hubs, PIs, personnel at Hopkins, Center, CIMIT. You've all been great. This wouldn't have been possible without the NIH team, the incredible program officers led by Michael Wolfson, Leonardo, Nick Langros, Stacy Gutowski. Emily was there before, and a whole host of them. They've been incredibly flexible, listening to us, helping us navigate as we learn the process. So thank you, the organizers, for featuring this seedling section. I thank you all for listening.
Let's see if we've got. We're going to use all of the mics. Okay. You may be. I've got one. Where's fun? Your eyes may be deceiving yourself. I'll explain the slide. We only have one slide that we're not going to talk from slides today. This is going to be an open discussion. We're going to invite you to ask questions anytime during the course of this. I'm Bob Story. I'm managing partner of Launchport, which is a for-profit venture and manufacturing center that's in Baltimore. I'm also the evaluation director for the Neurotech Harbor part of the program. So real honor to know many of you out here. My involvement in this panel is because there's a strategic initiative by the state of Maryland that's focused on life sciences, aerospace defense, and quantum dual use technologies. And so I had the opportunity to be in a meeting that involved many of these folks that was really interesting because it was bringing two different circles of researchers together who spoke really different languages. Talk about the folks in neurosciences and in the quantum side. They talked a lot about qubits. And from my childhood Bible studies, I remember that Noah talked a lot about qubits. And you think, after 5,000 or 10,000 years, we would have made more progress. But that's what this panel is here to talk to you about a little bit. I'd like to introduce the panelists first, and then they'll go into a little bit of their stories and some of their active programs. I think I'll sit so I don't get in the way. I'd like to start with Ralph. Ralph Edian Cummins is the Julian S. Smith Professor of Electrical Engineering and Computer Engineering and Vice Provost for Faculty Affairs at Johns Hopkins. Okay, he's back in the lab again. I'm sorry. My old introduction for you. My apologies. He's a pioneer in neuromorphic engineering, robotics, biomedical systems. His work bridges biology and computation. to develop prosthetics, biomorphic robots, low-power intelligent systems. He's the founding director of the Institute of Neuromorphic Engineering and has consulted for leading tech firms. Did I get this right? For Panasonic and Nova Sensors in the past, right? With over 3 decades of innovation. You look like you're about 30. Dr. Eddie Cummins continues to shape the future of human device interfaces and biological inspired computing. So thanks, Ralph, for joining us today. Next, Laura Jahe is Chief Research Information Officer at Cleveland Clinic and a professor of neurology at the Lerner College of Medicine. She's a globally recognized epilepsy specialist. She leads the Outcomes Research Program and serves as the executive program lead for the Discovery Accelerator, a strategic partnership with IBM that's focused on AI, quantum computing, and data science. Her NIH-funded research has transformed predictive modeling in epilepsy surgery and advanced computational medicine across the international collaborations. Dr. Jahe brings a global perspective to digital health innovation and translational neuroscience. My research says you're also fluent in Arabic and French. Is that correct? This panel will be conducted in English. Wolfgang on the end here is not on the slide, and this is a little bit of an interesting story because Wolfgang was invited to be on this panel, but he had an NIH conference that he was going to have to attend today. And so we were very sad about that, but Geetha Senthal with NIH, who is involved in a lot of the cross-functional activities in neuro and quantum, was going to be on the panel. So when one door shuts, another door opens. Unfortunately, Geetha couldn't be here, but Wolfgang's conference was canceled, so he was able to come back and join us. Dr. Wolfgang Losert is a professor of physics and interim associate dean for research in University of Maryland's College of Computer, Mathematical, and Natural Sciences. His interdisciplinary research explores the dynamics of complex systems at the interface of physics and biology, with a focus on cancer cell behavior, collective motion, and machine learning applications in living systems. He co-directs the UMD-NCI Partnership for Integrative Cancer Research and holds appointments in bioengineering and the Greenbaum Comprehensive Cancer Center. Named an M-Power professor in 2023, Dr. Losert is a leader in quantitative biophysics and integrative biomedical innovation. And a bit of trivia for you, Wolfgang, is that the managing partner of our in-house venture capital firm, Deb Hemingway, I think was one of your students, wasn't she? She mentions you with great fondness. So I would like to say a little shout out to Geetha before we jump into this great group. Geetha is a deputy director of the Office of Special Initiatives at the National Center for Advanced Translational Sciences, NCATS, part of NIH. She leads strategic programs at the intersection of biomedical innovation and emerging technologies, including NIH-wide efforts in quantum information science and translational genomics. Prior to joining NCATS in 23, Dr. Senthil spent over a decade at NIH, the Center of Mental Health, where she served as acting director of the Office of Genomics Research Coordination and co-led the NIMH genomics team. Her work bridges scientific disciplines to accelerate discovery, data integration, and precision health initiatives. And I wanted to give her a shout out because she's intimately involved in the various areas, interagency and inter-institutional center discussions about how quantum is going to integrate into the future of NIH. So make sure you have her name marked down and can coordinate with her in the future. I'd like to start with you, Ralph, a little bit. I think you're sort of a hardware guy. You've been involved in AI and machine learning technologies with conventional computing. And maybe you give us a little bit of a primer about how you've, your background and how you've kind of got to this point. And I'll come back to you and get your observations on quantum in a second.
Thank you so much for inviting me. Right. Yeah, and thank you so much to be here. So, I love the fact that I'm at Gallaudet, and it reminds me when I was, in college, I played soccer, and I played against them. They kicked our bums, but the interesting thing, it was so quiet. They were celebrating, but because it's a school for the deaf. For those who don't know, the field was so quiet. So coming here just reminds me of it. So anyway, I just thought that was a little bit tidbit. So I've been working at the interface of integrated circuits and brain science pretty much all my career. You may remember that last year's Nobel Prize for Physics was in basically machine learning and AI. John Hopfield was the kind of the hardware guy, and Jeffrey Hinton was the software guy that won the award last year. So the building of the hardware, essentially the systems that can be put together, started by essentially looking at brain, looking at at physics, looking at the engineering, and coming up with networks that would be able to be used to implement associative memories in his case. Well, that particular effort came out of a group that was put together at Caltech. Basically, that included, Delbrook, included Feynman, included Hopfield, and Carver Mead, who was basically a circuit design guy, right? And them together, and some folks like Anderson and so on in neuroscience, but they came together to form a group to study how brain can inform computing, right? And because of that, we had the Nobel Prize. We've had the kind of the flux of neuro AI. That's a new terminology that folks are using now, which is basically truly biologically inspired AI, and then ultimately how that can be used for computing. Well, we are entering, I believe, a new phase now where quantum might become what neuro AI, was, back in the 80s, right? Where there's a language of the physicists, which we will hear from Wolfgang, speaking the language of computation, which is like what I do, computer science, computer engineering, and try to put together new forms of computation that may be relevant to neurotech. I think the gap to neurotech is bigger, and that's probably where you come in to show how we can close that gap. But I think us two are a little bit closer, and then we'll see how you can then feed into this design.
Fantastic, thanks, John. So, Laura, just a little bit of your background, what your role is at Cleveland Clinic, and take it.
Sure. Yes, happy to. So Laura J. Hi, I'm a specialist in epilepsy. So the first half of the day with many use cases and examples that were provided about brain surgery and people with epilepsy, that is my bread and butter. So my personal research, though, has always been very data heavy. I build predictive models that can look at outcomes of brain surgery to treat epilepsy. I build models from EEG data, similar to what Shri showed earlier today, from scalp EEG to predict people who will do well versus not, study brain networks and such. And then it went into understanding the mechanisms, so needing to collect genetic data and tissue and blood. So in my personal research journey, I've had to solve problems. I needed to get brain tissue for my research. I needed to get electrophysiology recordings, EEG, to build these models. So I had to build infrastructure to support me. But then in my organization, others needed that. And then at some point, Cleveland Clinic came and said, okay, so whatever you're doing, do you want to do it bigger? And in 2020, I became the clinic's chief research information officer. So that's a job where I bring technology to research across the health system. In 2021, Cleveland Clinic was turning 100 years old. I had just been in the job for 1 year, but it was a good time because the organization was thinking strategically. When you're looking back 100 years, you're not looking forward for the next budget cycle. You're looking forward for how are we going to change the world 100 years from now. And it is that mindset that allowed me to bring a technology like quantum computing to the organization. And we will get into that some more later, but that was interesting. But that's in a nutshell, how I came to be where I am.
So I do want to hear about your 100-year plan when you have a chance.
Sure, no problem.
So Wolfgang, give us a little background.
Yeah, delighted to be here. And I have to say that I came to this
study at the nexus of quantum science and work on biology and biomedical research from our work on excitability. And so we had been studying mechanochemical excitability in addition to electrical excitability in cancers and healthy tissues. And over the last few years, we had noticed that mechanical excitability is also something that neural networks possess. And so that raises the question, if you're looking at cells from a mechanical perspective, you might need new sensors. So from a purely biophysical perspective, we came to the conclusion that these mechanochemical oscillations and waves would require new sensors and new kinds of actuators in order to access that additional signal in cells. Now, it may sound strange if you haven't heard it before, but what you heard this morning, I had to teach at noon 120 of my best friends. Well, they're my best friends because they're pre-meds and they really want an A. And I'm trying to teach them the physics of living systems and connect to their interests. And I was able to use one of the talks this morning. It was so inspiring to hear
Dr. Bellsberg.
Yes. The propeller,
Yes.
Yeah, it was scary for our physicists to see the pictures, but it was very inspiring to see the impact that the research has. And I was able to, almost in real time, 2 hours later, convey that to 120 pre-meds in a physics class. The reason I brought it in the physics class is that we heard this morning that there are physical signals. There's electrical signals, there's mechanical stretching that might impact the regeneration of the nerve. And so this is what I started thinking about and what I want you to think about when we think about the need for new kinds of quantum devices. And quantum computing is a great need, but quantum sensor is another huge area where the mechanical components of the biology could be sensed. So that's how I got into quantum.
Yeah, thanks very much. So this part of the goal of this discussion is recognizing this is sort of the starting point of thinking about alliances and collaborations between the different fields. So I'd like, each of the panelists has a little bit of story about some of the things they're involved in. Ralph, you were talking a little bit a couple days ago about where you are with AI now, what you can do with with conventional computing, and what would you like to see in terms of being able to discover what could be done in quantum? Maybe you might speak to what you see as some of the shortcomings right now in terms of quality control, et cetera. But.
Sure. Look, fundamentally, if you look at the term QML, right, quantum machine learning is a term that's used to describe some of the approaches that's being taken to try to apply it to apply quantum computing to medicine, right? So it's usually in the form of image analysis, usually trying to find some disease in some big database, and so on, using quantum computing. Now, firstly, the problem that you encounter is that translating these databases into the language of quantum computing is difficult, right? And you need, a whole bunch of I guess, layers of translation in order for you to be able to encode the databases in the bases that are used for computation within quantum computing. So maybe that's some of the sensing comes into play. And one of the things that I always notice is that ultimately quantum computing is an analog process. Databases are stored digitally. So you have to do this weird translation, but the world itself is analog. So there must be a way there that we can find a relationship that we can exploit, and that's where the sensing folks come into play big time, right? So that's one thing. The second thing is just the scale, the size of the problems that that we are familiar with in the classical AI, classical AI. You talk about billions of parameters, right, being processed and in these large language models. We are still talking about, I think the biggest computing system is about 6,000 qubits, and then on top of that, it takes about 100 qubits to represent one actual qubit, one logical qubit correctly without error. So at the end of the day, you're talking about small systems, right? There, maybe you can take advantage of storage and loop back and do sequential processing. But then you lose out some of the big benefits of quantum computing, which is parallelism, and just automatic entanglement, and then solving the problem in one shot, right? Although it's probabilistic, so you may have to do it multiple times. so that's another thing altogether. But so here you go, having thinking that we could make such progress, and you still kind of have to solve these fundamental physics problems, fundamental implementation problems that still makes it difficult for us to implement computation on these scales. And then, of course, let alone the fact that most of these quantum computers have to run at low temperature, which requires large cooling systems, and and so on and so forth, right? So that makes it again more difficult. So all that is to say that while we are making progress, we cannot yet use quantum computing to do large language modeling, right? We just can't. It's just not even close. So the type of problems that folks are using or are doing are very simple. What I would say is old school machine learning. That's being tested and that's being tried out in modern quantum computing. And most of it is being done in simulation. Folks like you who have access to real quantum computers is rare. For most of us, you're basically taking something like Penny Lane or Quispit, which are different software packages, SDKs that you can use to model what may happen in a real computer, quantum computer, but it's not easy.
Not ready yet.
It's hard.
So
for me, it is hard, but
so Laura has a 100-year plan for all of this.
Yeah, and I think we're getting there sooner than 100. I'm glad Ralph wasn't the guy talking to our CFO or the guy that was hyping up the clinic researchers. So it is true that the languages are very different. When the analogy I used when we were starting our program, where we were linking the computer scientists and the engineers with our biomedical researchers, is as if I had a room where half the room only spoke German, the other half only spoke Chinese, and they needed to generate a report in English. There is no way these folks understand each other. But let me back up, because actually I don't know how familiar the room is with quantum computing. We sort of dove right into it. But and I know there's plenty of engineers here, but just so we're not making assumptions, how many people in the room know what quantum computing is? Okay, so most people don't. How many people have actually read about quantum computing, anything, before this call? Okay, maybe a few more. So can I take 2 minutes just to explain? Yeah. So you all understand classical computing, though, bits, computers, how they work, the zeros and the ones, right? So a classical computer is based on the fundamental operating unit. It's called a bit. Imagine that was a room, and you walk in a room, and the room stores information by how much light is in it, right? Instinctively, as you walk into a room, you reach for the light switch, and it's either on or off. There's either light or dark. It's either 0 or a 1. And that is how much information that room, that bit, can store. We want to store more information in there. You could build many more rooms. You could put your friends to be flipping switches up and down in the building. That's building supercomputers or, parallel computing. But still, you are limited by the fact it's a light switch and it's either a 0 or a 1. Get out of there, go in another room, and that's the qubit. That's the fundamental unit of a quantum computer. There, it's not a switch, it's a dimmer. You flip it all the way up, it's light, all the way down, it's dark, but you can stop anywhere you want along the way. All of that information, all of these different combinations of zeros and ones are stored in this one tiny room. That is superposition. I'm simplifying it, but it's close enough. That's a quantum principle. All of that information is in this room. To add to it, whatever you're doing to this switch here is managing and changing the light in the whole building. And all of that information is also in just this one room. That's entanglement. And that is the difference between how much you can store potentially in a quantum computer versus what you can do in a classical computer. You can store the genetic code of, a human genome, one individual, in just a few qubits. You could store the genetic code of the whole human race, theoretically, in 32 qubits. There is no supercomputer that is big enough now to store that much information. The technology is not ready to do large language models. That is true. What is also true is the technology does not need to do large language models for it to be successful. The problems that we are talking about, Nobel Prizes. The Nobel Prize in Physics this year went to quantum computing. The Nobel Prize in 2022 went to quantum computing, algorithm development. The Nobel Prize the year before also went to quantum. This year is the International Year of Quantum by UNESCO. They labeled it. And it is because it is the technology of the future, right? So going back to my 100-year plan, I was sitting there thinking, okay, I'm in Cleveland Clinic. We have 240 individual PI labs. We have 400,000 square feet of wet bench research space. We just signed an agreement with the state of Ohio to invest with us $565 million to build an innovation district anchored around biotechnology. If I just invest in AI, I'm doing what the whole world is doing. There is nothing that is going to differentiate us. But if we invest in quantum technology, that is going to set us apart in few years when the world catches on. We made that decision in December of 2021. And mind you, I had just been there for I've been at the clinic for a while, but in this job, leading research informatics, it was like 1 year into it. So it was a big risk. And the calls that we got, half of them were some version of congratulations, this is great. The other half was like, what are you thinking? Kind of an answer. But then the year after is when the Nobel went to quantum and all the other Nobels that I mentioned. And then the NIH investing and the whole world investing and, UNESCO and the rest of it. So it's been great to have this advantage where we have a quantum computer on site in the middle of our research facility. The team this morning was talking about the barrier being the biomedical and the computational people not being together. We have it in the middle, and then the hospital is right across. So that has allowed us to really define the real use cases for quantum computing. They're not LLMs, we're not there.
So Laura, I'm going to ask to come back to you to talk a little bit more about the IBM relationship and maybe some of the targets you're working on and the collaboration opportunities. Wolfgang, I wanted to jump over to you though now, and I'm sure you've got some physics rebuttals to the entire discussion. But I'd also like to, since you led the Quantum Neuro Challenge that was part of the, I guess you and Laura collaborated on that. Tell us a little bit about that and also how you found that sort of intersection and what that kind of program meant to people who were thinking about how do I use these tools in the biospace?
Yeah, I think that's a great question that goes to the heart of how would you actually use a quantum computer? Why would you use it for neuroscience? And Lara gave a very concise explanation of this question of entanglement and what we believed. So we set up a NeuroQuantam challenge to provide neuroscience data, both electrophysiology data and also more biophysical-based data on animal models, looking at dynamics of groups of hundreds or thousands of neurons. And we asked folks building algorithms on quantum computers to identify what is the structure of the data, which of the many, many signals, if you have 100 or 1,000 signals, which of the signals cooperate together. And that is where we believe quantum computing is very well aligned with the needs of neuroscience. Because in some sense, a brain doesn't work like a digital computer. It's not like one neuron is connected to the other, it's connected to the other. Each neuron is connected to its 1,000 best friends and only fires in response to not a single input, but in response to some combination. I think about this as classical entanglement of each neuron. So in some sense, in our brains, we already do the kind of information integration that we think a quantum computer is capable of. Now, going back to the question of how do you make sense of neural data? If I have 100 traces for epilepsy or thousands of traces, if I look at clustering, typically we analyze pairwise clusters. Who is correlated with whom? But in the brain, one object may be correlated with 50 other objects. And computing that on a classical computer gets exponentially more and more difficult. Because if I have, let's say, 100 signals, I have 10,000 pairs, I have 1,000,000 triplets, I have 100 million quadruplets. And computationally, it becomes very difficult to look at higher order synchrony. But a quantum computer does that naturally because the signal is entangled and it can sort of sync up to structures of tens of signals, 100 signals, and identify these groupings in a more natural way. We're not there yet in terms of algorithm, but that was what the challenge was about. We had a large interest of the community, a number of phase 1 proposals what to compute. We had a number of finalists who actually were able to run their algorithm on a quantum computer. We're just at the beginning, but I wanted to highlight that it is not that we're looking for small data sets that where a quantum computer can actually do something. This might actually be a case where the quantum computer is really needed to elucidate structure. So this was very exciting. We look forward to continuing to push forward in that direction.
Yes, absolutely.
So the output is always the thing that, so how do you condition the right decoding in order to be able to pick, what you basically to ask the right question, right? It's like, I want to know, about cats. So how do I find cats in my entanglement? So what does one do?
I think that is exactly the question for which we need quantum information scientists and quantum computer scientists to answer that question. How do you encode information and how do you read it out? But frankly, we're struggling with the same question in neuroscience, in basic neuroscience. Do you read out only the electrical signal? Do you look at the chemical signals? So, yeah.
That's absolutely the right question to ask. It takes us about 6 months now, even however many years into this. It takes us on average 6 months to go from an idea to an actual computational problem that we ask of the quantum machine. It's you have to think differently. See, initially when we started with this journey, it was about, how can we do better than what we can with AI? And then you gradually evolve into what can I ask this that I cannot even ask of AI to answer? So it's It's a different mindset. And along the way, you end up finding the situations where you're doing actually much better than AI. So for those who are interested in drug discovery, for example, if there's anybody with that bent in the room, the most often, the gold standard for modeling how proteins fold, how do you go from a chemical sequence to a 3D structure that you can then test in the lab to see if that drug is going to work. That's where most cost, the billions that go into making drugs, most of it is that. And usually they have to go through 10,000 compounds. Maybe they get one at the end that works. So with quantum, we have been able to break down those proteins into pieces, bits and pieces, and then simulate the smaller pieces and then embed them all back together. And we have published that with the quantum approach, we surpassed AlphaFold 3, which is the AI technology that got a Nobel last year. So we did better than AlphaFold 3 in simulating proteins for drug discovery, particularly for rare diseases where, as everybody knows, AI is only as good as the data that it trains upon. And if you take something like rare diseases where there isn't really a lot of data to train upon, AlphaFold 3 usually doesn't give you accurate predictions. Quantum did much better. So there are these, and this is only one example. There's other applications with quantum machine learning, biomarker discovery, etc., where it's not that it will replace AI. It will never do that. But there are these applications where only quantum will be able to give an answer.
So let me ask the audience. So I'm going to ask the group up here to maybe have a little bit of a wish list that they might have in terms of the pathway for collaborations, the sort of collaborations you seek, maybe what you're trying to accomplish through the IBM partnership. I think I'd welcome anyone in the audience too, if you have questions about that, thinking about your own research, your own development. It's sort of interesting that Dr. Banerjee talked about the cost of change in medtech. So what are the possibilities in terms of risk mitigation in advance of clinical trials? What's well there? So did we have a question? Yeah, Nish.
Ralph, you and I have traced that journey of covered meat to now in neuromorphic, and it's really still not a mainstay silicon. It's always pops up and then it remains very, it has a power advantage over silicon standard digital. And so neuroscientists love neuromorphic encoding and it doesn't compare. So I just wonder whether quantum faces the same future or fate, or is there a long incubation period, or am I missing something? And more so even in neuroscience, because this neuromorphic is neuroscience founded and quantum inherently is not.
So maybe Wolfram can go and then maybe.
Yeah, I think there's multiple aspects to quantum, and I think there's quantum computing, and there's also quantum sensing and quantum communications. And if we look at the University of Maryland founded startup IonQ, for example, they started with quantum computing, but they've recently branched out and bought companies in quantum communications and quantum sensing, realizing that these will be the early revenue streams. And I am particularly interested in quantum biosensing because how many of you have used MRI in your research? All right. Okay, so MRI or in clinically. So MRI is a quantum sensor because you're using the nuclear spin of the atoms in the tissue to sense the status of the tissue. Now, what the modern quantum biosensing is about is we're trying to look at the quantum state of the electrons. The electron is screwed up because it communicates with everything around it. So you have to put the electron either in a nanocluster inside a protein to protect it, or you have to put it in a diamond in order to protect it a little bit. But then it serves as just as powerful a sensor as you have with MRI, but with much better spatial resolution. So there are a number of quantum sensors that are being used for a broad range of purposes. They're not yet used clinically because, frankly, the research that are developing quantum sensors are inundated by use cases. We have an opportunity as a community to foster the translation of these quantum sensors to biological applications that I wanted to pitch. We're running a quantum biosensing workshop where we're trying to get together the quantum scientists who are too busy with like other applications and get inspired to use the tech for biological systems. This will be next Tuesday.
I was going to say, go ahead, make your pitch right now. Next week, what's the time?
Next week, Tuesday, at the University of Maryland, College Park. Google quantum biosensing or search for my name, Wolfgang Losert. I have a recent LinkedIn post on the workshop.
So I see it slightly differently, maybe, or I mean, actually not differently, but just a different consideration, and that is explainable quantum computing, if you will, right? So that's the one thing I would say what neuromorphics maybe provides us, right? Is you can layer it, you understand what the different layers are doing and why the decisions are made. It's my sense, and again, this is my limited knowledge, that it's not quite explainable to know how these entanglements are happening in order so that you can fine-tune it in some way to make it do what you want to do. So I think that's all, that to me is a little bit of a challenge with quantum computing. Second thing is, I always think that, at the end of the day, to make progress, all of us has to be talking together, right? And that workshop might be the ideal scenario that you're talking about, where you bring the physicists, you bring biologists, you bring the engineers. I build chips, right? That's my area, right? I put together electronic circuits, so how can I learn from what the physicists are doing and then put that on chip and also make it affordable? That's the other part that's really tough, right? So as an academic, I can't compete with IBM and Google, right? So how can I get access to either, well, I don't want to get access to a pre-designed system. I want to build my own, right?
Take your time.
We learn by doing, right? We learn by building. So that's my approach. So how do I get access to technology that allow me to build my own?
No, but actually that's so some of the partnerships that we built since then have been with local universities that have engineering programs, and it's to give their students access to the machine so that they can ask engineering questions. There's plenty of engineering questions that still need to be resolved. The only thing I'll say to that is that some of these discoveries just come by necessity along the way. Like, for example, for us when we started, I never thought that I'd have to sit one day and worry about how do I give enough access to everybody who wants it. For the first 6 months after we had built the machine, it took 6 months to find where to build it, because it's so finicky with vibration, temperature of that. And then after that, it took 8 months to build it. 4 of those went into just calibrating the qubits. And then finally, it was available. And we have it in the middle of our cafeteria. So there's all these people walking around it. And I'd have people who would stop me and say, "Oh, Dr. Jehai, what is this box that we have now in this?" So you explain. For the first 6 months, the utilization we were tracking, right? 0. Nobody was using it because nobody knew what to do with it. So all of that time went into these workshops and office hours and, activities to start building the questions. And then within few months after that, we got up to 70%. For almost a year now, we've been at 90%. 95% sometimes, and that people complain about, we want more. The other thing that happened in that time is that we track also who uses it. Initially, it was mostly IBM, less Cleveland Clinic. And then you see that shift along the way, where now 3/4 of the users are just Cleveland Clinic.
And what's the distribution within Cleveland Clinic in terms of the researchers?
The researchers, it's pretty much right across our research areas. So I'd say there is a healthy third, which is all chemistry for the drug discovery, protein folding stuff. And then the quantum machine learning is in the cancer and immunotherapy space, so the biomarkers discovery, omics, genetic data, and imaging equal. And then there is now a small group that is growing, which is answering these types of questions with actually more how do we parallelize, like with GPUs, people worry about how do I make the most out of the GPUs that I have and how can I run workloads on different machines at the same time. We've had to do this with quantum to figure out how do we run different workloads on different qubits in the same machine because we have the line waiting. And then how do we make sure that when the one is done, the one workload is done, we can start with the other. So some of the postdocs are working on that, not answering the biological question, but answering how do you make the most use of the machine. And I'm letting them be because somebody has to figure it out.
So if anybody wants to collaborate with you, there's a lot of planning that goes into that, I'm sure.
Right? But we have these, okay, so there's a few things. So there is the quantum and there's the Healthcare and Life Sciences Working Group, which we launched, and that's open to everybody. We have now 23 entities outside of Cleveland Clinic that are using the machine. We just signed an agreement with UNESCO as part of the International Year of Quantum to allow remote access to it. We have a program for startups. If they want to use quantum for their work, we give them 6 months free access to the machine, and we link them up with venture capital. We have agreements with universities for collaborative research where we collaborate. It's all collaboration because people can get access to a quantum machine through the cloud from quantum providers. They wait in line, though, But for us, we didn't get the machine to sell quantum access. We got it to do the research. So people who want to collaborate on doing biomedical research using quantum computing, we have a whole hub system, for that. You can go, our website is Discovery Accelerator, Cleveland Clinic, and then you'll see some examples of the projects we have, or you can email me. We're always looking for collaborators.
So I know you're going to have to leave to the airport in not too long. I'd like to give all of you kind of a final word, maybe a comment. If there are any questions, there will be a little time to talk with the team afterwards. But maybe last word?
Yeah, so this was very enlightening to hear all the perspectives. So I just wanted to reiterate, the quantum has so many different applications, and from our perspective, quantum biosensing is a key, very near-term application where you can measure electric fields, magnetic fields, forces. The kinds of things that I heard this morning are actually involved in clinical outcomes. And the advantage of quantum sensors are that they're lightweight, they're non-invasive, they can be addressed with electromagnetic fields through the body, so you can have remote accessible systems. So there's a lot of promise in quantum biosensing as an immediate next step sensor to be deployed broadly in MedTech. I'm also very excited about the quantum computing revolution, and I'll be thrilled to hear what the next steps are from the other panelists' perspective.
Okay. So I think there's 3 steps that has to happen, and I'm hoping that the funding agencies that will be watching this video or listening will be putting money into this, right? So number 1, I'm happy that Cleveland Clinic does that one unit that can be used, that can be shared across the world. But it would be wonderful if government made such a thing available at a much bigger scale, so that so if I'm doing some work that needs access to such a thing, I could get access to it. Number two, again, pivoting on, leaning on what happened in the microelectronics industry, where, in the early 90s, late 80s, nobody was actually building their own chips, right? So that was when universities, MOSIS, which was actually funded by DARPA, came to being to actually encourage the teaching of microelectronic design, VLSI, integrated circuits, and then that basically created a whole, blossoming of an industry. TSMC would not exist today if it wasn't for that. The chips that we have, anyway, it was massive. So I would like to see a similar equivalence of that, happening for quantum. And the last thing is teaching, right? I looked around. There are, maybe a handful of universities, Maryland included. Hopkins, I think, has 2 courses, on this topic, right? If we're going to be, marking it for the next 100 years, we need to start teaching the kids now.
Yeah, what they said, both of them. So quantum sensing definitely closer to application than quantum computing. Having quantum centers that are available for people to learn on, absolutely essential. Workforce is absolutely essential. To help with that, at least for our purposes, what we did is we partnered with one of our universities in Ohio. They have an engineering school, and they are starting now an undergraduate, so bachelor, master's, and PhD, all just focused on quantum science. And those students will be spending 9 months each in our labs. So it's good workforce development for the state, and for us, it's a pipeline that will be coming in. Last word, I want to mention Geeta, because I want to make sure that we don't forget her. She has been instrumental in really pushing the NIH towards quantum. NIH, it's the agency that funds us all, and we all review. I sit on study section. I'm supposed to tomorrow. We don't know, probably not. But NIH's mindset usually is you have to be like 95% of the way there, and then we fund you, right? That's all of the preliminary data that we have to put in. That mindset doesn't really work when you're thinking quantum. Nobody has enough to put 95% of the way there. So that's what Geeta was able to change and allow the agency to think innovatively. And they have now a program called QBIT, Quantum Biomedical Innovation Technology. That's what the QBIT stands for. You can look it up. And they are funding awards in a different way, than they use. So it's not a grant, it's an award, so cash. They give the funding, and it's phased. You have to show milestones before you move to the next phase. The first cycle was already awarded. They awarded 10. We got 2 from Cleveland Clinic. But and then there is still a way to go, and they're looking for these practical applications. It's a long way still with quantum. We are expecting quantum advantage in 2026 in certain applications. So that means showing it is superior to AI in like a real world use, not a esoteric mathematical challenge. And then by 2029, we expect to have a fault-tolerant commercial scale quantum computer. So the roadmap for all of the big tech companies is accelerating to make sure that quantum is commercially available sooner rather than later. That's why it really falls upon all of us, no matter what our discipline is in medicine, to learn about this technology now, because it is difficult, it is challenging. The Holy Spirit isn't going to descend upon us the day that it becomes commercially available to teach us how to use it. So it is with that call for action. Be curious, reach out, ask questions that I want to leave people.
Beautifully said, and thanks for closing that. We don't know if any private citizens are listening to us today. But we do want to thank Geeta and all the work she's done. I have to say, I didn't hear one word about 40 days and 40 nights of rain, though. Yeah. So I'm a little confused about this whole discipline. But hey, this is a fantastic panel. I want to thank all of these folks for taking their time. Laura, coming from Cleveland and spending the weekend with us is great. Wolfgang, thinking about us and coming back and engaging, and my buddy Ralph, always there. There'll be a little time afterwards. I think there's a break now before the next session, and feel free to grab these folks. Thanks again to all. Thank you.
We've got one last panel, and it's the success stories and an interesting topic as well about the assistive and restorative medtech advances. And this is something that I think will be really interesting for you guys, so thank you for hanging with us. And to start it off, I'm going to bring up Dr. Henry Brem, and he will announce his panel, and we'll get started. Thank you.
I've seen the Tish. Is he here? Someone else will give his talk. Well, it's my pleasure to, firstly, my congratulations to you guys for your stamina and staying to the very end here. We're going to talk about success stories and panel discussion, and it's myself moderating, Paolo Bonato, who's the Director of Motion Analysis Laboratory at Spaulding at Harvard Medical School, Associate Professor there, and he's the co-principal investigator of CINTA. Nitish, who may have gone home, is my colleague at Hopkins. He's Professor of Biomedical Engineering at the Whitting School in Neurology and Neurosurgery at Hopkins. And Rahul Kaliki, who is the CEO of IBT, which you'll be hearing more about, and Adjunct Associate Professor of Biomedical Engineering at Hopkins Medical School. I'm going to very briefly go through, just as sort of an introduction. I hope we consider it a success story, but some of the work that we've done, it's sort of a different aspect of what I talked about this morning. One thing I would say is that, the title of this group's, this session is Assistive and Restorative MedTech Advances. I think neurosurgery has been mostly in previous generations about removing things. We take out tumors, we take out trauma, we decompress, we do craniectomies to take out bone to make room for the space or the spinal cord. We take out disc, we take out things. Even vascular surgery, we take out an aneurysm, we obliterate it so it's not part of the circulation. And that's been 99% of neurosurgery, maybe all the surgeries, about taking out things. I think that what we started to realize is that, and I think where the field has evolved and is evolving, and our work was, some of the early work on that was about putting things in and restoring the brain or treating directly in the brain. So the neurosurgeon's not only taking out a tumor, but also putting back a therapy into the brain. And on that, I just wanted to very briefly, show you just our own sort of journey with chemotherapy implants and where that's gone as an example of the challenges and also, I think, the success of being able to solve problems and change the way patients are treated. So when I finished my residency in graduate medical school and I trained at Columbia, there had been no advances in the field of, significant advances in the field of neuro-oncology in the way brain tumors were treated. There were surgical advances, but the outcome had remained stable for probably almost 100 years since Cushing's time, and that was that it was uniformly fatal. People died within 9 months, no matter what was done, and people had tried. When I was a resident, I was advised not to. The chair of neurology spoke to me as I was leaving when he heard I was going to Hopkins, and he said, "Congratulations, that's a great academic place, but I heard you're going to specialize in brain tumors. Like, that would be such a waste of talent." And I said, "Why is that?" And he said, "Well, because it's a fatal disease. There's nobody has been able to make any dent in it, and you're just wasting your career, wasting your time." So that was a good starting point. And in fact, the FDA had not approved any therapy in over 20 years. and the outcome hadn't changed during that time. And this is basically, I showed this earlier this morning, is basically the outcomes. Almost every hits people at their prime of life, and people die within a few months on the average. 9 months is the average, and by 2 years it was 100% fatal, or 99% fatal with an occasional exception. So if faced with that, the question we asked, so the question really, as I was starting after my training, was, could anything be done to change this outcome? One of the questions that we posed in working with Bob Langer, who was early in his career at MIT, but we had both worked together in Judith Folkman's laboratory at Harvard, me as a student and Bob as a postdoc, and where we developed the polymer slow release systems for anti-angiogenesis. But the question was whether the drugs that had been tested and all failed, whether the problem was that they just weren't getting into the brain. Were they? And the reality is, in the 1980s, after tons of clinical trials and everything else, nobody had really measured or had any method of measuring whether chemotherapy even got into the brain, even though they poisoned thousands of patients with all these different drugs and cocktails, and they got sick from it because it certainly went to the rest of the body. But they didn't even have a way of knowing if it was getting into the brain. They didn't do the experimental work to look at that either. So we postulated that maybe if there was a way of getting it directly into the brain using these biodegradable polymers that Bob Langer and his group at MIT had developed, we could get a safer way of delivering chemotherapy without the systemic chemotherapy, and whether we could get higher doses in the brain. Now, today we have Gliadel, which we're going to talk about, which are these chemotherapy polymers, Timidar, Avastin, Gliasite, Novacure, targeted therapies, imaging is better, surgical techniques are better, ICU is better. And what we've seen in terms of impact is that the median survival is now 20 months and not 9 months. So on the one hand, you can say, well, that's great. You've more than doubled the median survival, and we have more long-term survivors. Now it's about 20%, let's say, of are living 4 or 5 years afterwards, whereas it was really nobody was surviving more than a year or two. So on the one hand, you can say, well, that's great. You doubled the survival. That's a big deal. But on the other, to me, it is a big deal because it shows that if you use science and you use technology, you can change the outcome of what was thought to be an unchangeable disease. But of course, whether it's 2 years or 1 year, it's still a horrible disease. It's the prime of life. And so there's so much more that needs to be done and can be done. And I think that, I'm going to talk for a minute about Gliadel, but I think what we did, even more than whatever contribution was in introducing one new therapy, is I think that we showed that working with companies, working with technology, working with the FDA and the NIH, we had a approach that other companies could use, that they could base, if they did, more or less follow that model of what we did, they could go to the FDA and they could get approval, and that it wasn't an impossible task. It was just a matter of finding the right combination of treatments. So, the question is, how do you overcome the barriers of developing new treatments? Now there are lots of things in the pipeline. Well, firstly, if you have an idea, you go to the laboratory and you test it, because surgeons think of things, say, gee, it could be better if you did this, and oncologists and so on. So you test it, and there are models for testing. Then it's clinical trials to look at safety and efficacy. The FDA approval, which is very challenging for drugs. It's much easier for devices, but it's still a challenge no matter what. And then as I went on this journey, I realized I would have thought that the FDA approval probably isn't something that as a neurosurgeon I'd be involved with, but I learned that I had to be very much. But then it even goes beyond that. Even if you have FDA approval and you have a company working, there are marketing issues. What's good enough to merit investment? What gets hospitals to agree to take on new therapies? Who pays for the treatments? And I'm not going to go on, but we had to petition CMS, and it was very hard to get funding, not for us, but for patients to be reimbursed. They had to form a new DRG, and we had to petition really to Mark McClellan, who was the director of the CMS at the time, to both as it turns out, both for gliadel and for deep brain stimulation. But that opened the door to technologies being introduced in neurosurgery. And then there's this whole thing about clinical judgments and biases, which also, I would have thought good science, you don't have to worry about that stuff, but people, you do have to convince people to change their behavior and develop treatments. So the problem that we addressed, and I'm going to be very brief with this, is that could the ineffectiveness of cancer therapies be due to delivery and getting across the blood-brain barrier? And this, beautiful diagram from Ian Sook summarizes about 20,000 papers in the literature about how drugs get across the blood-brain barrier, either by understanding, lipid-soluble, small molecular weight substances get across, or you can open the blood-brain barrier, or you could use Ommaya reservoirs or convection-enhanced delivery and directly deliver it into the brain, or the work that we did with biodegradable polymers or microchips and just place them on the surface and bypass the blood-brain barrier in that manner. And this is gliadel chemotherapy, polyanhydride polymer loaded with chemotherapy. It takes about 10 minutes of operative time. This is the tumor's been resected here. And this is normal brain surrounding it, and you just line the surface with these dime-shaped polymer wafers. And, the concept is pretty simple. This is from a Scientific American paper that Bob Langer and I wrote back in the 90s. And it just says that, it just shows you resect the tumor, and the drug diffuses in a significant area around where the tumor is likely to recur. So when we were developing this work, and I think this is probably the most important slide, at every stage that we were presenting and doing the research, we got the same sort of response, both Bob Langer and myself, and that was, that's really great work, that's very cool, very well done, but you realize it'll never work, it'll never happen. And he said, gee, why? And it was interesting is that as we went on, whether it was scientific meetings, business meetings, Gordon conferences or neurosurgery meetings or oncology meetings, there was always the same sort of thing, like, that's cool what you're doing, because you've done that well. In whatever stage we were at, we did that well, but it's never going to happen. And I think it's instructive to look at some of the reasons that were the leading reasons. And I must say this slide I've shown many times in many different groups, and it resonates with everybody, because no matter what technology you're developing, it's universal. Whether it's in business, whether it's in medicine or science, it's the same thing. There's always a polite, generally, but a rejection of new ideas and new approaches. And the idea is not to get angry or crushed, but it's to respond. And what we did is we kept going back to the laboratory and answering these questions, or to the clinic. So the first series of the early years was, well, polymers can't be synthesized that would do this. They would react with the drugs. They're fragile. And these were things that the MIT group answered in papers in Journal of Biological Chemistry, and they showed that they could construct polymers that could release and biodegrade. Then when we started working on it in the 80s, they said, well, gee, if you ever put those toxic drugs into the brain, you're going to melt the brain. It's going to destroy the brain. So we looked at rats, rabbits, monkeys, and we showed that it was actually safe to put it in the brain. We could do it. Then they said, well, sure, it's safe. That's very nice. You've shown that, but that's because it doesn't go anyplace. It only goes a millimeter or two. And so we showed, okay, we developed a hold with Mark Saltzman, who was an assistant professor at Hopkins. Now he's chairman of biomedical engineering at Yale, but showed that actually it diffuses centimeters away, and you can model out how it diffuses by studying it in different animal models. And then there was a question about what drug we used, but that was the only FDA-approved drug at the time. And then we showed all of that, and then they said, well, and we did the clinical trials, and they said, well, the FDA would never, this is too complex a system. And so we did randomized prospective placebo-controlled studies. We used the same rigor in the clinic that we did in the laboratory, and the FDA was fine with that. And they said, shown in that way and doing that kind of rigor with a placebo-controlled randomized study, they did accept it. And then it became the issues I alluded to before about how it would be paid for, which patients would benefit, would the indications be broadened, Then we started with the prejudice stuff of, well, people started putting in as an exclusion criteria, we won't test a new substance if they've already gotten the local chemotherapy. And that was, I never understood the logic for that, because why would you deprive a patient of a clearly demonstrated benefit? But that's reality. And then, of course, that we need better drugs and more sophisticated approaches, which we've been working on. And I'm not going to go into a big thing on that, except that's the major work we've been doing for all these years. So the key paper was this randomized prospective placebo-controlled study. We published it in Lancet. On the basis of that, the FDA gave approval, both for initial and for recurrence and for all gliomas. And it was with this work that we showed, at Hopkins, but in many, many other places, which I'll show you in a moment, that we saw more than doubling of the median survival when we combined it with temozolomide, which is a newly, at that time, a newly discovered chemotherapy, now standard used. But you could see that we saw, almost a doubling of median survival. And you say, well, that's really old stuff. But this one is from 2023, but if you do a Medline search, even today, there are papers being published about its efficacy and its usefulness and how to best use it. But this is an example from France of 1,600 patients, at 42 institutions. And, they showed these are the things that are favorable: young age, female gender, concomitant chemotherapy, had a better overall survival. But there was clear cut that it improved survival. The conclusion from that study, was that BCNU wafer and with the chemotherapy regimen should be considered a standard of care for malignant glioma. The progress is incremental, but it only happens with enormous, relentless effort. And so I present that just to show that that's been the start of what I think has been, many other things that we're trying to accomplish. But it's a prototype of working with companies, working with academics, working with the regulatory agencies, working with the NIH, working with colleagues, working across specialties. And the outcome is both very satisfying and very stimulating that we need to do better going forward. So I think we'll have questions at the end, but I would call on our second panelist, or Paolo Bonato, to speak and present. What? Who is going to go last? So I just wanted you to know he's here, but he's going last. And Nitish, my colleague from Hopkins, is going to precede him. Yeah, we're saving the most powerful discussion for the last.
Last for the best, while they can help me plug in the Mac and all that. So if you could do that. While this is, be careful in this place, because I'm a professor. So first thing during lunch, what happened was my bag is disabled. Oh my God. Because I saw somebody else's bag and I kept looking everywhere. And then I went there. Again, suddenly my bag disappeared. So somebody switched the backpack. Okay, so make sure, don't bring black backpack that somebody takes away. Then second chaos occurred is I lost my glasses. Luckily, my wife had packed a second backup. So I think I'm okay. And I'm just giving a few minutes while he sets it up. So I think my goal right now is to kind of lay out a bit of a journey. And I'm going to weave in how different agencies play the role. And just let's go to the presentation. Yeah, please. So how different agencies play the role in the ecosystem. Through my own journey, bring in where Blueprint is and how we achieve translation. And I want to demystify the different funding programs that people hear, RO1 and SBIR and Blueprint and U44 or whatever. So I feel like I hope I can make that journey vivid within those 10 minutes or so I've given by examples. So that's okay. It's not critical. Somehow the sound it's not going in. It's okay. That's fine. Okay. Some little soundtrack is missing. Okay. So I'm Nitesh Thakur. I'm one of the co-PIs along with Sri and Ralph and Yusuf and a bunch of other people who may not be PI, but they are the mainstay like Santosh and Cabritan and Ivar. So we help manage the program. Okay. So what, like a starting point is like, if you look at it was MedTech Blueprint, but a lot of it is, as you've noticed, Neurotech. And so I just kind of want to demystify neuroengineering, which has so many elements that you can resonate what you see here. Neuro devices, you saw some posters, neuroimaging. There's a fantastic project that's been funded on photoacoustic imaging. Brain-machine interface, something is going to come along the line. Neuromorphic, we just had a session, and so on, right? And in the end, 7. But why I did this 7, or actually I want to go into the next slide, which is really, I published a handbook of neuroengineering. It's about 3,000 pages, edited book with 15 sections. And it really, at the end of it, my last 2 sections are clinical neuroengineering and translational neuroengineering. Because there's a lot of neuroscience out there. But if you start the entire journey from interfaces, which is this Blueprint program, you heard about quantum sensors, so sensing, number 9, neuromorphic, neuroprosthetics, which I'm going to talk about. So this field, like I want to be like, go to undergrad or grad school all over again, because there's just so much going on here. And my presentation, sort of, I'll kind of like focus on this, brain machine and neuromorphic robotics, and hand over to Rahul, who will follow me on prosthetics. Okay, so this is my demystification of where we are. So I will repeat this slide at the end. A lot of my journey started through DARPA program in mid part of my career, where I used to do clinical translational work, but DARPA came in, said, let's revolutionize the prosthetics. And it morphed into later funding from NIH, various mechanistic and some translational work, and it really needs to go towards commercial activity so that we can get devices to the patient with regulatory and all the GLP, GMP, all the things that have to be done, right? So I will repeat this slide later, but that means if you are here, you may well follow this kind of a journey that get R1 if you're an academic. If you're a startup, you'll get undiluted SBIR funding for a proof of concept or something. And then somewhere that Blueprint fits in, right? It's something that is to take it first in man, if you want to take that one phrase when you take back, go home. And if you want to do preclinical to clinical, then there are other programs. So you need to learn to separate out these different mechanisms so you don't get caught up in R01 mechanism or pure clinical study. Okay, so let's see how my journey kind of came about. I mean, you know, way back when DARPA said, you know, control the robot or processes with the mind, right? So, you know, this was, I think, back in 2007, we connected up a student to the EEG cap, decoded that information, and the prosthetic hand opens and closes. So that was kind of like the catalyst to DARPA, because we needed to give a fairly quick result. And that's what we did. Rahul recently tells me that he just went to some venture, I don't know, some startup fair or something, and a couple of young kids are getting millions of dollars to do the same thing. I said, deja vu, you know, but they give money to all these young prodigies. So we'll see what they come up with. But this was BMI way back, right? And DARPA took that risk. That, and this work, I'm going to, you know, blend in a little bit. Some of that work was at Hopkins and APL. Some of it at Rehab Chicago, where they did this regenerative way to put nerves into the muscles for this transhumeral impurity. And, you know, now you can see that this person can control prosthetic hand with regenerated nerves into the muscle. So it's called targeted muscle renovation. But you can start seeing that there's a lot of experimental clinical science. I mean, this is, some of it was done in Europe, but here DARPA to NIH, there was that transition, but you're still developing research, algorithms, and so on. And DARPA funded still the development of the limb, etc. So how the journey goes on, right? So, you know, I'll skip the details of how it's gone, but this was sort of second phase of things. Along that way, NIH played a really important role in this kind of demonstration of brain-machine interface, some of the first pioneering work at Pittsburgh, but there was other groups, and Brown and Stanford, others, who showed that through direct cortical implant, you can control a prosthetic hand. It's still slow motion, use your brain waves, go out and control something. It's still DARPA continue to play a role, but some pendulum started to shift to NIH. And so you record and, some brain machine and you control the prosthetic arm. So around that time, I started to realize that you are always controlling the processes, but what about sensing, right? Giving a feedback. And we had a bilateral amputee, this young man, George, who he came to our lab, and this prosthetic limb was assembled by Rahul and his company, Infinite Biomedical, with some of the touch bionic, bionic hands. And he can not only just control the hand, but also perceive sensation, but to deliver to the skin. So much so that you can feel even a painful sensation, like if you look at sharp objects, you can feel the sensation. See, again, the journey went from DARPA to NIH to some basic science. Some of it is neuromorphic encoding that we talked about last time. So touch sensors on the fingertip are neuromorphic. And this is still science, and some of it is robotics. Okay. Now moving further, Applied Physics Lab now continues to take that forward. I think Matt Pfeiffer is here, Francesco Tenere, all those folks funded, I think, presumably mostly DARPA, but lately NIH, worked with colleagues in neurology, Nathan Krohn and others. Now they've shown that you can do bilateral manipulation in real world or virtual reality. This man is, implanted with 2 probes, and this is intracortical. Now you can see that this is getting harder because now we are getting from non-implanted to implanted world. And while DARPA-style proof of concept, NIH-style decoding science is well and good, but how is this going to go into patient and first in man, and beyond that, it's going to go on and go further. So that is where I would now start saying Blueprint can play a role. So incidentally, the same group then went on, Luke Osborne, my former student, and against their team at APL. Sorry, the volume isn't coming, but he is now feeling sensation from this implanted probe. So it's not only is controlling, but is taking the sensory information back into the brain. So you see how the field has evolved from the non-invasive to implanted, from motor to sensory. We are still at a high-powered research phase funded by major agencies. So how do you kind of commercialize? And I'm going to borrow an old video from Rahul and Infinite Biomedical. I have to put up a disclaimer. I'm a co-founder. And here is an amputee who is operating the prosthetic hand as if like an activity of daily living. Far cry from that APL, that brain machine demo that you saw into real world. And I'm hoping that the next 2 talks will tell you how to bring it to real patient, real amputees world. Okay, so I went pretty quick to lay out the history and just to now start summarizing that this field of neuroprosthetics, this field of brain-machine interface, you can lay its foundation perhaps in one argument from DARPA. These yellow and green bubbles, you can't read it, but they are also invasive and non-invasive approaches. And it has taken all the way forward. We had this interesting conversation about quantum. And sure enough, like DARPA, one of the latest programs was, can you even decode single neuron activity non-invasively from the brain? So in its own way, DARPA has been a great supporter. But really, NIH has had to step in to do the science and the clinical translational work. It's founded on neuroscience, funded by NIH, and then translation more recently. That and the buzz and hype and everything and industry money and venture money has now spawned a lot of companies. You heard in the morning from Colonel, and there are Neuralink and a number of Synchron and all these major companies. There are a plethora of startups that have come up in BCIB. I mean, I don't know if we can even keep track of that, right? So this is, right now, it's the high, I should have brought in the hype curve, which is a very popular way to present how the fields take shape. And today, that brain-machine interface from very esoteric DARPA world and NIH science and clinical world is now into commercial activity. So, where does this future lie and where does Blueprint lie? So I want to kind of characterize in the context that left side, motor, sensory, restoration of, all of that is sort of science on which we are founded. And we need to take it to interfaces. This is that Blueprint program. The hard problem of building this interface, more likely than not implantable, to the brain, to the spinal cord, to the peripheral nerve. And you'll see this sprinkling of the project. Not all for brain-machine interface. Some of them are for neuromodulation, in fact, many. And that is where we reside in this Blueprint program. Henry just brought up that it's not about just take out the brain and all of that, but now we're in the restoration and even augmentation world, so that these neuromodulation, neural interfaces will perhaps restore function without having to take out something and augment function, which is a very interesting thing to talk about. So I am envisaging, building on my work, that I don't have time going to it, but I'll kind of pictorialize it, right? That if I were one of you, and my lab does some work, these were a couple of PhD students who did this work on saying, peripheral interface. Nerves, if an amputee loses a limb, nerves regenerate, and they regenerate very slowly, as you heard earlier in one of the presentations. And so how do you prove that these regenerated nerves can move limbs? So we did rodent study, a couple of PhD students, and you can see that we can make rats limb move. That is science stuff, right? Animal study. I have a proof of concept. Now, maybe I should take it further. If I were one of the Blueprint inventors, I would say I would generate intellectual property and develop a stronger proof of concept of this restoration ability. But remember, now we are talking about hard problem of interfacing to nerves and muscles. So I would take that bit further. And then we want to take it first in man, that fully interface system, that not a clinical trial, but take it to the first in man demonstration of this neural interface. And I think Paolo might present one of the projects out of MIT, which is probably moving along that line. So you can see that academics here in the room can go from R01 to some translational research, maybe through SBI program, to go into Blueprint program, and then first in man. So that's my general story, that Blueprint resides in this kind of ecosystem where DARPA, NIH, and now increasingly commercial investor venture companies are playing a role. The companies are investing, like this high-tech brain-machine interface company to real-world practical processes company like Infinite. Tap into R01 mechanism, if you like. Tap into these SBIR mechanisms so that you can get proof of concept or develop a prototype that can go into amputees. Come to the Blueprint program for that. You have that proof of concept or you have a prototype, but you need to de-risk it before it goes into the patient. You need to get regulatory path established. If you need to get your intellectual property is strongly established, maybe a proof of concept on large animal model, so that then you can go into a patient study. So that's my summary of, I just wanted to weave in from my journey to how you all can take part in this Blueprint program. And last but not the least, try to be advocate for all our agencies are doing who are under funding stress. So you as citizens, I think, morning talk was very eloquent about it, that we as citizens have to support NIH and all the agencies who are laying this groundwork for this journey. And that's all we can do to advocate to the Congress and our leadership in Washington that support these programs. So thank you for your time, and I'll hand over to Rahul.
Switch back to computers. Nitish, while we're switching computers, because that'll take a half hour. Can I ask you a question? You showed the federal funding mechanisms and the sequence. Do you want to mention about if we are not dependent on federal funding, since the government's in a shutdown right now?
It's terrific. Yeah. I hope we can have time more on the discussion, and Rahul and Paolo can also fill in. So of course, as you know, SBIR STTR mechanism is, in a sense, industry partnership. So first thing, we are coming out of academic bubble. But the second part of it is, even then, the industry is getting de-risked by the SBIR STTR mechanism. It isn't the other way around. And now the question is, from there, what's the gap, right? Who is filling the gap to the pure productization Medtronics and others will do? As far as I see at the moment, it's an era of venture capital into this business, as opposed to somehow an industry will put some few $100K, $1 million to get you started. And I think we are living in a heyday of investing in neuromodulation and neurotechnology through incredible venture investment going on. I think we need to draw more of them because Blueprint is doing a hard work of identifying top-tier translational stuff, and that would be my thought right now. But it's a tough topic.
Okay. I would add, though, that philanthropy is underestimated with a massive investment in the United States, maybe worldwide, but I certainly know in the United States, is made to academic centers to support research that's translational. In their foundations, there's so many in the neurotechnology area, there's so many foundations that are disease-oriented that really will pour money into helping develop new technologies and new treatments. So I wouldn't underestimate, because I think it's leads to frustration in young investigators. They think, well, if I don't get my R01 or SBIR or the Blueprint right away, then I can't go forward. But there are lots of ways, lots of alternatives that we need to take advantage of. Rahul, I'll turn it to you, and hopefully that you will further illuminate us.
Great. Thanks. So, Nidis, thanks for the intro as well, for all the work that I'm going to show today. I'm going to talk, go a little bit more broad, and then we'll narrow up a little bit. But first, I'm going to talk about, what this population and kind of looking at it from an investor's perspective. So I run IBT. It's a company that, like I said, it's spun out of Nidish's lab, but we're only focused on upper limb prosthetics, and we've been able to keep that mission-driven focus largely because the people of MIH that have continued to fund our work. But if you look at it from, let's say, a private investor, if you're thinking about this, we have a really small market, thankfully, in our world, in the US, about 50,000 people. And actually, I just learned that in Ukraine, unfortunately, they have even a larger population of upper limb loss individuals. So, just that we're thankful that we don't have to have those conflicts, but there are other places in the world where this is unfortunately a growing need. So we have a small population. And really, success is driven in this population by the number of hundreds. If you fit hundreds of people a year, you're considered a successful company. Then we have, on the other side, you have a really large technological barriers and regulatory barriers as well. You can imagine that you've got things like small motors that you're dealing with. You have machine learning in some aspects. You have hardware, software that you're developing, parts that touch the body, parts that may even go and get implanted as well. So a lot of complex technologies that you're potentially working with. But on the other side, of course, there are regulatory bodies, one being the FDA, where you have a class 2, potentially even class 3 devices that make it really challenging to build devices. So meaning that we need a significant amount of investment to take things to the market. So we got a small patient population, a large cost that's associated with bringing these to market. And then, of course, we've got the other side, the payment side, is that you have insurance companies, at least here in the US, that are not looking to spend a lot more money. And they're going to say that anything that comes out that's new is investigational, and you're going to be categorized as what we were initially, a miscellaneous device. So most major insurance providers are not going to pay for your device. So, how do you actually commercialize technology in the space? Because again, small, high dollar cost to get to market. And once you get to market, very few people want to pay for your technologies. So I'm going to take a look at the history of innovation in the upper limb space. And so I'm just going to jump back only about 110 years and look at, what the technology growth has looked like over the last 100 years or so. And so what we see in 1912, we have Dorrance along with Hosmer that came out with a split hook design. And really this body powered type of device was even invented 100 years before that. But the reason it became a mass market adopted product was because of World War I. We had a large population of people coming back from World War I. And we had now the Industrial Revolution where we could produce these at a large scale. So now we see the adoption of this body-powered technology that really kind of took off. Jumping ahead, if we go into the 1960s, we see the first myoelectric arm that emerges. But what preceded that? Well, we had World War II, which led a lot of people internationally, the Soviet Union, here in the United States, efforts led by the Department of Defense. They ended up funding various groups. Harvard, MIT was among those groups as well, to develop devices called myoelectric devices, which take advantage of muscle signals from the body to control robotic limbs. And so this was the first one in the US, came from Liberating Technologies, which was a spin out of an insurance company, believe it or not, that was built off technology that was at MIT. So that was really when we first seeing myoelectric devices emerge. Now, since then, I'd say since 1960 to about the early 2000s, there really wasn't much in terms of new technologies that were out there, in terms of ways to control, number of degrees of freedom that were operable, very small incremental changes. It wasn't until, as Nilesh kind of talked about this, DARPA, as a result of the wars in Iraq and Afghanistan, decided to invest in trying to bring about innovation in the space. And they spent on the order of $150 million across 3 different programs. And they really pioneered funding a large group of labs and a lot of thought leaders, Nilesh being one of them, several others that I'm sure you all are familiar with, including my lab, which I was doing a PhD from 2004 to 2009 at USC, and Jerry Loeb was the PI there. And so we were all part of this group that was working on revolutionizing prosthetic arms. And one of the groups was led by our friends at APL down the road here. And so we have a lot of now really great thought leaders thinking about this space, thinking about how do we make innovative products in this, or innovative research, and then eventually, hopefully, products for this market. So, and that led to, and at the same time, we had other technologies that were kind of converging. We had small motors, like I mentioned, that were now being able to put inside a palm of my hand. So now you could have multiple fingers moving simultaneously. You saw Nilesh's video as well. And so these were now emerging as commercial options. In 2007, ILM came out. That was the first one. Bebonic, 2010 thereafter, and Michelangelo. And then you had all this academic energy now spent towards, bringing about new innovation in this space. And we had, as the DARPA program started winding down, this is where a lot of effort from the NIH, NSF, DoD picked up that space, because you had PIs that were in these labs doing this great research, and they want to continue doing the work, moving it forward. And so this is where NIH, NSF, DoD kind of stepped up to kind of bridge that gap into the next era. When you have people like myself, who is interested in not necessarily academic work, but really taking technologies and bringing it to patients. And then similarly in other institutions, Colorado, they created a company called Point Designs, which makes mechanical fingers. At Rehabilitation Institute of Chicago, which is now Shirley Ryan Ability Lab, founded a company called CoApt, which made machine learning and take pattern recognition technologies to the market, and they launched a product in 2012. They both, ironically, just got acquired in the last year, so they've completed that cycle, I'd say, of like going from initial concept to commercialization, and now leading to a successful exit. And then here at Hopkins, taking on Nidish's pioneering work and trying to bring it to the market through our company, IBT. So I'm going to talk about our specific product at IBT and what did we do now that I've kind of given you a little bit of history of upper limb prosthetics. I'll talk a little bit more about what we did. So I met Jacob and Jacob Siegel, who's a PhD student in Richard Weir's lab. Richard was also funded through the DARPA program on various projects. And they had a concept they're developing called the postural control algorithm. And so the way that worked is you had multiple electrodes, and that's what these little arrows represent, going around this circle. And as each electrode contributed to the overall pattern of a movement, you had a vector that was drawn, which was a linear algebra summation of all these different individual vectors, and that vector could point to different parts of the circular map. And so that's what you see here. And that's what was originally funded through the VA with Dr. Weir and Siegel. And then we came into contact through a mutual friend and started working on our first NIH Fast Track grant in 2016, which we got funded. And then our job was to take what was a proof of concept in the lab that Jacob had done with maybe 2 or 3 patients and now bring it to the market. We had partnered with Hanger Clinic, which is the largest prosthetic clinical group in the country. And they were involved in the design of our technologies to make it better, make it clinically driven. So we were talking to them on a weekly, if not more frequently basis to make sure that we were building something that they liked. So we converted this into initial take-home prototype that you start seeing here. You can see the vectors as you point to different parts of the map and how that creates a translation in a movement. We've changed from a continuous space to a discrete space. You can see that here. That's what these wedges represent. And so in 2020, we sent the first patient home with the device, and they were able to use it. Interestingly enough, that patient is still using the system now, the commercialized version. So. And then thereafter was really what I consider the hard part. You've got, what we did was a proof of concept. Essentially, we built a one-off prototype. But as we heard Riki talk about earlier, there's a large amount of work that's being done from taking that initial proof of concept into something that's reproducible, that's manufacturable, converting these things into design inputs, those convert into design outputs, and so on and so forth. That eventually gets packaged into a group of documents that we eventually submit to the FDA for a Class 2 FDA approval, which we got in 2024. And then following that, we got a CE mark in January of this year, and then we also applied for a CMS code last year, which along with 3 other groups, and which got approved, and then we've got a code that covers this product now in the U.S. under Medicare. So this is my favorite part of the discussion, is kind of showing you what we've actually done with the technology. And so I'll show you 3 cases here of how we've been able to take this technology that originally started as a concept in the lab into the market. So this patient here was somebody that hadn't been able to use any prosthetic control other than one electrode to open and close his hand. He said he was very frustrated to use it. He would not use his prosthesis. And it was because he had a mixture between multiple channels that made it really difficult for him to separate out the products. And he had tried everything. His journey had been taking years, and he was very frustrated. And so I had met him through our group, the PMR group at Hopkins, and thought we would try to put this system on him. And we found that he quickly was able to use the system, and you can see how his signals we were able to separate out into these 2 different locations, and he was able to get consistent control. And it was really remarkable to see him use it, and you'll see a video of him using it now, where we even added more than just those 2 open and close movements. He's able to add rotation as well. And then he's able to move his hand in space and doesn't get inadvertent movements and things like that. And so this is someone who had struggled, like I said, for multiple years, and now is able to control 4 different movements consistently. The second case is somebody that recently came to us and was a young man who had lost his arm in an industrial accident. And we had actually started now working with the surgeons to start planning on how to place nerves into muscle fibers to be able to restore a more intuitive sensation. So his surgeon had actually gone in and placed the muscles for his thumb abduction and adduction in various parts of his forearm, so spatially separated them in consideration of our technology, which is really neat to see. So we're actually now going full circle working with the surgeon up front. And this patient, as you'll see here, he can now do he can, of course, shake somebody's hand with a power grasp, open and close. And you can see his smile there. This is 1 week post-delivery. And so he's getting that confidence to use the system. Here you can see him switching into a tripod grasp and opening up this bag of chips. Oh, sorry, key grasp here. And opening a bag of chips and is able to do that and pull that apart. And he's doing this, of course, there's a large team that's at Hughes Surgery, and Dr. Shaw was the surgeon that did this, and Altafried is his OT that we're working with him prior to even receiving his prosthesis. So this is a lot of people's work. I can't take any credit for it. And then on the last video you see here, he's switching and using his wallet and going from using that tripod grasp, pulling out his card, and then now being able to switch into the key or lateral grasp. And so again, this is a large team that's working with him to make sure that he's able to get those outcomes. The technology is only one part of it. Without the team that we have here, we wouldn't be able to see this type of result. So now he's being able to use his prosthesis in daily life. This last case, someone also a very special person who came to us who had a prior prosthesis, was very frustrated with the way that he had to recalibrate it quite a bit because he basically said it wouldn't listen to him. And so again, and this is someone that with a lot of our patients, what we see is that with traumatic incidents, there's a lot of depression, a lot of self-doubt, will I be able to get back to where I was, or and that frustration leads to device abandonment and further issues. So I think in this case, the team again that was around him, the same team as before, said, all right, well, let's try him with Glide and let's see what we can do. And what we saw is that the visual feedback that he was able to get from the system was really helpful for him because he could see, again, his previous complaint was the prosthesis is not listening to me. Well, now he could see what the prosthesis wants him to do or what he needs to do to get the prosthesis to do the different functions. And you can see that vector pointing, doing open and close. Here I was actually going and visiting another patient. He came in and volunteered to come in, and he offered to be a peer advisor to this other patient. So this is someone that's grown in his confidence and has become somebody that is willing to come in and shoot a video like this. Let me share it with you all. And it's been really great to work with people like this because now, he goes back and he sends me videos over the weekend of him doing lawn work. And so this is, why we get in the business of doing what we do. And so really, really just thankful to see that. And so, with him, like I said, it all came down to confidence. Was the prosthesis going to do what he wanted to do? And with that confidence, that gave him the ability to feel like, well, now he can do things that he can go back to his life and do things like working in the yard and things like that. Great. Well, that's all I've got for you.
That's all is an understatement. That is truly stunning, and we saved the best for last.
So I'm not sure it's going to be the best, but I'm going to switch gear the tiny bit here, and what I'm going to do is to give you just in a few slides a very, very short presentation that actually I prepare half of it, and half of it I ask some of the PIs that actually lead projects supported by our program to prepare, because what I would like to do is to emphasize how the, really I can only say because I didn't set it in place, Yusuf and Mike Dempsey, right, are responsible for it, and Bob and Susan, right, for the evaluations, the fabulous system that we set in place to select outstanding projects really led to the selection of projects that are at the forefront of the field. The way I'm going to show it is by showing some of the work that has been going on in the hospital, the rehab hospital where I work, right? And so what you see on the left side is the exoskeleton technology that we used about 15 years ago or so. Nothing wrong with that technology that came out of UC Berkeley, Caseroni's lab, right? There was, Spalding was part, as many of the other rehab centers that were mentioned in previous talks, right? And so we're part of a selected number of sites that actually evaluated this technology from a clinical point of view. But as you can see, bracing up to the hips, of course, for applications in which that is appropriate, right? But very cumbersome to use this technology, right? And so what we have seen gradually, and we have an example on the right side, is technology that is a lot less obtrusive and it's a lot more responsive to what the users would like to do. This is technology that came out 25 years ago out of the Human Augmentation Program at DARPA, and then it's still utilized, as you can see on the bottom right, for some military applications, but we have been extensively used it for clinical applications, for instance, both in stroke, as well as to unload the anatomy in individuals, for instance, with tendinopathies. For those of you who are not familiar with the technique that we're using musculoskeletal modeling to actually estimate internal forces. We're using OpenSim and MyoSweet to do that. When we have demonstrated that generating torques at the ankle with this technology, we can actually unload significantly the Achilles tendon. So why is this relevant to our program? It's relevant because one of the projects that we are supporting, right, that the program is supporting, right, is a program that kindly led by Zach Lerner and Ray Browning, right, and so we have a representative here of the company, right, and so have a slide that they have kindly provided, even if I asked just at the beginning of last week, so very much appreciated. But it is very much along this line. So it goes actually beyond purely easier to use, less obtrusive, really compact and focusing on a single joint. But what we're seeing in the field, as we were seeing in these projects and other projects that we have been considering for the program that we're discussing today, is that this system not only will be able to actually augment function as it was originally proposed by DARPA, right? And so, but they actually have the ability to facilitate the motor learning process that is associated, in fact, with the recovery. This specific group is focusing at the moment on facilitating improvements in motor functions in a pediatric population, but you can easily imagine that this could be utilized in adults. So the other two aspects of these type of technologies, so exoskeleton technologies, to either augment function or facilitate the restoration of function are represented in these slides. So one interesting area of development has been the one of looking into soft robotics. The pictures on the left side represent some of the technologies that have been utilized, that have been developed for industrial applications. What you see in the middle is technology that it just was easy for me to get on the slide simply because we have tested it at Spalding, and we have a collaboration and familiarity with Conor Walsh Group that has developed this type of technology, soft robotics, and then through a licensing agreement commercialized through Rewalk, right? But also to augment the capabilities that the soft robotic systems that are incredibly wearable, very comfortable, but complex to on and off, and limited in the amount of torque and bandwidth that they can deliver, some of the pioneer work that has been done in this area is to look into applications of functional electrical stimulations in this context. And you see in the middle of the picture, that's almost historical of a system that was developed a very long time. You saw the video on top of the Banerjee L300 system, which is broadly used in the clinic, and on the bottom you see a new company, Psionics, that have been working a bit in this area. So again, we have really a champion in this area, Lou Awad, at Boston University, working with Connor Walsh at Harvard Engineering, has been leading this project. I'm not, again, discussing this in a detailed manner, but Andrew DiMeo is here, who is mentoring and following them, so you can hear more about that. But you can see from the slide and the progress represented here in the animation that they have done really a fabulous job at taking essentially the problem of augmenting the amount of torque generated by these systems, not only to facilitate the restoration function through augmentation, but actually allowing through smart ways to deliver stimuli to muscles to control not only the plantar flexion, which is fundamental, and it's part of a great amount of work that this specific group has pioneered to improve ambulation in this patient population, but also controlling dorsiflexion, as it's done in the Bionet system that I showed just a minute ago, as well as inversion aversion of the foot. They can actually get into essentially developing a system that, again, not only provides augmentation, but also provides an opportunity for retraining motor functions in this population. In this specific instance, the group is primarily targeting stroke survivors. That is the case also in prosthetics, and both Nitish and Rahul really show you the marvelous work that has been done for upper limbs, but there is really analogous work that has been done for the lower limbs. And we're way past what we used to do about 20 years ago, which is study prosthetic knees, which is what you see on the left side. It's kind of historical, a publication that we published a little while back in the American Journal of Physical Medicine and Rehabilitation, in which we look into the comparison of a mechanically passive system, the MAUC, right, and so a C-leg, right, so an hydraulic system, and a Rio knee, that technology that was developed at MIT and eventually commercialized by OSU, that we characterized in my lab to show that we could actually get better stability during stance, and the leg was swinging more easily, right, to more complex systems that actually provide propulsion, which is what you see on the bottom right. It's now a system, again, developed at MIT as the Biome, eventually licensed to Autobach and commercialized as the Empower, right? And so, and there are new frontiers both there as well as in what is very critical from a clinical point of view, which is the interface with the body. You know, how do we make it comfortable? How do we avoid issues with the residuum health, right? And so oftentimes a major issue in these vascular amputees. And it's the figure that you see on the right side, right? This is a system, again, developed by MIT that we're studying extensively and now integrated actually in the clinic at Spalding, in which we do a scan, essentially, the CT scan of the residuum. We match the impedance of the residuum by designing a lanyard socket that essentially distribute the pressure in a way that's optimal as to preserve essentially as much as possible the health of the residuum. So this is what is happening in terms of trajectory. But what's next? Next, even in lower limbs, is to be sure that the subject has the level of control that Nitish and Rahul actually demonstrated for the upper limbs, right? And so they could respond to a perturbation with actuations, right? And this is a long journey really led by Juher at MIT. And for this specific project, they actually spun off a company that Rick Kastler is leading. And it does take advantage of the equivalent to what Nitish brought up to our attention earlier, which is targeted reinnervation, is a pseudo, I would say, equivalent of that, which is what you see on the left side, which which is the surgical technique developed by McCarthy at Brigham and Women's Hospital that actually connects agonist and antagonist muscles. And in a recent paper, it has been shown how that actually improves per se, so the control of a prosthesis. But could we do that with something that is stable, right? It's mechanically stable, and it's very muscle specific. And so a bit of, in my opinion, originally, a bit of a crazy idea, right? And so that this group had, and in parallel, there is a group led by Christian Cipriani in Europe that's looking into it for the upper limbs, right? Is to actually inject small magnetic beads into muscles and then actually monitor externally, essentially with magnetometers, right? And so the equivalent of an array of magnetometers. What is actually happening mechanically inside the body, and then control the prosthesis accordingly, right? This is absolutely at the forefront of the field, right? And so besides augmenting functions, right, when we want to restore function, and this is the last project that I want to briefly mention before Henry perhaps brings us back to a short discussion if time allows, right? And so besides the restoration with technology that I mentioned, the improved control and the capability of essentially relearning, right? And so in those patients that are not responsive to the interventions that I mentioned earlier, the field has had a humongous attention on neuromodulation techniques, right? And so traditionally that means priming by using TMS, or facilitating essentially activity in cortical area through TDCS, right? Essentially depolarizing members, membranes, and so on and so forth. You see in the middle a picture of some of our experiments in the lab using a combination of robotics and interactive gaming facilitated by data gloves, essentially. And then recently, that's what you see on the right side, a Lancet paper by a colleague at MGH, led by a colleague at MGH, but involving a lot of other centers, is the stimulation of the vagus nerve. And that's because through, about cholinergic and monomenergic pathways, there is evidence that you actually are facilitating essentially the production of BDNF, right? And the results are indeed promising. This is a technique that we really like, and gradually we're getting into clinical adoption. But if you look into their license paper, the motor gains are still limited on an average basis. So if you look at the group results, for those of you who are familiar with rehabilitation, on a Fugger-Meyer scale, you get about 3 points of difference between your control group and the intervention group. And for those of you who are now familiar with rehabilitation, that's a scale that has as a minimum clinically important difference 5 points. So the difference between the groups on average is actually less than the MCID. Right? But on an individual basis, the results are extremely good. So there are 2 lines of work, one which is toward assessing what is the right phenotype to actually use this type of stimulation. And the other line of work is to say, what if we increase massively dosage? And that's what Batel is doing. And Dave Friedenberg was kind enough to actually put together this slide for us so that we could actually highlight this project. And the idea is complex in its implementation and then relatively simple in conceptually, right? And so it's a fabulous platform, and it basically says, what if we could address one of the major shortcomings of the use of the technology so far? And the shortcoming is the fact that in the clinical trial that I just mentioned, to make, in fact, the stimulation effective, the stimulation has to be synchronized with the performance of movement. So the way it was done in that trial, right, it was basically by having a therapist observing the patient and pushing a button every time the patient was going through a repetition, right, which is, of course, limits really very much the applicability of the technique, and certainly does not allow to expand upon that with massive motor practice. So the concept here is, as I said, relatively simple with actually a fairly complex implementation, is to say, can we actually detect through sensing technology embedded in a sleeve that goes around the arm, the intention and then the performance of movements in a way similar to what the therapist was actually doing during the clinical trial. And can we do that and therefore deliver these stimuli automatically in the field, right? And so, and the pilot results that you see on the bottom left are a clear indication of a significant motor gains that are associated with that. So these are the 4 examples that I wanted to provide. And again, I hope I was convincing that the program has been good enough, thanks to fabulous system put together by Mike, Yusuf, Bob, and Susan, to actually select projects that are really at the forefront of the field. They're not purely a good investment that so any investors group could actually pick, right? But there is quite a significant level of risk. And of course, you know, we have been talking around during the day about de-risking these projects, but I feel like as a researcher that this is quite a unique program, and the quality of the projects that have been supported, I think it's a testimony of that. Thank you very much.